Yo, welcome back, guys. This is me, Totality. I know it's been a long time. 1 minute, let me just get hit by a textbook. I don't know why I forgot to take that. Let me change the scene. Here we are. Hi, man. My hair has grown a lot. I did not notice that. Oops, oops. What happened here? So, yes, I'm finally back, and here we are doing our job. That is to literally study on stream. And if you don't know what I've been doing the last week, I guess, because I didn't stream for 7 days, but I was continuing my challenge through those days, and it was a bit difficult because we had Eid too. But that's not the reason though, because I wanted to study a bit. I wanted to actually feel more, private. Sometimes whenever I go live, I don't know why I have this uncomfortable feeling. I don't know why. But yes, since I have some experience now, so it's not that much, but studying I wanted to study like off streaming, so just to see like how safe I feel with this, and here we are finally. So I just wanted to mention you one thing about this is that we have been I have it's written as recap of day 42 to day 48 what I've been doing with those days. So I have completed some like some subjects actually, like biology is completed, chemistry is completed, English half is completed. Then maths I'm progressing it actually. I still didn't complete the full thing. Then physics the only thing I have to start out. So that's a problem here. But it's fine because we have done a lot of progress, and it was a bit time taking, but I wanted to study each subject thoroughly, and I feel like I have done it. It's the maths only which will take some time because during exam days I'll be able to like go for it nicely and take it out on the exams itself, like cracking the exams. Anyways, let's just move on. I didn't notice that it was muted. My bad. I mean, I just muted on purpose. I don't know why. But okay. Oh yeah, I forgot to take my control panel. So as you can see, okay. Wait, I did not mention this. Oh shoot. Let me. I forgot to mention the day number itself. Let me like edit the title on on stream. I'll edit the title right now. I know it's taking some time. Let's just edit the title. Edit the title. I'm gonna edit the title. I forgot to put the day number. It's day 49. Save. And if I run it back, yeah, it's day 49. Let's move on to this. Alright, so right on the bat, nobody's here, that's fine. It's data is excellent, that's great. Then yeah, let's start. You know what? Let's just start. So what I'm gonna give you a small context what's going on. Tomorrow is my biology exam, so we're gonna fully revise biology all again, even though I've completed it. Because revision is one important key here to actually like get good marks. So I'm gonna revise the whole biology with each and different steps, and we'll also try to attempt to complete some maths. We have some maths work to do too. Like it's gonna be a bit time taking, but we're gonna do it. So this is what I'm gonna do actually. Like first of all, let me just take this out. I don't know why it was there. Yeah, as I was saying, we'll have only 5 hours left here for the work, and then we'll progress on. In those 5 years, we'll complete our full uh, textbook reading. Sorry, not textbook reading. The notes completion. Then we'll complete the full part of drawing diagrams. I have to draw the diagrams, and some textbook questions, because textbook questions are really important. And phone is gonna play a main role in this, so I'm gonna use my phone in this. Mm, let's try. The problem is I have to like take this challenge out then. WhatsApp is so important sometimes, man. I really block it, but then I have to unblock it again and again, because it's something like you don't want to waste your time on, right? So yeah, you will be like, yeah, you should be not wasting time, but again, that's the thing, like it takes up a lot of time, like it is still important though. It's not like it's not important at all, but then the fact that it is really really important. Okay, wait. So wait, well, where should I start with? We'll start with molecular basis of inheritance. Till then, I've made my own notes based on this, so this is a bit advantage thingy. So let me just keep my phone here. How much charge? 94% is good. If I need the charge, I will bring my charger. That's it. And I don't know where's my pouch. I cannot find it. I don't know what happened to it, but I do have my pen, pencil, and scale and eraser. All the requirements actually, to be honest. But yes, let's just move on. So here we are. Let's complete this. Okay, let's start. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. I recited that really fast. I should be doing that, but okay. 7:37. We'll be completing at 12:37. Let me keep the timer. Of let's say 1 hour each, and this no, let's say 3 hours. In 3 hours, in the span of that, or let's say 2 hours, are capable. We should be capable of completing full notes and all, because we shouldn't be giving much time to each thing. So where is it? Come on, tools, scripts. This one. Give me 120 minutes and then timer finished. Yes, close. So if I open this up, this is 2 hours. Nice. So 3, 2, 1, go. Let's start. Biology. Mendel's time traits were according to Mendel, the traits were passed from parent to progeny, which is called as inheritance. This is because of the factors which is today known as genes, which wasn't explained well. But however, after the 100 years that has passed by, we get to know that DNA and RNA is a genetic material, and these DNA and RNA are the nucleic acids, which are polymers of nucleotides. Now, nucleotides, by the sense of it contains N bases, that is nitrogenous bases, adenine, guanine, cytosine, and thymine, and then sugar, that is pentose, which is basically deoxyribose sugar, and then we got phosphate group, which is PO43 minus. Then genetic material of virus is basically RNA, because most of them contain RNA, and these types of viruses are known as retrovirus, and they are mostly functional by mRNA. So that's one thing. So genetic material of virus is mostly functioned by mRNA. Nucleic acids are polymers of nucleotides. Then DNA and RNA. So deoxyribonucleic acid and ribonucleic acids both are genetic materials, and yeah. DNA is made from deoxyribose sugar, RNA is made from ribose sugar. Central dogma. Now, this is something I don't know why it's given in the starting of the textbook, but we'll have to go through that too. Wait, again, and this is more recap. So, DNA, RNA, genetic material. Yo, Gabe, owners, how are you doing, bro? Hi, and we are actually doing a small recap. I'm actually revising the chapter of molecular basis of inheritance. I have made my own notes, so I'm still going through that. So basically, we need to know that nucleic acids like DNA and RNA, those nucleic acids are basically, which are termed as nucleotides, are known as nitrogenous base sugar phosphate group. Of course, that's the thing. And the genetic material of virus is mostly mRNA, and there are three types of RNA: mRNA, tRNA, and rRNA. mRNA, messenger RNA. This one is mainly the genetic material. tRNA, I think I mentioned that earlier. I think it's in the textbook. Okay, I'll check that later. tRNA should be the structural and catalyst thingy. I'm not sure. Okay, then take care. Well, study well. Bye. Yes, have fun, bro. Bye, bye. And then three. What else is there? rRNA. Like, you know what? Let's just go through that because it's not there in my notes. It's there in the textbook though, so we'll go through that. It's important. Where is it? Okay. Hmm. It's given here. Okay, so tRNA is adapter, mRNA is the main genetic material, RNA is structural catalyst. My bad. Structural catalyst, bro. I had no idea about that one. Sorry, my bad. Because I usually study from those notes. Okay, so okay, it's given here. RNA is genetic material, tRNA is the adapter, and rRNA is the genetic material. Sorry, not genetic. Structural catalyst. Then what? Yeah. So DNA undergoes replication, and the process of conversion of DNA to RNA is called transcription, and the making of the proteins from RNA is known as uh, translation. Yes. Then we got the deoxyribose sugar. What I need to know so far is that there is a OH and no, not OH. So again, the usage of textbook. Let's use that to check this out. Uh, the double helical structure of DNA. We'll check this out too. Um, here right now. Okay, so here it is. So this is ribose and this is deoxyribose sugar. So according to me here, when there are multiple connections, they are actually showing OH group here and no OH here. So you're just gonna draw a line from here and CHH. You know what? We'll just mention that this is CHH. So it's gonna be like first, second, third, fourth, fifth carbon, and we're gonna draw that structure when once we are like doing drawing of the diagram. But however, it'll have a great look up here. So overall, what we need to know is that this is the first prime carbon, second prime carbon, third prime carbon, and fourth prime carbon. This is the fifth one, which is connected to the both hydrogens and one phosphate group, and this is gonna form the 5 prime end, and this one is basically called as phosphoester bond. um, However, if the let's say there we take a nucleotide in between the strand, we get to know that the carbon which is attached to phosphate is also attached to another carbon of the other nucleotide. So this type of bond, overall bond that is formed, is called phosphodiester bond. And then what else? 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 We have wait the yeah, N-glycosidic bonds. The N-glycosidic bonds, I believe that these are the ones that are attached to bases. So yeah, the nitrogenous bases, the bonds that are attached to nitrogenous bases with the first prime carbon is known as N-glycosidic bond. Didn't I mention this? Okay, I should be mentioning it here. I don't know why I did not mention that. That's something which is very weird. And yeah, I did not mention it. Let me just mention it here. So this is what for phospho. Bro, what is wrong with the pen? Yo, what? Yo, what? Why is it not working? Yo, what? I swear the pen was working earlier. Nah, come on, stop scamming, bro. Really? How much force do I need to apply to make this work? Why is it stuck? No, it's not working. No, what? It's not working, guys. I don't know what happened. Okay, never mind. Let's just use the pencil. Okay, so this is N-glycosidic bond. Sorry, wait, no. This is N-glycosidic, and this is phosphodiester bond. Where's phosphodiester bond? Okay, I don't know why I didn't mention these, but okay. DNA is more stable and as it is an abundant genetic material. DNA is very stable. RNA is highly unstable. DNA is made of deoxyribonucleic, which is consisting of sugar, phosphate, and base. Deoxyribose sugar, phosphate, and the nitrogenous bases of adenine, guanine, cytosine, and thymine. But for RNA, it's not going to be thymine. It's going to be so for N bases, we have purines, which are basically adenine and guanine. But for pyrimidines, it is uh, adenine. Sorry, not adenine. It's cytosine and then thymine and uracil. Then we got the N base plus sugar. If we attach the N base with the sugar, that is the pentose sugar, we'll get a nucleoside. Now this nucleoside is like is the one that we studied. Okay, in biomolecules also, that is deoxy, or let's just say adenosine, guanosine, cytidine, thymidine, uracidine, and we can also say adenosine, deoxyadenosine, because it can be it's an N base pair which can be found in DNAs. If it is in the case of RNA, then we'll not mention that D. That's it. But uracil is confirmed that it is in RNA, so we are not going to put D there. And thymine will have their D always because deoxy ribose nucleic acid. DNA is the one that only has this thymine, but not uracil. That's one thing. So uracidin, the 5 prime end of the DNA is the phosphate group, and the 3 prime end of the nucleotide is the OH group. That's one thing. Two nucleotides linked to form a dinucleotide. More than two nucleotides are polynucleotide. Okay, the PO4 3 minus is connected to the 5 prime carbon due to the phosphoester bond, and when the PO4 3 minus is attached to the other nucleotide with the help of the that is to attach to the uh, you know let's say 1 minute, let me just check. So if it is this 5 prime carbon, should be attached to the 3 prime carbon, all right, should be attached 1 2 3. So yeah, it will be attached to the 3 prime carbon of the other nucleotide, which is basically the one reason why we are getting a phosphodiester bond. As the overall forms the overall phosphodiester bond. The N bases are connected to nucleotides by the N glycosidic bond. The length of the DNA is measured through the number of base pairs seen in the DNA. So the length of the DNA is measured through the number of base pairs in the DNA. And then one thing about the N bases is that there are two types: purines and pyrimidines. Adenine, guanine, both are purines. Pyrimidines is cytosine. Why am I saying that? Cytosine. Then the thymine, and then we got uracil. But if we talk about their nucleosides, it will be D-adenosine, D-cytosine, D-cytidine, D-guanosine, D-thymidine, and uracidin. That's it. No D is there. So yeah, the length of the DNA is the number of base pairs seen, and in the 5 prime carbon, the PO4 3 minus is present, which is attached to the 5 prime carbon, of course, uh, forming the 5 prime end. Uh, but that is true. The bond is that is attached. The bond formed when they are both attached is phosphoester bond, and there is another overall bond that they would be attached with, where the phosphate would bond with the 3rd prime carbon of the other nucleotide, forming the overall bond known as phosphodiester bond. This phosphodiester bond is one such characteristic. Phosphoester bond is also a bit different from phosphodiester bond because it just shows a specific bond of that phosphate and that nucleotide's 5 prime carbon. That's one idea you can get from here. And then another thing about this is that what did we study about it? Okay, 5 prime and yeah, number of base pairs seen. Then we will check the this one 5174. So 1 4 5 1 2 5 3 8 6 533 5386 base pairs and 5174 bacteriophage base pairs, and E. coli has 4.6 into 10 raised to 5, no 6. Oh, it's 10 raised to 6, my bad. I have to remember that. This is 3.3 into 10 raised to 10 raised to 9. This is 6.6 into 10 raised to 9. This one should be 48,502. This is base pairs. Okay, these are in terms of base pairs, and that's interesting though. That's good. Now nucleotides. Nucleotides. Okay, this is an extra topic that I have added. If the nucleosides are attached with the phosphate group, we get the nucleotides. These are known as D-adenylic acid, D-cytidyllic acid. Then we got D-thymidyllic acid, D-guanylic acid, and uracetylic. I think it's uracetylic. Is that uracetylic acid? Yeah. Then the backbone of polynucleotide chain. Okay, we are coming to another part. Interesting. This is gonna take a bit time, but no problem. Frederick Griffith's experiment is also there, right? So yeah, we have to do it fast, guys. We are not having enough time. Let's go. So backbone of the polynucleotide chain. This backbone will be basically these phosphate and the for the phosphate group and the pentose sugar together. These form the backbone of the DNA as they are the strand as they're forming the strands, whereas the nitrogenous base are towards the inward direction, forming hydrogen bonds with each other, and then which are called as base pairs. And then also one thing about this is that we have studied the number of base pairs. 5174 bacteriophage has 5386 base pairs, and then the lambda bacteriophage has the 48,502 base pairs. E. coli has 4.6 into 10 raised to 6 base pairs. An unhaploid human cell, that is gametes, basically mainly gametes, those have like 3.3 into 10 raised to 9 base pairs. Then for a diploid cell of a human, it will be 6.6 into 10 raised to 9 base pairs. Now that's one thing. Then wait, who coined the term nucleic acid for DNA? Let's just search that up. Let me just open that, and then who coined the term? Who coined the term of nucleic acid? Nucleic acid for DNA. What's this? Yeah. Okay, get off. Okay. DNA, not DNQA. Richard Altman. Let me write that down. For nucleic acids, because we know that DNA was termed as nuclein by Frederick Mischer in 1869, but then I wanted to know the name of the other person who named it as nucleic acid. Alfred. No, it's Richard. Richard Altman. Richard Altman. Let's go. Identified by Frederick Mischer, 1869. Okay, that's a good weird name. But okay. Described DNA as nuclein. Due to technical limitations of long polymer intact of DNA, it was really difficult for people to actually like prove the structure of DNA for a long amount of time. Elucidation, the explanation of DNA structure take time, and the N bases are connected to the moiety group from backbone. Okay, this one, sugar moiety group. The N bases are connected to the moiety sugar group, which is forming the backbone of the DNA along with the phosphate group. Um, okay, wait, 1869, Frederick Mischer. Okay, what was the year though? Okay, 1889. So it gives 1889. Can you guys see the screen? Like, oh, okay, wait a minute. Let me just decrease that. Here we go. 1889. Not much of a decrease though, but it's fine. 1889. Just after 20 years after Frederick Mischer, okay, let me just check this spelling too. Meisher. Okay, Meisher. Let's go W. Then, okay. So Friedrich Mischer, who in 1869 discovered not coined the term nuclein for the DNA, and then Richard Altman coined the term nucleic acids for DNA in 1889, which is not there in the textbook. It's a new, it's like an outside knowledge. But then, due to technical limitations of the long polymer intact of DNA, it took a lot of time for people to for people to elucidate the structure of DNA, or let's say just elucidate the DNA itself. And then, yeah, we of course know that the N bases are connected to the sugar moiety group, which means that they form long polymers, and that's the backbone along with the phosphate group. In 1953, the proposal of double helix structure was given by whom? It was James Watson and Francis Crick. Both of them used the help of X-ray diffraction, which was done by the uh, Rosalind Franklin, and who was that person? Morris Wilkins. Okay, due to X-ray diffraction data, it's X-ray diffraction data, Morris Wilkins and Rosalind Franklin. Then we got Arvind Chagraph. Out of nowhere, he just said that the ratio of adenine and thymine and guanine and cytosine are constant, and their ratio gives 1. So that's what he said here, but I don't know why he said like that. But that was a really random guess, though, if I'm being honest. Like, I don't know if that was random or not, but that looks random, bro. Like, out of nowhere, it's like, yeah, A plus D by C plus G is 1. That is adenine plus thymine divided by cytosine plus guanine is equal to 1. Like, what? But yeah, there are some exceptions to this. That's what I've noticed. The sequence of base pairs of a strand would be complement to the sequence of base pair of another strand of for the same genetic material, which means that we can predict the base pairs. This is because specific base pairs like adenine would only have to bond with thymine in case of DNA, that is only two hydrogen bonds, and it can also bond with uracil in the case of RNA. And then guanine will only bond with cytosine, with the that is to the help of three hydrogen bonds they make. That is one bond. That's the bonding they have. Then base pairs are the unique properties to the polynucleotide chain. Okay, that's one thing. Um, Yes, so the sequence of the base pairs of of one strand is complement to the base sequence of the base pairs of another strand. That's one idea we get. So parental DNA should be identical to daughter DNA. Okay, this one is a bit one thing I've noticed in textbook. Uh, let's just go through that. So base pairs confers a unique property to polynucleotide chains, and then this one. Then we have the salient features, and then central dogma. Okay, so the parental DNA would be identical to daughter DNA. If strands of parental DNA acts as a template for the synthesis of a new strand. This is a future topic, by the way. I think they won't ask this for the exam, but I'll feel like, let's go through it. So what we do need to know is that the parental DNA can undergo the process of replication through the help of central dogma process, the whole thing, where the parental DNA will act as the template, and the other strands are formed, forming the overall daughter DNA, which are going to be the new DNAs being formed. However, what we get to know is that these daughter DNA that the new strands are produced will be identical to the parental DNA. This is because of the sequence of the base pairs being complement to the other sequence of base pairs. So that's one thing I've noticed, and that's given in the textbook too, right? Yeah, it's given here. Like, I literally tried to write down every point of textbook here in this book. Then salient features of DNA. I learned through the mnemonic of bands. What is band? Backbone. The first point is about backbone. So we can say that DNA is a double-stranded structure where the backbone of each strand consists of a sugar and a phosphate group, whereas the and nitrogenous base pairs are uh, towards each other, forming hydrogen bonds with each other. Then that is the overall base pairs being formed. Then the second thing is anti-parallel. The strands of the DNA are anti-parallel to each other. If one strand ends with 5 prime to 3 prime, then the other strand will end from 3 prime to 5 prime. Then we got nitrogenous base and bands. Okay, I have to remember this mnemonic. It's a nitrogenous base. We're gonna say that the nitrogenous bases like adenine will bond with the thymine to form two hydrogen. Sorry, they will undergo. So they will actually form a bond that is two hydrogen bonds, and the guanine will only bond with cytosine, and three hydrogen bonds will be there between. And if you are talking about the case of DNA, adenine will only bind with the thymine, not with uracil here. Then the sequences of N. Okay, wait, that's already again. Oh yeah, okay. The sequences of N bases are complementary to the sequences of the another base pairs of of the other strand. Then D, distance. Okay, so this one is basically showing that the pitch of the DNA is 3.4 nanometers, and 0.34 nanometers is, or let's say 3.4 angstroms, if I'm correct. Is it? Yeah, the distance between the two base pairs is 0.34 nanometers. Let me just check. Yeah, 3.4 angstroms is the distance between the both the base pairs, and what else is given in that? I don't think so there's much given here. Yeah, and then the last one is stability. So I feel like this is a really important question. So if they ask for this for 5 mark, we should be able to answer this easily. Stability of DNA because base pairs stacks over each other, and in addition of the hydrogen bonds. Uh, okay, so I think that's salient features of DNA. I have to go through this again. So bands, backbone, the what was that? Okay, I don't know what was there in the screen. I just saw something, but okay, never mind. Bands. Okay, so backbone. In the backbone, we have the DNA is a double helical structure where both these strands consists of the backbone. That backbone consists of the sugar, deoxyribose sugar, which is bonded with the phosphate group, and the nitrogenous bases bonded with the first carbons are towards each other, such that they form hydrogen bonds. Okay. Forming the overall base pairs. We can also mention that. That's some extra points. Then extra point. Oh, okay, wait, what else? What was I talking about? Then yeah, anti-parallel bands, anti-parallel, right? So anti-parallel in this case, we can say that the object. Okay, what am I saying? Um, my bad. Let me just switch on to my screen, focusing. Okay, just kind of, let me just take this out. Okay, so anti-parallel here, in this anti-parallel case, what we need to mention here is that in the anti-parallel case, the DNA, two strands of the DNA are anti-parallel to each other, such that such that one strand might have their ends from 5 prime to 3 prime, and the other, so that the other strand will also have the ending from 3 prime to 5 prime. So that's one thing about the ends that we can talk about anti-parallel. Then the end base, that the sequence of base pairs found in of the end base are the sequences of the nitrogen bases of one strand is complement to the other, or due because of the hydrogen bonding formation. And then we can also say that the ow, is it painful? Okay, nose is paining. Why is my nose paining? What am I saying? Why is my grammar not gramming? Okay, what grammar, right? That's what I'm saying. Oh shoot. Oh, okay. So then, the adenine will only bind with thymine with the help of two hydrogen bonds, and that is only for the case of DNA. And then the guanine will only bind with cytosine with the help of three hydrogen bonds. That's one idea we get from this. Then the overall thing we can talk about is distance. So the pitch of the DNA is 3.4 nanometers, and the distance between each base pair of the DNA would be overall 0.34 nanometers or 3.4 angstroms. Then the stability of the DNA is high. This is because of the base pairs being stacked over each other with in addition with the hydrogen bonds. Let's go. We know the you know what? We know the salient features of DNA now. That is great. Okay, so let me just go through. The polarity of other strand is 3 prime to 5 prime. Okay, I should be mentioning it as polarity, my bad. Not ends. Polarity. Okay, that's one idea I got. Then the proposition of the structure of DNA and the simplicity explanation. the proposition of the structure of DNA and explanation of it was revolutionary. So again, 1969, Frederick Misher was the one who actually termed it as nuclein, and 1989, sorry, 1889, not 1869, 1889. Then another person came whose name is Richard Altman, who was able to like coin the term of DNA as nucleic acid, as he observed the acidic nature of it. Then uh, nuclear, yeah. Okay, in 1953, they were able to like find out the structure of the DNA, which was proposed by Francis Crick and James Watson. Sorry, is it correct? Is it James Watson? Okay, correct. James Watson and Francis Crick, and then what else? 1953, yeah, that's correct year. This is due to the diffraction, X-ray diffraction data that was given by Rosalind Franklin and Maurice Wilkins. Okay, that one was true. Okay, now we got the central dogma. Here, the DNA would undergo replication, and the making of RNA from DNA is called transcription, and the making of proteins from RNA is known as translation. And if we want to talk about the process, if we want to talk about uh, the process in which the retrovirus would undergo through, it would be called as reverse transcription with the help of the enzyme reverse transcriptase. I don't know why I didn't write here. Reverse transcriptase. Reverse transcriptase. Let me write it down here, and that is gonna be written as the enzyme. So this is the enzyme involved, reverse transcriptase. Now, this is only for the case of retrovirus. Okay, the proposition of the structure of DNA was revolutionary, and yeah, proposition of the structure of DNA and explanation, blah blah blah. That's one thing. In retroviruses, where RNA content is the only genetic material present in them, where they will undergo the passing of information from RNA to DNA through the process of reverse transcription by an enzyme called reverse transcriptase. Okay, it's mentioned here, but it's in the form of points, so it won't be that directive. That's one thing. Okay, so length of DNA. How are we going to find out the length of a DNA? So we'll just say that length of DNA is equal to number of base pairs into the the length distance between two base pairs. So that is 0.34 nanometers into let's say how much. If you're taking a human cell diploid, it will be 6.6 into 10 raised to 9. Then once we actually like calculate this overall, we get 6.6 into 0.34 into yeah, that's it, and then we get 2.2 meters. Imagine 2.2 meters of DNA, which is packed in a micrometer length of a nucleus. That's the length of the nucleus, if anyone's wondering. And here we need to know that the packaging is very crucial. So packaging is one topic for me which takes time to learn. And then we have the Griffiths experiment and the biochemical characterization. Okay, we are doing great, guys. It will take time. So okay, let me just read this through because it will take time for me to understand. So the DNA helix, which is a negatively charged nucleic acid, okay, are attached to the positively charged proteins. They don't have a defined nucleus, so DNA isn't scattered and for large loops and form large loops, yeah, that, and together they form the nucleoid. That's one thing. Then in eukaryotes, which we call it as the histones for these proteins, the DNA, negatively charged DNA, would be binding with the or folds upon the positively charged proteins, which are basic in nature, called histones, and they depend on the abundance of amino acids like residues like lysine and arginine. Okay, lysine and arginine, histones rich in amino acid residues of, yeah, okay, what, mentioned it twice. Let me read this topic and then I will explain it by myself because this topic is a bit weak. So, the unit of 8 molecules of histone from the form is called the histone octamer, which is positively charged, and the DNA wraps around it, around the histone octamer to form the nucleosome. Okay, that's one thing I've noticed. Okay, you know what? Let's just say nucleosome. Then H1 histone is also there around the nucleosome. One thing about this is that the repeating unit of nucleosome is called chromatin. Okay, that's one thing I should know. Repeating unit of nucleosome is called a chromatin. Then yeah, forming beads on. Okay, wait. Nucleosome contains 200 base pairs. I think if I missed anything, any point, I'll just check. just in case if I missed any point. Histones are rich. Yeah, basic amino acids resides like okay, blah 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 blah. Blah 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 blah. Blah. Okay, what am I doing? Okay. Yeah, okay. So the repeating unit of the nucleosome is called chromatin, and then these are forming beads on string structure seen in electron microscopy (EM). The beads on string structure in chromatin is packaged to form the chromatin. Is packaged to form the chromatin fiber. Okay, higher level of packaging requires non-histone complex, sorry, chromosomal proteins. What am I saying, bro? Okay, okay, yeah, this one. Yeah, this one. In typical nucleus of the cell, regions of chromatins are loosely packed and hence they acquire less stain. This region of chromatin is called euchromatin, and whereas there is another region of the chromatin material which would be densely packed and would densely have a dense stain, and this is known as the heterochromatin. The euchromatin is like transcriptionally active, but heterochromatin is not transcriptionally active. That's one thing. Let me take the charger out. And then search for genetic material. 1926, the quest to determine the mechanism of genetic inheritance at molecular level. Okay. Okay, so let me just go through this. I know we are taking a lot of time for this chapter only, but I think it's worth it because this chapter is pretty much new for me too. Even though I've revised it, I still need to go through it nicely so that I don't forget. Anyways, let's move on. Let's move on. So, as I was saying, that the DNA packaging is the packaging of the genetic material is really crucial. In the prokaryotes, what happens is that the E. coli would actually have the DNA packaged with DNA, which is uh, negatively charged. DNA helix will bind with the positively charged proteins, and these together forms a loop called a nucleoid. The DNA doesn't scatter in this case, and yeah, that's it about RNA. However, in DNA, this is a bit different. What happens is that the DNA helix structure, which is already negatively charged, will bind with the positively charged base in the base state of proteins called positively charged base state of proteins called histones. These are dependent on the abundance of amino acids residues like arginine. They're also like let's say arginine and lysine. Then yes, histones rich in amino acid residues of arginine and lysine, and then the unit of 8 molecules on the histone octamer. This histone octamer gets wrapped around by the help of, is wrapped around by DNA helix, which is negatively charged. 200 base pairs of DNA can be seen in one nucleosome, because together the histone octamer and the DNA structure when they are wrapped will form the nucleosome. Around it is also there as H1 histone, which is mentioned in the textbook, and I don't know what's the use of that. It's not mentioned in the textbook. It just says that there is H1 histone. Anyways, what we need to know is that the repeating units of these nucleosomes will form the chromatin material. This chromatin material will is also known as the beads on strength structure. The beads on strength structure that we see, okay, what is mentioned here, is seen in electron microscopy (EM). The yeah, the beads and string structure of the chromatin material is packaged to form which is the chromatin fiber, to form the chromatin fiber. Higher level of the packaging is done by non-histonal chromatin chromosome material. What am I saying? Non-histonal chromosomal proteins, which are seen for higher levels of packaging of the DNA. And then not only that, there is another thing in the by observing the nucleus where of the chromatin material, some places are densely packed, which get a dense stain. When a stain is put on them, they get a dense stain, and that is known as a heterochromatin. In some other regions of the chromatin material, they are loosely packed, and thus they get less stain. This part is known as the No, euchromatin. The euchromatin is transcriptionally active, and heterochromatin is not transitionally active. Okay, the search for genetic material. Now, this is where we start about Griffith's experiment. This one is not important. It's like just the previous things. So yeah, we're so close in completing this part. In 1926, the search for genetic material has or the mechanism of genetic inheritance at molecular level was important. Took time to prove that DNA is genetic material. What molecule is actually the genetic material? Uh, Question that hasn't been answered. Okay, wait. If I mention this in Google, what will it say? Like, what will it say? Okay, like, what happened? No, what? No, what's going on? Yeah, what molecule is actually the genetic material? This is the question that they didn't answer. Do we have the answer in this modern life that we are living on? Molecule, material. No, it's just so fun studying life. Like, that's so fun, bro. Okay, that's just DNA. It just says DNA. Okay, I'm gonna answer DNA. Okay then, DNA actually. But yes, question that hasn't been answered. This is given in the textbook that the question hasn't been answered, that what molecule was actually the genetic material responsible for this type of genetic inheritance at the molecular level, that is DNA actually. And then Griffith's experiment, which is basically a experimental method of explaining the transforming principle. Okay, this is done in 1928, according to the textbook. And in 1926, the search for the genetic material or the genetic inheritance at molecular level became important. Then, yeah, uh, we had done a lot of things. Okay, just a small recap of what we did. Okay, I'm just going through. I'm not reading. I'm just going through. That's it. Small revision of what we did. Okay, because I'm not gonna waste time. Okay, wait. Oh, okay. Think about it. Yes, yes, yes. Takes time to go through. Okay. Yeah, 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 yeah. So, okay, wait. Arvind Chagraph. 1 minute. I didn't go through this one. The number of adenine and thymine. Is it thymine, right? Yeah. Should be adenine and thymine. Yeah. And guanine and cytosine are constant, so they give the ratio as 1. Okay, that's it. Yeah, the base pairs confer uniformity. I'm going through this. Just going through this. Okay, I have to go through this topic once. Parental DNA acts as a template to form the new daughter DNA, which is identical to the parental DNA due to the sequences of the base pairs formed. Okay, that's one thing. Okay, that's one idea. Let's move on. Selling features, bands. That's it. Just go through bands. Bands, right? So, ANS is anti-parallel, nitrogen is base, distance, and stability. P is backbone. Then central dogma. Revolutionary, yes, yes, yes. Retrovirus, they just do that reverse transcription. That's done. Then this one we just gone through. It's an interesting topic, if I'm being honest. Then here we are. Okay, so yeah, we did a quick revision. I just didn't want to waste time. So 1926. Okay, we have not much time. Just have 1 hour 18 minutes left. Dang, this topic is just like so small, and I'm giving it so much time. Like, that's crazy, bro. I shouldn't be giving that much time though. That's crazy. That's crazy, bro. I think I should have given this 3 hours and diagram and other work 2 hours, but I don't think so it would have been complete at that span of time. Let's just complete. 1926, the quest to determine the the genetic inheritance molecular level. Then at 1926, right? Yeah, took time to find out whether the DNA is the genetic material. That what molecule was actually acting as the genetic material. Then Griffith's experiment, which is a transforming principle, explained the way that how the genetic material experimentally explain that. And 1928, okay, let's read this. Frederick Griffith did a sequence of experiment for the bacteria Streptococcus pneumoniae. Streptococcus pneumoniae, remember that bacterial name, where he observed that it can change its physical form, and when grown on a cultural plate, there are two types of strains formed, the S strain bacteria and the R strain bacteria. The S strain bacteria contains a polysaccharide smooth polysaccharide coating, which forms smooth shiny colonies, but there is another core of colonies formed by the R strain bacteria, which were not containing those coats. But however, the S strain bacteria were virulent, which means that they can affect the person or the animal with the disease pneumonia. But R strain bacteria were non-virulent, which means that they can be easily killed, and they cannot affect the person or any kind of animal to undergo through the disease pneumonia. Contains mucus smooth polysaccharide coat, doesn't have a layer on it. Okay, this is the one main thing. So the Frederick Griffith, what did he do is that he actually injected S strain bacteria in the mice, and the mice died. But once he actually like injected R strain into another mice, R strain bacteria did not affect the mice, so our mice literally lived. Then what did then the bacteria can be killed by like heating. So heat killed S strain bacteria. The S strain bacteria was killed by the heat, so this was injected into the mice, and the mice still lived because S strain bacteria wasn't active here, it just died. Then we got the R strain bacteria, where where it was injected into the mice along with the estuarine heat killed bacteria. It is found out that the mice literally died. Why? Because the R strain bacteria would transform the genetic material to the heat killed S strain bacteria, and thus the S strain bacteria becomes active, causing the problems to the heart and forming the pneumonia disease over the rat. Thus the rat died. Okay, I just explained it. That's what I remember. So Griffith observed this and stated that there's something which is acting as a transforming principle, that somehow the genetic material transfers from R strain bacteria to heat killed S strain, forming the overall virulent S strain. Now wait, I wanted to know. Okay, I think most probably the bacteria as you see here DNA. So yeah, I'm not gonna go for that because they are not virus. Is the only one that is containing RNA, right? So retroviruses. So that's one thing. So many biologists were convinced with this experiment proving the transforming principle. Uh, it didn't describe the biochemical characteristics of genetic material. Okay, this is one is important though. So Griffith's experiment wasn't able to explain the biochemical characterization of the DNA, the genetic material, and this wasn't convincing for many other biologists in that time. Using of explanation of the transforming principle of the proving the transforming principle, and then yeah, we are not gonna do the Hershey and Chase experiment because that's not there in our syllabus. Uh, Biochemical characteristics of transforming principle. This is the last topic, and then we'll complete this chapter. Hooray, bro! We are doing great. We are doing great. We are cooking now. Okay, that's great. I'm actually fully focused. I know this is. I'm not. Why did I boast, man? I shouldn't be doing that. Trust me, I do not boast like this. I just somehow say it accidentally. Okay. Oswald Overy, Macklin McCarty, and oh, these names are hard. Oswald Avery, Macklin McCarty, and Leon McLeod. Right? No, Colin McLeod, not Leon. Colin McLeod. They proved the biochemical characteristics of the genetic material of the transforming principle. Actually, is it given us that? Yeah, biochemical nature of genetic material. for the previous one. So, biochemical characterization of transforming principle was proved by them. They purified the DNA, RNA, and proteins to check which one was responsible for the transforming principle. Turns out that RNA and proteins weren't responsible. It was the DNA who did this. So, DNA, what happened was that there are some enzymes called RNase and protease, which were basically those digesting enzymes. Or like it would digest the RNA and the proteins. Basically, these were not involved in the transforming principle at all. However, the DNA was there, which was responsible for this, and it turns out that the DNase enzyme was basically inhibiting the transforming principle. That's what I remember. Let me just check if I'm being not wrong. Digestion with DNase did inhibit transformation. Okay, digestion, digestion of DNase inhibited it. If I want to expand it into long form for DNase, it's deoxyribonuclease. DNase inhibited transformation. So DNA was the one responsible of all this. DNA is the main character of this chapter, what I know so far. Thus, DNA is the hereditary material. DNA alone transformed our strain to strain, but not all biologists were confirmed. Even though there are other characteristics like that we'll learn, of course, but DNA is the main one here. The DNA is actually having its domain here. Okay, so we have completed the chapter, but I think we have missed some small parts which we need to go through, and I think it was this one, the limitations again. So many biologists were convinced of the transforming principle proved by Griffith's experiment, and then what else? What else? Because it's a bit weird to understand that how did the Estrine bacteria, which was killed by the heat, got alive? That was really confusing, and that when he said that, it was a bit confusing because they never actually might have seen this type of thing. So I might agree with the biologists because this is a very confusing type of phenomenon. And then another thing about this is that he wasn't able to prove the biochemical characterization of genetic material. Okay, yeah, what did Frederick Griffith observe that when he was adding heat killed estrine bacteria and archaean bacteria together, and then he injected to mice. If when he saw that, he found out that mice was dead. He even dissected if you don't know, it's an outside knowledge, it's not in the textbook. He found out that he dissected the mouse, and he actually found out that there were estrine bacteria in the heart, and those estrine bacteria convinced him that, yeah, the what happened was our strain bacteria somehow revived the heat killed strain bacteria. What actually happened was there was something that was acting as a transforming principle, where the our strain bacteria, it was transformed from our strain bacteria to the heat killed strain bacteria, forming the overall living strain bacteria which killed the mice. So that's one thing, because of course strain bacteria is virulent. And again, I'm explaining those because I want to keep them in my mind so that I don't forget. Last, biochemical characterization. I need to remember the names. Oswald Avery, McLeod. Oh my god. Man, the names. Colin McLeod, Oswald Avery, and what's that guy name? Another Macklin McCarty. Ah, the names. OMC. Nah, that's not. That's calm. Okay, let's go for calm. So whenever I want to know the name, I'm just gonna write the mnemonic calm. Colin McLeod, Oswald Overy, and Macklin McCarty. Okay, not bad. I just need to remember the first names: Colin McLeod, Oswald Overy, and Macklin McCarty. Okay, they proved the biochemical characterization of transforming principle, where they purify the DNA, the RNA, and the proteins, and it turns out that the proteins and RNA were the ones that were responsible for this transforming principle, as the RNAs and protease enzymes, which are the digesting enzymes, were not were not affecting the transforming principle. However, the DNA was one responsible for this, as the DNAs would digest to inhibit the transformation, and thus they concluded that DNA is the hereditary material. DNA is the hereditary material, but all biologists were not concerned. Destroy and we completed this. Let's go. W, guys, we did it. We did it. So here we are, completed this. Now we need the textbook and a phone. Okay. Here's 4.1. Do you guys want to do this through laptop? Let me just check. We can do this through laptop too. It's not problematic. Yeah, you know what? Let's just do it through a laptop. The problem is I just want to not showcase it up. But okay, let's do it. Let's do it. Let me go to this. You cannot see anything right now, but I will show it to you. To create another window capture device, because if I show display capture, anything can go wrong. Display capture shows everything, like I mean everything. That's why it won't be good. Oh, she didn't. Oh, 86. I mean, maybe it could be the new textbook. I don't know. 1 minute, guys. I'm gonna show it to you. Just wait. This is gonna be the one. Okay, so we start off from here. 1 minute. Ah, my eyes. Oh, what is going on? Okay, let me just create another one. Okay, let's not waste time. We just have 1 hour. Wow. Guys can only see a white screen, right? Wait. Let me keep it here. Here, what? Back here. Okay. Um, you guys see it now? What? Why is it showing blank? What if I do this? Oh, what? I have no idea what's going on. You know what? Let's just delete this one. Let's make another window capture. No, not this. Window capture. Okay. For this one, I'm gonna open this. This. Or wait. Wait. I don't remember if I have it, but I'll use WhatsApp for this. Wait, because I literally made chapter-wise notes, like full. Like even though those are only PDFs, it's not mine though. It's from my biology teacher, but I've sorted them out. Like I literally sorted them. It will be really helpful. I don't remember where I had done this, but let me just check. Let's go for this. I'm not gonna actually show it because I'm scared if I can leak any number. Yes, I found it, guys. 1 minute, guys. I'll go to toilet and come back. It's a bit urgent. Alright, we are running out of time, bro. I'm sure that I'm not gonna complete it, but let's try, man. I'm not gonna give up. Let me just switch this off, okay? So, can I like open the window for this? Let me just check. This is gonna be weird, bro. Where's window capture 2? Wait, oh yeah, it's another one, right? Yeah. So let's keep it back. Let's double tap and okay, here we go. It's not big. You know what? Let's not do that. You know what? Let's just if I open this, it's gonna just disappear. That's what I know for sure. It's gonna disappear. Never mind, it didn't disappear. It's opening Word though. Okay, you know what? Let me just like switch this to Word. That's what I'm gonna do. Yo, guys, come on. Man, I don't know whether I can even screen share because it will take a lot of time. And why is Word taking so much time? Yeah. Okay, so okay, no problem. I'm not gonna bother with that because I'm not using Microsoft. But you know, like Microsoft, unless if and until if I need to edit it, I mean, I'm just gonna see and check. That's it. So wait, here we go. Yeah, this is the one. Let me increase it because we have less space. Okay, that's pretty impressive. I can just like do that. Don't worry, this side won't be cut off. It's clear enough to just check. Um, okay, can you guys see now? Oh yeah, that took a lot of screen, huh? That took a lot of juice. Let me just stretch it out this much. Okay, this one is fine. This one is perfect. Come on. Okay, so the male reproductive system. Okay, it's just skipping to that. A pair of testes, accessory ducts, glands, and external genitalia. The testes are situated outside the abdominal cavity within the pouch called the. Uh, really, I don't know the name of the pouch. That's crazy. Wow, scrotum. Why did it take me so much time? The scrotum helps lowering the temperature of the body. Like the scrotum helps in the lowering of the body temperature by 2 to 2.5 degrees Celsius for the help of spermatogenesis. In an adult, each testis is oval in shape with a length of 4 to 5 centimeters and width to 2 to 3 centimeters. The testis is covered by a dense covering. Okay, just by having a dense covering, and then each of them consists of 250 compartments known as testicular lobules. These testicular lobules contains 1 to 3 highly coiled seminiferous tubules in which the sperms are produced. Okay, let's go. We are doing great. 1 to 3 highly coiled seminiferous tubules means that there are main germ cells known as spermatogonia, but these are called as the mother gamete cells too because these are the ones that are forming gametes through the help of spermatogenesis, and that's like you know the process. And then there is another one that is known as the interstitial spaces where there's Leydig cells, there is incomplementary sorry, is it called as immunocompetent cells? Yeah, immunocompetent cells, blood vessels, small blood vessels by the way. And yeah, it's called interstitial cells. What, did we see that coming? No. But yes, this Leydig cells will secrete androgens. Then the Sertoli cells will provide nutrition to the germ cells, the male germ cells. Kids, if you're watching this, I just want to say that you know you're gonna study this in future. Right now, we are just gonna focus on this. It's just gonna be a bit of you know, this is not for kids at all. This is not for kids. Remember, trust me. Please, YouTube, please do not like switch this off. I'm not showing anything bad. I'm just like steady. I can help out others while like you know doing this revision. Male germ cells from spermatogonia, sperm formation, Sertoli cells provide nutrition to the germ cells. There are many types we can actually check. Yeah. The male accessory ducts are reticestus, the vas efferentia, and the epididymis, vas deferens. The seminiferous tubules of the testis opens into the vas efferentia through the reticestus. The vas efferentia leaves the testis open. These are just exact biology textbook ones. And open into epididymis located along the posterior surface of each testis. Okay, so epididymis is located. It will move on to the epididymis, which is located on the posterior surface of the testis. The epididymis leads to the vas deferens, and that ascends to the ascends to the abdominal loops over the urinary bladder. It receives the ducts from the seminal vesicles, and then it opens into urethra, which is the ejaculatory duct. Then these ducts store and transport the sperms. That's what they do from the testes to the outside through urethra. The urethra originates from the urinary bladder and extends through the the I cannot even, I don't know whether I can even say this. To the male external genitalia, one part that is elongated, to its external opening called urethral meatus. Okay, man, the problem is I don't like swearing. Now, of course, this word is not meant for swearing, but the problem with this people is that they will feel like, oh, this guy literally swore. But bro, this is just biology. I'm just studying this. Who knows? Even if this comes for exam, I might have to write this out. Like, yeah, this goes from there and that's blah 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 blah. I have to write this down, bro. Think about it. The male accessory glands, paired seminal vesicles, prostrate glands, and the bulbourethral glands. The secretions of these glands constitute the semen that is in the seminal plasma, which is consisting of fructose, calcium, and certain other enzymes. Then, yeah, the female reproductive system, which consists of a pair of ovaries, pair of oviducts. Oh yeah, this one is a bit big. Pair of ovaries, pair of oviducts, uterus. Then we got the uterine walls also, but uterus. Then cervix, cervical canal, but just say cervix, vagina, and then what else? Along with a pair of mammary glands. Yeah, mammary glands is also taking place as a part of the female reproductive system. Ovaries are the primary female. Those are how do I mention it? Um, Okay, ovaries are just primary female. Uh, I'm just gonna say sex organs that produce female gamete ova. Man, is anyone watching this? I swear, if anyone's watching it and they're like, "Yo, what in the world is bro doing?" Okay, that's crazy. And several steroidal hormones known as ovarian hormones like progesterone, estrogen. It is capable of also like secreting, for ovulation, and then yeah, the length is about like 2 to 4 centimeters about this, and then it is connected to the pelvic wall and uterus by ligaments. Okay, so ligaments are the ones that is connected by to the pelvic wall and uterus. Each ovary is covered by a thin epithelium which encloses the ovarian stroma. This ovarian stroma is divided into outer cortex and inner medulla. Ouch. Ow. The ovary. Okay, the oviducts, fallopian tubes. This is gonna take time, guys. This is gonna take time. Ouch. We have completed the big chapter work, and then this. Okay, two small chapters versus one big chapter. That's the thing. The primary female sex organs to produce is a blah blah blah. Okay, that's done. Located on each side of the lower abdomen. They're located on each side of the lower abdomen. Then what else? They are 2 to 4 centimeters long, and then what about that is that they are connected to the fallopian tube. This fallopian tube is 10 to 12 centimeters long. It consists of the infundibulum, ampulla, and isthmus. Ampulla is the wider part of the fallopian tube. Isthmus is the just narrow part to which it is having a cavity open to the uterus. Then the infundibulum has a fimbriae, which are finger-like projections which collects the ova after ovulation takes place, or after the rupturing of the graphene follicle. Then peripheral cortex and inner medulla is the ovary is covered by thin epithelium, which is known as the ovarian stroma. Yeah, outer cortex and inner medulla. That's what I've mentioned. Extends from the periphery of each ovary to the uterus. You know what I'm actually worried about. Like, I don't know if I can write the exact points, but I just want to be like very sure about this. Okay. Fimbriae, which help in the collection. Infundibulum leads to the wider part. Okay, that's done. Last part of the oviduct has a neural and joins the uterus. Okay, the uterus is a single inverted pear-like shape, and then it is also called as worm. And yeah, it is supported by ligaments attached to the pelvic wall. So the ovaries are also attached to the uterus and the pelvic wall through the help of the ligaments, and then it is also supported by ligaments because uterus is also attached to the pelvic wall, and then the uterus opens into vagina through a narrow cervix with the help of the cervical canal. The cervical canal and the vagina together constitutes the the Ballard's canal. Then we got the walls of the uterus, which are very important. These have three layers of tissues. The external thin membranous tissue, which is known as the perimetrium. Then the inner ground. Sorry, not that one. The smooth muscle middle layer that is known as myometrium, and then which helps in the contraction or child labor. You know, during birth, yeah, parturition, let's say. Then endometrium, which is basically the inner layer or inner granular layer of uterus, is known as endometrium, that lines the uterine cavity. It undergoes cyclical changes of menstrual cycle, while the myometrium exhibits strong contraction during the delivery of the baby. I don't know why, but I feel like my endometrium has another importance. I don't remember. Yeah, implantation. Implantation. I forgot about that. Then external major genitalia, if external female genitalia, mammary glands. I don't know why, but people usually skip this concept, but this is also a topic that we need to study. Okay, let's start with mammary glands because it's a bit weak for me. So it is containing glandular tissue. I didn't know that, and variable amount of fat. Yes, like the fat is here. This is areola. Then okay, I don't have a functional mammary gland, of course not, because I'm not a female, but still. Glandular tissue is divided into 15 to 20 mammary lobes, which are containing clusters of cells called alveoli. These are milk secreting cells and can also store these milks in them. Then the alveoli open into, okay, this one is a bit complex, mammary tubules. Then these joins to form a mammary duct. Mammary ducts join to form a wider mammary ampulla. Then lactiferous ducts. That's crazy. Mammary tubule, mammary ducts, then mammary ampulla, so mammary tubules, mammary ducts, mammary ampulla, and lactiferous duct. And through which the milk is sucked out. Yeah, this is for breastfeeding, by the way. For external male genitalia, you know what I'm gonna say? The penis is the male external genitalia. The enlarged end of penis is called glans penis, and it is covered by loose fold of skin called foreskin. Yeah, this one is strong, and an external female genitalia. The mons pubis, which is a like cushion of fatty tissues covered by skin and hair, pubic hair. Then the labia majora, which is the portion through which it is found under the mons pubis. The fleshy folds of tissues are found, fleshy folds, okay, and then which extend down from mons pubis and surround the vaginal opening. But that's actually a bit labia minora because there are paired folds, under the labia majora, and paired folds of tissues under the labia majora, and then a hymen. The opening of the vagina is often covered partially by this membrane. The clitoris, which is a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening. Yeah, it's there in the textbook diagram about the, like, I don't know what do you call it, cross section. Ouch. Secretes androgens with the help of the Leydig cells and can also like help in the formation of sperms. No, the temperature. The temperature is the main factor. To doesn't actually uh, give the enough decrease in temperature from 2 degrees to 2.5 degrees Celsius. That is the main requirement for the spermatogenesis, or else it won't happen. Fructose cells and certain other enzymes. So we're doing great, right? Are we doing great? I feel like we can do this. I feel like we can crack this up. It will just take time, but we can do it. Yes, we have confidence. Let's go. Hmm. Okay, let's go through this again. Small glance. Oh my god, I don't know how much time this took. This is just one PDF, by the way, guys. One PDF. We have like 5 PDFs of these, right? How many do we have? Let me just check. I don't know whether I can show it to you, but let me just check. Like, that's one thing. Oh man, I have to open WhatsApp because that's where I keep the folders in. Let me just check. I think you know what? Let's not close WhatsApp. I need it. I need WhatsApp. I'm not scared about it because window capture won't leak anything. I'm sure about that. Unless and until I show WhatsApp, so I'm not gonna do that. Yo. Where is the person, bro? Yeah, here you go. I just wanted to talk with you, man. Why can't I go up? Yo, what? My laptop froze, I guess. Yo, what just happened? Okay. Yo, what? My WhatsApp literally crashed. My WhatsApp crashed, bro. What? Bro, how am I gonna study then? Come on, do it fast because we have to move on, bro. I'm gonna open another one. Yo, what? Bro, how am I gonna study then? This is not gonna look. This doesn't look good. You know what? Let's just open Teams and do it because it's wasting my time. I don't know why is not anyone cooperating with me, man. Like, what's going on? So let me close that up. I'm gonna open another one, guys. Okay, this time it's not gonna open on Microsoft Word, but it will open on Microsoft Edge. So I have to like literally open Microsoft Edge. Where's Microsoft Edge? Let me check. Microsoft Edge. I hope you're available, bro. There we go. We got it. What is this? Okay. I think you guys can see it now. Let's go. That's what I want. Yeah. Okay, so we got gametogenesis, which is the first process that takes place in the human reproduction. Spermatogenesis. Now, this is in the case of male, where the spermatogonia, which is the mother gamete cell or the germ cells, which will multiply and divide, basically, can just say, to increase in number through the help of mitotic division. And yeah, that's one thing. These some spermatocytes. Sorry, what am I saying? Some spermatogonia will act like primary spermatocytes will periodically undergo a meiotic division. That is, in the first meiosis, they will form secondary spermatocytes, and then after the second meiotic division, remember, guys, I'm not teaching or anything. I'm applying Feynman technique, which is just like teaching, but you're just basically like studying. That's it. I'm revising, by the way. This is not you like going through it. So secondary spermatocytes, which is in the second meiotic division. Now, after the second division, we get second meiotic division. We get these spermatids. Now, spermatids are basically not in the shape of and yeah, also one thing, secondary spermatocytes are haploid, because in the first meiosis this reduction division takes place. So two haploid cells will be formed, that those are secondary spermatocytes, but primary spermatocytes is diploid. These spermatogonia is also diploid. One idea about this, okay? Then we get these spermatids, the haplospermatids, haploid spermatids, by the means of that it will undergo another process to form those very unique type of structures called sperms, which are also known as spermatozoa. Let me just take the scroll down. I should use mouse. What do you guys say? Spermatozoa, but let's not. Let's just focus. Spermatos. Yeah. These four things. I like the way the diagrams are given. Like those balls. Like this is spermatogonia. This is primary spermatocytes. These divide into two haploids, four secondary spermatocytes. Now these divide into four spermatids, and then these form these sperms. And then the sperm heads becomes embedded in Sertoli cells. Then they are finally released by the from the seminiferous tubules by the process called spermiation. And then they are released by from the they arise from the seminiferous tubule. They will be released from there as they since they were embedded in the Sertoli cells, and spermatozoa that sperms are formed. And you can see from this diagram, actually you can see that these are spermatogonia. These are primary spermatocytes which are going under the secondary first meiotic division. They will form secondary spermatocytes. These secondary spermatocytes will form these spermatids. Now, what I feel is that this diagram is a bit wrong with the labeling. Like, I don't know why this is mentioned as spermatids, because trust me, uh, spermatids should be this, I guess. Why? Because this is a sperm, literal sperm, literally like after the process of spermiogenesis. I don't know why it's mentioned as spermatid. This is a bit confusing. And if you're, and this is basically spermatid, and yeah, you can see that sperm is emitted here,
and by this process of spermiation it will be released, and then it moves on from the seminiferous tubules to the retitestis. Then the human sperm, we can see this. This is an important diagram. I need to learn to draw this too. This is the tail, which is very crucial for the movement or the motility of the sperm. Then we got the head, which is elongated. Then the acrosome, which contains certain enzymes which will help in the fertilization. Plasma membrane is also there. You can see that there is plasma membrane. Nucleus containing chromosomal material, that is haploid, so N chromosomal material. And then neck, here there is neck. Here there is another thing actually, which is not mentioned in the textbook. I don't remember what it was, but in NEET we study about this part too. There is some small part, you can see something minute. I don't know what it was called. Anyways, this is the middle piece. This middle piece consists of several mitochondria, which are the energy source for them to swim or for to show their motility. Yeah, again, just a small recap what we did. That there is plasma membrane. Head is also there. This head is containing those nucleus material. This is the middle piece, contains several mitochondria, which is which are the source of swimming. Source of energy for swimming, and this is helping for the motility. And then the plasma membrane. That's it. Let's go. Hooray! Then we got the hormonal control of spermatogenesis. At puberty, we are getting okay. At puberty, okay. If you don't know, this starts at puberty. This one, this one is important because the hypothalamic hormone, that is Gen RH, which is also known as a gonadotropin releasing hormone, will stimulate the anterior pituitary hormone. Okay, this anterior pituitary hormone would stimulate the releasing of the FSH and LH. FSH means follicle stimulating hormone, and LH is luteinizing hormone. The luteinizing hormone will act up along with the Leydig cells for the secretion of androgens. Okay, this one was important. I wanted to know that. Whereas FSH will act upon the Sertoli cells, where it will help in the what do you call it, for helpful in some factors, like it says that it helps in spermiogenesis, which is great. But there are many factors that we study in NEET too, so that's one thing. We'll have to go through that for the NEET level. These are those types of things which I might have to study tomorrow, like at the morning time, for those extra things that I need to cover. But for now, these are the ones that I've checked. And then, yeah, the diagram. This one is important. Okay, so 1 2 4 2 2 4 4 4. Okay, 2 2 4 4. 1 2 4 2 2 4 4. Okay, so what happens is that this is the spermatogonia, which are mitotically dividing to form more cells, increasing in number. This spermatogonia would some spermatogonia will actually be called as primary spermatocytes, because they will periodically undergo meiosis, forming these secondary spermatocytes. These secondary spermatocytes are undergoing the first had undergone the first meiotic division, so they are haploids. So 23. No, sorry. This is primary spermatocytes. They will divide to form the first meiotic division, secondary spermatocytes, which are known as which have 23 chromosomes, and these will undergo to form the spermatids after secondary meiotic division. So this is the second meiotic division, where it is the equational division, and this will form the spermatids, and these will undergo through spermiogenesis to form the sperms, and that's the spermatozoa that we are looking for. Okay, that's the diagram. My bad. Uh, Structure of sperm that will help. So it consists of head, neck, middle piece, and tail. Okay. A plasma membrane envelops the whole body of sperm. It consists of agrosomes, which consists of certain enzymes which helps in the fertilization of fusing of the sperm into the ova. Then sperm heads contains elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure called acrosome. The acrosome is filled with which helps in the fertilization. Middle piece possesses numerous amounts of mitochondria, which helps. Okay, wait, let me charge. Which helps in the which gives the energy source for swimming. Then, uh, okay, middle piece possesses. Yeah, that's done. Okay, so it consists the plasma membrane envelops the body of sperm. Then the head is actually the elongated elongated part which contains the haploid nucleus. Then what else can we say? Then yeah, uh, which produces energy. Okay. Now the human male ejaculates about 2 to 3 ml of semen, that is 200 to 300 million sperms, which is pretty much a lot if I'm being honest. During a coitus, which is basically called as something which I want to say right now because this is a family channel. You know what? I'm happy. I'm really happy that there are no people watching me right now because, bro, if anyone was watching that, it could have been messed up. Yeah. So, oh, it's only spermatogenesis here. Okay, my bad. I didn't notice that. Then vas deferens, seminal vesicles, and prostate are essential for maturation and motility of sperms. The seminal plasma, along with the sperms. Okay, seminal plasma, along with the sperms, give the semen, and that's one thing I forgot to say. So secretions of epididymis. This was there in the textbook too, like in the earlier page. Secretions of epididymis, vas deferens, then seminal vesicles, then bulbourethral gland, and the prostate gland helps in the transport of the sperms or maturation and motility of sperms. Okay, are essential for maturation and motility of sperms. Are essential for maturity and motility of sperms. Okay, so we need to draw a label diagram for sperm and explain the functions of all part. Okay, we can do that. Define spermiogenesis and spermiation. We just did that. Name the hormones involved in the regulation of spermatogenesis. Those are LH and FSH, which are the gonadotropin hormones, stimulated by the help of the gonadotropins, which are stimulating the anterior pituitary, which is the FSH and LH. The LH hormone will like stimulate the androgens. They will act along with it to secrete the androgens, whereas the FSH hormone will stimulate the along with the will act upon the Sertoli cells to help in some such factors such as uh, spermatogenesis, helps in spermatogenesis, and that's what I could say for now. Spermiogenesis. Okay, let's say spermiogenesis. Yeah, what is spermatogenesis? It is a type of gametogenesis for males. I'm just gonna say that. I know it's not like that. You should not be saying that. I think both the questions are same. The first two. Uh, okay, that's it. Yes. Okay, we are doing great. Let's go. All right, we have completed this, and we are moving on to the next one. Okay, so we are gonna move on to oogenesis, which is a bit more complex. Let me just show this. All right, I got another one. Okay, Microsoft Edge is also great. So human reproduction, lecture 2.3. Okay. Oogenesis, which is a process of gametogenesis taking place in females. The process of formation of mature female gametes, that is called oogenesis. Okay, wait. Oh, so the process of formation of sperms is called as male gametes is called as spermatogenesis. Okay, my bad. Yeah, this could be coming as like 2 mark questions. I have to be really careful about that. I didn't notice that. So the process of formation of mature female gametes is known as oogenesis during the embryonic developmental stage. After the child is has reached his after the child was born, the literal oogenesis literally stops from there. Sorry, the formation of oogonia stops now. I feel like ovaries are just too OP for females. There are a lot of things that ovary does. Ovulation, and then helps in the secondary sexual characteristics. Then it is the one that is releasing estrogen and progesterone, which are literally helping out in the implantation, not implant fertilizer after the fertilization, how to take place implantation and all that stuff for the uterine wall. It's really useful. Bro, what? And guess what? This oogonia is formed by the ovary itself in the fetal stage. So when in the ovary of the fetus of the girl, so it's fetal ovary. No more oogonia are formed or added after birth, and then they enter prophase one. Some oogonias will enter prophase one, which gets temporarily arrested at that stage, which means it completely stops. So it forms primary oocytes. and these primary oocytes will get surrounded by a layer of granulosa cells. These granulosa cells, in which they are surrounded by, will form the primary follicle. These large number of these follicles will degenerate. So during the lifetime of females, only 60,000 to 80,000 of these primary follicles will remain. However, what will happen is that they will form a new theca layer and more granulosa cells, and that develops into secondary follicle. What happens is that, yeah, 1 minute. Why? Okay, the points are a bit too jumbled. No, not jumbled. Like, a bit. The positions are a bit different. This is for the secondary follicle. This one is the tertiary follicle. The secondary follicle will develop into tertiary follicle, which consists of characteristics of fluid-filled antrum in them, in cavities, and then it also consists of new theca layers, that is the internal theca, inner theca interna and outer theca externa. Then what else is the tertiary follicle, which yeah, that's tertiary follicle. My bad. Now, there is a note that we need to know is that the primary oocyte will complete its first meiotic division in the tertiary oocyte, forming uh, the unequal, that is the large secondary haploid oocyte, secondary haploid oocyte, and then a first tiny polar body which will degenerate. It won't live for long, and then this large haploid secondary oocyte which is formed will be again in the tertiary follicle. This tertiary follicle will become matured, and they will mature, and they will form the Graafian follicle or the matured follicle. This one, where the secondary oocyte will form a new membrane called zona pellucida surrounding it, and then this ruptures, releasing the secondary oocyte. Now, I don't know, they didn't give this here, but I think in the textbook there is the diagram. Yeah, this one. Here, this is the corona radiata. This is the perivitelline space. This is plasma membrane of ova. This is in between. Here is the zona pellucida. All right, that's what I wanted to look at, because it's not given here. Maybe it's given in the future topics that we are going to look at. Okay, so we need to learn to draw this, which is a bit coughing or choking thingy. Uh, okay, there's this one too. I didn't notice that. Draw a label diagram of Graafian follicle. Okay, we'll learn to draw this one too. So this is the secondary oocyte. What's this? Cavity of follicular fluid, follicle cells, theca. Okay. Okay. Wait. You know what? Let's chill. Let's chill with this. Let's chill with this. Because we need to give time. Okay, we are kind of cooking, guys. We can complete this too. I'm confident that we can complete this, and then the other chapter is very small, so we can also complete that easily. But the problem with this is that the next one that we are gonna focus on is a really big, really big chapter. So we have to do it as fast as possible. I feel like we'll take 3 hours for this, bro. This is not gonna take enough time. But I do know that we'll complete full biology so fast today. Like we can even get enough time for maths. I'm sure about that. So yeah, let's go. Why is my FPS dropping? Wait a minute, let's work. My FPS is dropping. All right, is it? Yeah, it's fine. So again, because oogenesis is a bit more complex, I want to actually revise again. So the process of the formation of mature female gamete is known as oogenesis. The mother gamete cells, which are known as oogonia, are formed in the fetal ovaries, in which this takes place during the embryonic developmental stage. After the birth of the child, the no more oogonia is added or formed. Then these some oogonia will undergo meiotic divisions called as primary oocytes. These primary oocytes undergoing meiotic division will enter into phase 1 and will get temporarily arrested at that stage. These primary oocytes will develop a new layer of cells known as granulosa layer of cells, layer of granulosa cells, forming the primary follicle. This primary follicle will develop into forming secondary follicle. Primary follicle. A large number of these will degenerate. So only 60,000 to 80,000 remains in the females in their lifetime, and then these primary follicles will develop into forming secondary follicles by more layers of granulosa cells being added to it, and Yeah, ethical and ethical layer being formed on it. So that's one thing. And then these secondary follicles formed will actually like undergo further development to form the tertiary follicles, where the tertiary follicles will have more detailed tunicle layer, that is the inner theca interna and outer theca externa. Not only that, about this we also focus and get the idea. What am I doing? What am I saying? Yo, what, bro? No way, bro. A fluid filled layer called antrum. Yeah, the theca layer is organized into an inner theca and outer theca external. Okay, what was I thinking about? No, wait. Yeah, fluid filled antrum. There is an antrum which is a fluid filled cavity, filling the cavity of this, I believe. That's what I was saying. And there is a note that the primary follicle would sorry, the primary oocyte will complete its full first meiotic division, where it will form the two unequal haploid cells. That is the secondary haploid secondary oocyte, large haploid secondary oocyte, and then the its first unipolar body, which will degenerate. Now what happens after that is that the follicle will get matured. This will form the Graafian follicle. This Graafian follicle contains the second yeah, during that time the secondary oocyte would also will exhibit a layer called as the zona pellucida, and from there the overall ova is formed. So ruptures to releases the secondary oocyte ova from the ovary by a process called ovulation. So this Graafian follicle will rupture, and then ova is formed. After that is the formation of corpus luteum, which only secretes progesterone, and then the diagrammatic view, which we need to understand, and that's it. Okay, let's go. We are going great. Okay. Now this one is 1 2 4 1 2 2. Okay, that's a bit weird. Now this is important actually. It says that 1 minute. Is it there in the point? What? Okay, wait, this is confusing. It says that the second tiny polar body is also released in the adult reproductive when it goes in the second meiosis. Okay, in the second meiosis of yeah. Second secondary oocyte will also go under the second meiosis. I remember that. It will form second polar body and ova. But it is not mentioned here. It's not big matter, but then why is it not mentioned here? Yeah, I remember it was mentioned in the textbook, but it wasn't mentioned here. That's weird. Okay, so it's 1 2 4. Okay, this is a diagram. I know we need to like learn that too. The problem is that diagrams will take time because what are these diagrams? Well, these are so complex. And then yeah, we have the Graafian follicle, which is a theca. Then the layers of cells like follicle cells, and then what is this? Cavity of follicular fluid. I think that was fluid filled antrum, right? Antrum, yeah. And then this is the secondary oocyte. Now the secondary oocyte goes under the second meiotic division, forming ova and second polar body. What is oogenesis? Draw a legable diagram of ovary. Yeah, we'll do that. It's something like small bladder cells, and then we got the Leydig cells too in this, actually. I think Leydig cells also exist in female reproductive system, I believe. Do Leydig cells, I believe, bro. I remember saying it. Yeah, see, already you can see that it produces estrogen. What? Is it true? I did not know that. What? I did not know that. That's interesting. Okay, wait. So, what are the interstitial cells? What are the interstitial cells? I want to know because cells in ovary. Can you guys even see this actually? Like, oh, okay. You guys cannot see that, right? Let me just open that. Oh, I know. You might be like, wait, what's going on? I'm just checking. Steroid producing. What? Where is it? Mesenchymal cells. Okay. In short, we can just say these are so complex. In short, we can just say that, yeah, it is there. Hmm. Okay, we don't have enough time. Let's move on. Too much brightness. Let me decrease. Why is there too much brightness in my laptop? Let me increase this much. Yeah, that's enough. Like there's too much brightness in the laptop, bro. Oh, okay. Here we go. This one, and then what else? What is oogenesis? Yeah. Oh, okay. See, it says that give a brief account of oogenesis with schematic representation. Is it talking about this? Let me just check. Bro, come on. Do we need to learn that? Like, those are so complex. Yeah, it says schematic representation, bro. This is the one. Oh, okay. However, we need to learn this one. So the sativa follicular cells, follicular fluid, then this is the secondary site. Okay. Alright, I feel like I'm confident in this. Let's move on. 2.4. We're close, guys. We are close. Wait. How many lecture? How many more? Okay, only two. Only two. Let's go. We can do this. We can do this, guys. We can do this. And then this chapter is also complete. Oh man, this is a bit choking. But okay, let's try completing this chapter and another chapter in just 20 to 30 minutes. Because I think the other chapter is very small, we can complete it fast. Let's go. So menstrual cycle, yeah. The first menstruation begins at puberty, which is known as menarche, and then in human females, menstruation is repeated at average intervals of 28 to 29 days, and the cycle of events starting from menstruation till the next one is called menstrual cycle. So the cycle of the menstruation cycle from which it starts from the menstruation to the next one is called as the menstrual cycle, and then it takes place in an interval of 28 to repetitive interval of 28 to 29 days. Then the four phases of menstrual cycle. The first one is the menstruation. The second one is the follicular or proliferative phase. The third one is mid phase. Is it given here? No, it's gone. Not. It's not given. Okay, it's given as ovulation, not mid phase. Ovulation. Okay, that's it. Then luteal phase, which is the last one. Then the next cycle begins. Okay, so this one is a bit depth. In depth, there's a lot of things to it. And then fertilization. Both cases are given. We have to understand the graph. LH and FSH will be at peak during ovulation, and estrogen is also at peak during ovulation. The progesterone will only form when the double corpus luteum is there, and estrogen is responsible for the proliferation, and the progesterone is responsible for the maintenance of estrogen. Now these are blood vessels, if you're wondering, and after this, after the corpus luteum degenerates because there is no fertilization, there will be no progesterone to maintain it, so it will become unstable, and yeah, menses are formed. That is, it will literally break down. So the next cycle begins, and that is menstruation. That's the next one is menstruation itself. So yeah, menstrual phase, that is the menstruation itself. Menstrual flow occurs and it lasts for 3 to 5 days. Results due to the breakdown of endometrial lining of the uterus and its blood vessels. It forms liquid that comes out through vagina. Yes, that's true. Then menstruation only occurs if the released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy. However, it may be also be caused by stress and anxiety, not anxiety. Okay, poor health, etc. Some other underlying causes. I did not know that actually. Menstrual phase is followed by follicular phase, so this can be could be an a significant indication that maybe there could be pregnancy for that female if there is a lack of menstruation. However, it can be caused by poor health and stress, which can be the thing. And then menstrual phase, looks like my battery is literally on gonna die, but no, it's not. because of the charger I kept. The menstrual phase is followed by follicular phase. That's the proliferative phase. Sorry, during this phase, the primary follicles. Okay, this one is new. I didn't know that. The primary follicles in the ovary grow to become a fully matured Graafian follicle. Oh yeah. Okay. Okay. Simultaneously, the endometrium of the uterus regenerates through proliferation. Yeah, that's true because that is done to the help of the estrogens released. These changes in the ovary. Okay, who releases estrogen in the ovary? Just tell me that. Like, who exactly does it? Like, is it the Leydig cells? Who is it? Who is responsible for release estrogen? Like, I have a doubt based on that. Like, who's that thing? Granulosa cells of ovarian follicles. So you're saying, telling me that like Pika cells stimulated by? Oh, okay, okay. I mean, there are Leydig cells too, right? So what? I have no idea about that. Okay, so it's granulosa cells, Leydig cells, with the help of FSH. That's confusing actually. Okay. I mean, that's not the higher biology I'm right now learning, so let's not go through that, you know. Then, oh yeah, okay. So simultaneously, the endometrium, okay, it regenerates to proliferation after it revises. Secretion of gonadotrophins increases gradually during follicular phase and stimulates follicular development as well as secretion of estrogens by the growing follicles. Okay, okay, you know what? As well as estrogens is being secreted by the growing follicles. That's what I wanted to know. With the help of granulosa cells of the growing follicles, okay, both LH and FSH attain a peak level in the middle of cycle about 14th day. That's why the ovulation takes place. Okay, so you can say that these two are the ones that are usually useful in ovulation, and estrogen is the reason why, because first of all, these pituitary hormone releasing hormones stimulates the follicles, granulosa cells, and that's why it forms the estrogen. I don't know which one was it, FSH. Why is it FSH though? Like, why not LH? Because LH is the one that is stimulating the Leydig cells and forming androgen in the males, but how is it not that in females? That's the thing. But yeah, it says follicle stimulating hormone, so it will stimulate the follicles of the granulosa cell. So technically, that's also possible. Okay, I mean, yeah, okay. Then what else? Rapid secretion of LH. Okay, they're like, this should be mentioned in this point, by the way. Ovulation, ovulatory. Rapid secretion of LH leading to maximum level during the mid-cycle called LH surge induces rupture. So the rapid rise of the LH in the ovulatory phase, ovulation attaining the peak, such that LH surge takes place, which induces the rupturing uh, of the what of the graphene follicle, releasing the secondary oocyte, which will undergo the secondary meiotic division to form ova. That the ovulation ova is ovulatory phase is followed by the luteal phase. Okay. The remaining parts of the Graafian follicle transforms as corpus luteum. The corpus luteum secretes large amount of progesterone, which is essential for maintenance of endometrium. Such an endometrium is necessary for implantation of the fertilized ova and other events of pregnancy. Yeah, during pregnancy, all events of menstrual cycle stop, and there is no menstruation. In absence of fertilization, the corpus luteum degenerates. This causes disintegration of the endometrium, leading to menstruation, making a new cycle. In human beings, in human beings for females, menstrual cycle is around 50 years of age, called menopause. Okay, that's the whole part. Let me just have a quick recap. So, what did we study? we studied about the menstrual cycle, which is an interval of repetitive interval of 28 to 29 days taking place from one starting from one menstruation after to another menstruation. Here, it consists of the menstrual phase, the ovulatory phase, the proliferative phase, then the ovulatory phase, then the luteal phase. In the let's say in the for menstrual phase, the menstruation takes place for 3 to 5 days, where the breakdown of endometrium lining and also with the small blood vessels in the form of liquid is taken out through the vagina, and this takes place. Okay, this does take place. Okay, of course, and then this menstrual phase. I think that's what I remember. Yeah, this happens only when the fertilization hasn't taken place. But yes, lack of menstruation can also be an indication that there's pregnancy. But but the due to poor health or stress, it can also induce the menstruation. That's one such idea that we can get from this. And then another thing about this is that after this phase is the follicular phase or the proliferative phase, where the primary oocyte will develop from primary follicle to Graafian follicle. In this stage, the follicles developing will have granulosa still stimulating estrogen, which would gradually increase, gradually increasing, helping in the formation or the proliferation of the endometrium in this phase. Through which the yeah, we can say that is also done by growing follicle, and then follicular phase. Yeah, these changes in the ovary and the uterus are induced by changes in the levels of pituitary and these changes are induced by the levels of the changes in the both ovarian and the pituitary hormones. The FSH and LH hormones are also increased gradually during the follicular phase. Uh, yeah, and stimulates follicular development. Okay, that's one thing. As estrogen grows and as forms, and then the luteal phase. The remaining parts, ovulation. The rapid rise of the LH, which is known as LH surge, induces the ovulatory phase, which is the ovulation, which is the rupturing of the Graafian follicle. Now, what happens next is that this ova is released, so no fertilization takes place. That's one thing about this, and the corpus luteum starts degenerating, resulting in the breakdown of the starting the second time starting the another cycle of menstrual cycle, so starting the menstruation of the other menstrual cycle, and that is known as the luteal phase, that we can say. This happens due to the absence of fertilization. That's one thing. uh, yeah, then this sees in human females, the menstrual cycle will cease around 50 years of age, known as menopause, and it starts during puberty, which is known as menarche. Now, fertilization. It can only occur if ovum and sperms are transported simultaneously, or else that's the reason why sometimes when the copulation has taken place but the fertilization hasn't. The ampullary isthmic junction. The ampullary isthmic junction is the site where the fertilization takes place. Sperm comes in contact with the zona pellucida layer of the ova, induces change in the membrane that block the entry of additional sperms. Yeah. So if once the sperm enters into it, the acrosomes which are containing into it will induce the change in the membrane that blocks the entry of the additional sperms, and then it ensures that only one sperm can only fertilize an ova. The secretions of an acrosome helps the sperm enter. This induces the completion of the second of the hell of the meiotic division of the secondary oocyte. Wait 1 minute. I feel like the timer has ended already, right? Come on, just tell me. Only 4 minutes left. Oh man. Like, bro, come on, bro. Like, I, I literally did not do anything this time. Like, I'm just focused, fully focused. Anyways, implantate. Okay, no, where was I? Yeah, due to the help of the secretions of these chromosomes helps the sperm enter and fertilize with the ova, thus blocking the entry of the other sperms, ensuring that only one sperm can enter the ova. And then the second meiotic division. This induces the complete division. This induces the completion of this meiotic division of the secondary oocyte. The second meiotic division is also unequal and results in the formation of second polar body, which will degenerate again, and haploid ova. I think it will degenerate. I'm not sure. Because what will be the use of second polar body? It will not do anything like first polar body. And then this is forming the haploid ova that is rooted. The haploid nucleus of the sperms and that of the ovum fuse together to form a diploid zygote. So this induces the completion of the full meiotic division, where the second meiosis takes place, in which the secondary oocyte will divide into a second tiny polar body and the large ova. I would say ova and a haploid ova. The haploid nucleus of the sperm and the haploid nucleus of the ova would fuse together to form the diploid zygote. Mitotic division of zygote takes place as it moves through the isthmus, which is known as cleavage. So, what is cleavage? When the uh, man forms 2, 4, 8, 16 daughter cells called blastomeres. The embryo at 8 to 16 blastomeres is called morula. The morula continues to divide and transforms into blastocyst. Oh man! Oh man! Oh man! Anyways, what am I doing? So mitotic division of the zygote takes place when it moves through the isthmus region of the fallopian tube, which is known as cleavage. As it divides, it moves into two cell stages and four cell stages, 16 cell stage called blastomeres, forms 2, 4, 8, 16 daughter cells called blastomeres, and the embryo with 8 to 16 daughter blastomeres, the embryo consisting of 8 to 16 uh, blastomeres is called a morula. As the morula further develops, it forms the blastocyst, which will further go into the uterus to undergo implantation. The blastomeres in the blastocyst are arranged into an outer layer called trophoblast, an inner group of cells called inner cell mass, which is having this totipotency as I remember. Is it given? Maybe the last one. What does this say? What is menstrual cycle? It is the cycle of the. It is a cycle of an interval where it starts from the first menstruation and then repeats the cycle and to the next menstruation. बोल सकते हैं. Which pituitary hormone regulates menstrual cycle? Uh, FSH and LH, right? Both. It says hormones, so FSH and LH. I'm gonna say that. How is poorly spermly prevented in humans? Oh, wait. So basically, what we can say is that one sperm will fuse with one ova. So that one sperm fusing with one ova, it's because that the acrosomes induces changes in the plasma membrane of the ova, blocking the entry of the other sperms, ensuring that only one sperm can enter to it. And we also studied about the secondary meiotic division. That's not given here. So, name the site of fertilization. List out the isthmic ampullary junction. List out the order of events after the sperm encounter the ovum. Basically, what happens is that its zygote will further divide to become embryo, which is containing two cell stage. This embryo that moves to zygote will further go into development into the mitotic divisions, and while it moves to the isthmus, that is known as the cleavage, where the zygote will develop into the embryo from 2 celled, 4 celled, 8 celled to 16 cell stage, and so on. The embryo with 8 blastomeres to 16 blastomeres. Okay, the daughter cells seen in embryo is called as blastomeres, and 8 to 16 blastomeres and the embryo is known as the morula. As the morula further develops, it forms the blastocyst and it moves through the uterus where it will implant on it. Implantation will take place. Now, what happens after the implantation? We'll check that out. The morula continues to divide and transform into blastocyst. The blastomeres in the blastocyst are arranged into an outer layer called trophoblast and an inner group of cells called the inner cell mass. The trophoblast layer then gets attached to the endometrium. Okay, the inner cell mass gets differentiated as embryo. Man, I feel like the time has already ended. Yeah. Man, 2 hours just to complete these two chapters? That's not enough, right? Man, what? Oh, okay. We are gonna come. We are about to complete this. In fact, just one more lecture note, and then complete this. These are useful, by the way. Like you don't need to go much through textbook. Even though I had gone through the textbook, like I read the textbook, except for reproductive health, I have to go through that. Reproductive health also has some things in textbook. I might have to go through that one thing. The remaining parts we'll deal with that later. Like first we'll complete the notes. It shouldn't be taking much time either way. Bro, the more time that we need to give next is for the genetics. Like I'm scared. Like literally, there are 9 notes, 9 PDFs just for that, bro. Like yeah, I don't need to go for molecular basis of inheritance. I'm confident that I've studied that, but then think about it. Oh yeah. That's one thing. Okay. Okay, let me just study implantation once because implantation is one thing. Now the blastomeres in the blastocyst are arranged in an okay. Okay. So the blastomeres in the blastocysts are arranged into an outer layer of trophoblast and the inner layer inner cell mass. Now the trophoblast, as it reaches the endometrium of the uterine wall, endometrium, it will attach to it, where the trophoblast will attach to with the endometrium, and the inner cell mass will get differentiated with the to become the embryo, to embryo, and then what else? The uterine endometrium will rapidly divide more. Rapidly go under mitotic division for more cells to cover the blastocyst, and thus the implantate. As a result, the blastocyst becomes embedded in the uterine wall, or becomes embedded in the endometrium of the uterus. This is called as implantation. It's crazy that how the body works almost so like perfectly, man. I'm like, thanks to God, mashallah, long about it. Allahu Akbar, Subhanallah, Alhamdulillah. How many views did I get, by the way? I'm just confused. Cool, let's go, man. Nobody's watching. I like it because the type of thing I'm studying right now is a bit too not comfy. But I have a suspicion. I don't know why. Please, YouTube, don't take this down. Please, YouTube, please, please don't take this down, YouTube. Please, for the sake of God. Okay, let's go. It's still there. I felt like I also feeling something is wrong. Like why is there no person coming up? But then that's also fine for me because right now I'm studying this thing. Like the problem is that I have no problem in studying this. It's just like a chapter that I'm studying. It's important biology, but then there are some people who do this nasty freak stuff like which I don't want to discuss about. So my bad. Anyways, let's move on to what else. Yeah. Once we complete this, I'm gonna restart the timer to 1 hour. I think at that time we'll complete this chapter and the reproductive health one easily. The next chapter only has 3 lecture notes, but then after that is the main big deal, dude. Like that's gonna be crazy. Do you want me to like first do the biology textbook questions, then the like diagrams, or what do you want? Like I feel like first doing biology textbook questions, then the last like diagrams. We'll practice diagrams later. Like what else do we need to draw in diagrams? Like sperm, ovary, then the Graafian follicle, 3. 4, the schematic representation of both the gametogenesis processes that we see in males and females, so 5. And then the double helix structure. So 6 things to draw. That's pretty much a lot, bro. Last year in 11th standard, we only had two diagrams to draw. That's a lot. Oh god. Honestly, semantic representation is like a flowchart, so that's one thing. What else? Oh yeah, okay. What else I was saying? Let me just look at this because okay. After attachment, the uterine cells will rapidly divide, covering the blastocyst, and in which the blastocyst gets embedded in the uterine wall, that is the endometrium of the uterus. This is called as implantation. Now I'm confident that I can do this. Let's move on to the other one. And before that, I'll also start another timer. Let me start another one. A timer will be playing a really good role in today's live lecture, I believe. Because today, I think we are not going to take any breaks because, we have to lock in now. 2.5. Only one lecture remaining for this chapter to be completed. This one will take some time. Yo, what's this? Dang. I did not notice that. Okay. Can you guys see this first of all? I want to check. No, you cannot. So, if I do this, 2.5. Oh wait, did I not show that 2.4? Oh, I did not. My bad. That's my bad. I did not check that out. Okay. 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 Let's move. Why is there a cursor? Yo, what? Why is this separate? Okay, okay, okay. Let's go. So, pregnancy and embryonic development. Let me use a mouse because it will be much easier for me to scroll and do that work. Mouse is much easier. Like my table is income. My table is so free right now. I can just use it. My phone here. Okay, I need to put it here. My bad. So, in this case, here we go. Alright, this is much easier. After implantation, the finger-like projections appear on the trophoblast. Yeah, this one will take time. This one is not easy for me. This one will take time. This is not easy. This one is a challenging thingy, actually, if I'm being honest. Uh, then this one. Yeah, this will take time. Okay. Okay, wait. The important topic I remember is that the milk produced during the initial few days of lactation is called colostrum, which contains several antibodies, absolutely essential to develop the resistance for the newborn babies. Then process continues, resulting in stronger and stronger. The resulting in the expansion of the baby out of the uterus through the birth canal is known as the parturition, and the mammary glands undergo differentiation during the pregnancy and starts producing milk up towards the end of pregnancy by the process of lactation. Okay, fate of stem cells. Okay, okay, so we need to explain this thing. I feel like this can come for like 3 to 5 months, maybe 3 months because this is not that big. What is colostrum? This will come. Did you guys see that notification? No, right? Yeah. Because okay, let me just give you that idea of notification sounds if you hear any. Because usually I use window capturing thingy because display capture will literally capture everything. Like it can even leak numbers. That's why I don't want to. Like if there is any notification where there's where my contact is not saved, so directly the number would be literally released or leaked. So that's not going to be a good thing. Finger-like projections appear on the trophoblast called chorionic villi. The chorionic villi and it's not going to be easy. Supply of oxygen, nutrition to embryo, excretory waste are removed by it to the help of the blood vessels, right? Produced by the embryo will be removed. Removal of carbon dioxide. Oh yeah, there is a diagram based on this. Let me check. This is a challenging thing though. Like I have very less idea about this part. Okay, umbilical cord. Wait, I think I forgot there was one thing I should check out. What was it called? Pubic symphysis. Okay. Hmm. That's ligaments. Okay. Hmm. Let's look at this. Finger-like projections appear on the trophoblast called chorionic villi. The chorionic villi, and that is okay. And the uterine tissue become interdigited with each other. So this is chorionic villi, this is the uterine tissues, and then jointly form a structure and functional unit called placenta, which forms supply of oxygen, nutrition to the nutrients to the embryo, removal of carbon dioxide. Okay, so the word interdigited would be a great way to actually say. So the uterine tissue of the mother and the chorionic villi of the trophoblast will be interdigited with each other to form a structural and functional unit of connection between the embryo and maternal body known called placenta. The placenta helps in supply of oxygen, nutrition, and also in the removal of carbon dioxide and excretory wastes. Placenta connected to the through an umbilical cord, which helps in the transport of substance. This is the umbilical cord with its vessels. This is the fetus, embryo, breast, placental villi. Hormones, human chorionic gonadotropin, human placental lactogen, estrogens and progestogens, etc. Laxin is secreted by ovary. Oh yeah, we need to know about this one too, right? Yeah. Ectoderm, mesoderm, endoderm. Totipotency, the potential to like give rise to like, the fact that this means totipotency is literally known as like totality. That's literally me, man. Like if you don't know what totality means, it means anything or everything. But according to philosophy, it has a bound, limit bound, but infinity does not. It just has limitless bound. That's what it means. According to philosophy. I'm not a humanities student though, or I don't even know whether people study philosophy in humanities. Like, I just know that. Like I think I searched it up in Google once. I was like thinking about which can be like something greater than totality. It could be infinity. Infinity is also a great name, but other YouTubers also have this name. That's why I chose this. Like I know that there are some YouTubers with these names, because this name is so unique. The username that I chose is so unique that alhamdulillah, because of God, I was able to do this. Like, people can easily find my channel even through search page if they just type totality high. They'll either see my channel with my videos, or you can also see some videos of Robert W collaborating with me. That's one thing, you can actually search it up and find it out. Human chorionic gonadotropin hormones. Okay, placenta acts as an endocrine tissue, so it will HCG, HPL, estrogen, and progesterone. Lactogen, oh no, that's not lactogen. Then, oh yeah, one thing is that human chorionic gonadotropin and human uh, placental lactogen secreted by the ovary is relaxin. Okay, I did not know that. Okay, I need it. That's another thing I needed to know. So that will help me now. During pregnancy, the levels of other hormones like estrogens, progestrogens, cortisol, prolactin, thyroxine, etc. are increased, supporting the fetal growth. So estrogen, progesterone, prolactin, thyroxine, and cortisol, etc. These helps in the supporting in the fetal growth, metabolic changes in the mother, and maintenance of pregnancy. Prolactin, thyroxine, cortisol, just estrogen, progestrogens, like those hormones, will help in the supporting of the fetal growth, the maintenance of pregnancy, metabolic changes in the mother. After implantation, the inner cell mass embryo differentiates into ectoderm, mesoderm, and endoderm. These three layers give rise to all tissues, organs, and nodules. The inner cell mass contains cells called stem cells, which have the potency to give rise to all the tissues and organs, known as totipotency. So, the major features of embryonic development. This will take time, guys. This one is a bit time taking. I think I cannot edit it because this is not my page. Oh, I didn't notice that my mustache has grown a lot. Mashallah. I didn't notice that. Like literally, I can literally stretch this thing out. Anyways, yeah. Chorionic villi of the fetal growth, maintenance of pregnancy, helps metabolism, changes in the metabolism, metabolic changes in the mother metabolism. The inner cell mass embryo differentiates into ectoderm, mesoderm, and endoderm. These three layers give rise to all tissues, organs, and nodules. Okay? Potency. It has the potency to give. After 1 month of pregnancy, embryo's heart is formed. Fetus is developed limbs and digits. Most of the major organs are from limbs and external genital organs are all together. The first moments of fetus and the appearance of hair on the head are to which lips. Fine hair, eyelids separate, and that is parturition. Actually took me fifth. I don't know who's watching me. I don't know. There's someone watching me. Hi there, man. Hi. How are you doing? Oh, I'm just studying. After 1 month, the embryo's heart is formed. It is fully developed. Its limbs and leg digits. So first month, heart is formed. Then first month, right? Second month, limbs and digits are formed, fully formed. First trimester, organs are well formed, like especially the limbs and external genitals are well formed. Fifth month. Now we're skipping to fifth month. First moments. Oh, okay, first moments. So this is like end of each. Only during the fifth month, the first movements of fetus and appearance of hair on the head are usually observed, and end on the second trimester. The body is covered with fine hair, eyelids separate, and eyelashes are formed. By the end of the ninth month, that is the trimester. You what? Okay, trimester. Uh, the fetus is fully developed and is ready for delivery. That's parturition. This thing. It is a neuroendocrine mechanism. Yeah, this one I have to explain. Let's do it, guys. You know what? Let's just do it. I want them to give me like small marks for based on short marking questions based on this second chapter, the reproductive health. Ouch. I want to again go through this, so this will take time, guys. 1 minute. This part will take time. I'm just going through that. I'm thinking. Okay. Oh, second trimester. Oh, okay, okay. There is a difference with this one and this one. I did not notice that. This is important. That's why I'm going to. Then we'll go through this, which is also important. This is the weak topic I have right now in this chapter, if I'm being honest. That's it. And then this chapter is really strengthened up. Oh my god, come on, let's do it. Oh. Yeah. Okay, it's external genital organs. I have to be exact. That's one thing. Then here, first movements are observed and fine uh, hair on the head are usually observed. Observe appearance of hair on the head are observed. Here, fine hair is developed and with eyelashes formed and eyelids separating. It's crazy that eyelids and eyelashes are very small changes, actually, if I'm being honest, but they take place so late. I think, yeah, it's because there are the minor changes and the major changes should be there. That's why. I see. Okay, so I feel like I'm confident with this one. This one. Okay, it's a supply of oxygen, supply of the food, and then that's nutrients to the embryo. It's not food. Supply oxygen and nutrients to the embryo, or removing carbon dioxide, and uh, you know, what do you call it? Removing of. Oh no. Let me keep it here. Removal of the excretory waste from the embryo. Okay, let's do this for the last time because this topic is very confusing. I don't know why I confused with this topic, even though it's easy. Uh, then we'll move on to this, the last one, the last topic of all the chapter. This one is very important. Like I can also see that graph. What else is important? This thing. Yeah, this is human gonadotropin. This is human prolactin. Sorry, human placental lactogen. This is estrogen and this is progestogens, etc. Yeah, because this is an endocrine tissue. That's why. And yes. So first of all, during the first month, the fetus develops its heart. Fetus heart is developed. Then, because it's really important, very important organ, by the way. Then the fact is, the brain takes full development after 25 years. Like not, like literally, that's crazy, bro. Like even after birth, you have to wait for your the brain to fully develop. It develops after 25 years. That is really crazy. Like you can just literally say that my brain is not developed if you are under 25 years. That is crazy, bro. Whoa. Thinking about it. But you feel like your brain is doing great. Your brain is doing that. But that's fine. That's actually great. But wow, like over 25 years after that it's completes its full development. That's how it matures. That is crazy, bro. Never mind. What am I doing? Just remember that brain is consists of fat, bro. What? The brain is consisting of fat. Wait, there were 5 weaves? Oh my god, 7 weaves. What in the world? How come did we get 4 weaves that fast? Like, what? Okay, at least it's at this topic. It's not bad at all because the previous topics were like something which is very sensitive to talk about. At least this topic is fine. We can talk about it because even kids know that this happens. So that's fine. Okay, so human pregnancy lasts for like 9 months. And I don't know if I can age restrict this thing. Like I want this to be only for those people who are watching above 9 years. Like no, I want people who are above the age of 13. You know what? Let's say 13. Like now you may be like, wait, what? Chill out. I am third. Like bro, I cannot even show it to like those 9 years old or 10-year old people too, right? Like literally, if I tell you that yeah, there are kids that are of like age of 1 to 12. They're literally kids. 13 is the age where they like become teenagers. That's why. So 13 and above is the one thing I want to get. The fetus develops limbs and okay, fetus develops its limbs and digits. The first feelings or heart was that okay. At the end of the first trimester, what happens is that majority of the organs, that the major organs are developed, well, or like the limbs and the external genital organs are well developed. Then what else is there? Um Yeah, the in the fifth month, what happens is that the hair starts appearance of hair in the head, and then also the first movements are observed. Then second trimester at the end of that is during 5 month, fifth month by the way. But for the next one, at the end of the sixth trimester, sorry, second trimester, um, what happens is that the eyelids are separated, eyelashes are formed, and fine hairs develop. And then the last one, that's the last trimester, end of third trimester or ninth month, the fetus is fully developed and it's ready for parturition, ready for delivery. Okay, so yeah, the period about average duration of human pregnancy is about 9 months, which is called the gestation period. Not much talked about. Vigorous contraction, by the way, this is the gestation period that we were talking about. That's it. And causes expulsion. The vigorous contraction of the uterus at the end, in which it causes the expulsion of the baby out from the birth canal, is known as parturition. Or why do we call it a fetus? Now it's literal baby, right? Or we call it baby after the birth has taken place. Okay, so I'm gonna call it as fetus. You know what? Let's call it as fetus. The vigorous contractions of the uterus at the end of uh, the vigorous contractions at the end of the what do you call it, at the end of the pregnancy causes expulsion of the fetus out through the birth canal such that the baby is born. This process of delivery of the fetus, childbirth, is called parturition. Parturition is a neuroendocrine mechanism. Signals of parturition originate from the fully developed fetus and the placenta, induce mild uterine contractions called fetal ejection reflex, release of oxytocin from the maternal pituitary, pituitary. Parturition. So close, bro. So close. We're so close. I think we are already completed. Literally, I took half an hour for that. Wow, just for this thing because it just takes so much time. Because this is a weak topic, man. I cannot like deal with it. That's the problem. But I do know that the human reproductive health will only have main concepts as contraceptives, as they're really important. Like I've literally seen many neat questions only talking about contraceptives. So yeah, I think some people have messaged me in WhatsApp. I just saw that. Like did you ask teacher or anything? That's what. I have one doubt. You know what? 1 minute. Um, Let me take this page. Let me share this. How can I share it? Can I share it? Share it to ChatGPT. We'll first do ChatGPT and check this out. First, I need to crop this out, by the way. I didn't crop it. Okay. Oh, I found a question. I found a question. All right, all right. Okay, this is a good one. Let me ask teacher about this biology teacher. Yo, what? Yo, what? Oh my god, her description says Karma, the best teacher. Yo, she's a whole Gen Z, bro. What? How does she know these stuff? No, what? Man, why is she so casual with this? Yo, what is this? No way, bro. What does that say? Yo, what is that, bro? That's crazy, yeah, man. It's crazy. It's crazy to see those types of teachers who have those ideas of those memes. Like that's crazy. Let me message her and say, "Respected ma'am, uh, let me send that image."
Let me just put that explanation up. Is it? Answer to exist in this predicate. The mutant trait is shade black. Okay, wait. Mutant trait means recessive trait, right? Let me just check. Mutant trait is recessive. I have a great feeling. Dominant. Can be either recessive or dominant, but okay. So this is recessive as far as I know. Shade black. If it is recessive, okay, let me just okay, man, man, that was a crazy case, bro. I had to help. Okay, that was just out of topic. Let's move on, man. That's waste of time. Uh, like, what's going on? I just asked it out. That's it. I hope nobody's number is getting leaked here in this case. Okay, so the signals for parturition originate from fully developed fetus and the placenta. Why is there a single take? What? I don't know why people have to like Wi-Fi off right in this case. Graphs are you talking about? I have no idea of type of graphs you're talking about. DNA double helix structure. Might come. Sperm. Oh, I think it's only for labeling. Only for labeling. DNA double structure might come. Bro, she's practicing that? No way, come on, bro. That's so much time taking. Yo, that is really time taking, bro. Yeah, DNA double helical structure might come, sperm, and then no, I think it's only for labeling. Also practice giraffe and follicle just in case diagram. I don't know what he's typing. He's taking a lot of time. Till then, I'll just check this out. Yeah. So where is Microsoft Edge? Here we go. Induced uterine contractions called fetal ejection reflex. Signals for parturition originate from fully developed fetus and placenta. It's a bit disturbing when people like do WhatsApp messages to me, but what can I do, man? I have to help them out. Like one person was like, "Did you ask the teacher about this doubt?" So I had to right now ask the teacher because yesterday we were like discussing about this at late night. Plus nutrition. Phosphate is connected. Man, like bro, that was so funny. What I was doing, I was just replying to him. He thought I was offline, and he just literally like busy. Yes, but phosphodiester bond is the connection is the bonding from the phi prime carbon to and p, who's that? Okay, and p to 3 prime carbon of the other nucleotide. Nucleotide. But phosphodiester bond is the bonding from the 5 carbon carbon to phosphate. Draw 3 uh, nucleotides for each strand. For each strand. Man, my time is going on. Okay, fetal ejection reflects release of oxytocin. Acts on the uterine muscle, causes stronger uterine contractions, further secretion of oxytocin. What is this? 1 minute. Let me just help him out, guys. Like, let me edit this thing. How can I edit this? Guys, why can't I edit this? Moment. Yeah, got it, bro. Shows the phosphodiester bond. Got it, bro? It's up to. How is he actually replying? What? But the phosphate group is already there, right? Then why should we mark it again? Anyways, I got it. Not the group, bro. Like, bro, come on, man. I hope he knows about N-glycosidic bond. It's N-glycosidic bond. Jason. Yep. Sorry, Jason. Yes. Hey, man, my time. Oh my god. Like, can you chill out? Induce mild uterine contractions. Okay, so induce mild uterine contractions called fetal ejection reflex. Finally, please, now I can study.
The process continuation resulting in stronger and stronger contractions leading to the expulsion of the fetus out through the birth canal known as parturition. After following the parturition, the mammary glands undergo differentiation during pregnancy. They would start undergoing differentiation during pregnancy and start producing milk, which is known as the lactate towards the end of pregnancy by the process called lactation. Yes. Okay. Okay, so what happens is that it is a neuroendocrine mechanism originate from fully developed fetus and placenta. Okay, so where the oxytocin is reduced, yeah, fetal ejection reflex from maternal pituitary and then acts on the oxytocin it's on the uterine muscle, causes strong uterine contractions. This process is actually easy if you look at it normally because it's a repetitive process. You can look at it. What happens is that it starts from fully developed fetus and it starts from the placenta. It does mild uterine contractions from the like in the uterine contractions for the expulsion of the body, which is known as fetal ejection reflex. Just know the name, and then if you get from here, it gets easier. The release of oxytocin from the maternal pituitary. That's one thing, and that helps in attacks on it and then causes stronger uterine contraction. From that, it will further secrete oxytocin and then more of the stronger uterine contractions takes place, and that's how expulsion is done. And also remember, guys, this is a really painful process. Like mother undergoes a lot of pain here from this. More oxytocin released, more stronger and stronger contractions, so that the baby can come out from through the birth canal. You can just think about it. The cervical canal is very very narrow, guys. It's very narrow. Vagina is a bit okay, fine, let's say that, but the cervical canal, like I've studied this in the diagram, you can see that the cervical canal is very narrow. That's the thing. Yeah, so it starts off from the placenta and the fetus, like putting some moments in the fully developed fetus and the placenta, where it will start mild contractions from through the uterus. From the uterus, with the help of fetal ejection reflex, more oxytocin is released at the anterior pituitary, which will act on the endocrine sorry uterine walls to like contract further and further secretion of oxytocin and stronger contraction, blah blah blah. Colostrum, it is basically after the following the parturition, the lactation takes place where the mammary glands will differentiate and produce milk. This milk is known as colostrum, but it is important to the baby because it contains several antibodies which helps in the resistance, which is absolutely essential to develop resistance for for born babies. What are the okay? So absolutely essential for newborn babies to develop resistance. What are the different layers of embryo? Trophoblast and inner cell mass, right? What are they given? We'll check. We'll check. What is the fate of stem cells? Well, stem cells are will give rise to all types of in tissues and organs in adults, such that have the potency to give rise. That's a potency known as totipotency or totality. Okay. Hey yo, what is going? Okay. Let me just take this out. Okay. Finally, man. Finally. Okay, wait. Just have a doubt on this one. What are the different layers of embryo? So let's check that out. We completed the chapter. Let's go. Uh, I don't need that slash here. Double slash it. Germ layers. Oh. Ectoderm, mesoderm, and endoderm. How can I forget that? Yeah. Ectoderm, mesoderm, and endoderm. I cannot forget that. Finally, let's move on to the next one. No breaks, guys. Trust me, I'm not actually having any breaks. Even my friends are just helping, just giving me some questions and all. Okay, total health, well-being in all aspects. That is PEBS or BEPS, behavioral, emotional, physical, and social. Let's go. Create awareness among people about various reproduction related aspects, provide facilities and support for building up a reproductively healthy society. So RCH programs. Well, actually, it was initiated. India was the first one to initiate, if a type of reproductive health program known as family planning program, which was in 1951, and then it got renamed as reproductive and child health care RCH programs, where creating awareness among people about various reproduction related aspects. Uh, there are a lot of ways you can actually discuss about this. Oh, shoot, population explosion. What is this, bro? Okay. Okay, so we are starting from here itself. That's this is the second last topic as far as I remember, and why is it here? This is first lecture, guys. Chill out. Okay, there's a lot of things in the first lecture, man. Okay, so if you don't know, I'm just going through the notes for the first time because usually I don't go through the notes. I usually go through the textbook. So that's one thing. So yeah, audiovisual and print media to the non-NGOs, parents, relatives, and teachers and friends. Discussing it with friends will be very weird actually because they won't take it seriously. That's funny. They'll be like so dirty about it. That's the thing. Information about reproductive organs, adolescents, and related changes. I mean, this is fine, and discussing it with relatives is even more weirder if I'm being honest. It's cool information about reproductive organs and adolescents and related changes, STDs. Yep. Educating people, especially young couples and those in marriageable age group, about available birth control options. Yeah, like contraceptives. You can say, care of pregnant mothers, postnatal care, and perinatal care is also there of the mother and child. Equal opportunities for the male and female child. Yeah, like there are some female feticides, which is the thing that they will abort the female fetus, which is not good at all. People usually want male child. That's what they want to do, but that is very like gender inequality. That's literal gender inequality. So we need to like have an equal opportunities or equality for both the genders. That's why there is an awareness for this too. Then it should be prenatal care because prenatal care is also mentioned in the textbook. Statutory ban on amniocentesis. This is an important one, I believe. Uh, is this already highlighted? Like I didn't highlight it. A fetal sex determination test based on the chromosomal pattern in the amniotic fluid surrounding the development embryo. Developing embryo. Like the thing about this is that it's a very general chapter. Like I feel like they shouldn't be giving these types of questions. That would be very weird. Like why do we need to learn these types? We are studying biology. I mean, of course it's important to actually know the awareness and all that stuff, but do you think it's useful in like writing an exam? I don't know. Like this is so general. Like this is what you would usually study in social science. Like mm, I don't know why. Like you can study about amniocentesis, which is a bit biological. Then there are other things. Then the number, they might ask about the number though. That's one thing. Medical termination. This one was initiated in 1971, as far as I remember. It is also known as induced abortion, and to check indiscriminate and illegal. Yeah, there is also restrictions because we need to check indiscriminate and illegal female feticides. This is banned. Uh, why MTP? Because it is considered relatively safe during the first trimester up to 12 weeks of pregnancy. That is 3 months. Oh yeah, this is disturbing trends. I'm going fast, guys. I'm going fast because I don't want to waste more time. Okay, so again, small revision for of what else, creating awareness about people about. Okay, so according to WHO, reproductive healthy means that it should be being well-being in all other aspects like physically, emotionally, behavioral, and social. Then reproductive and child health programs. Their main aim is to like create awareness among people. Uh,
Wait, teacher just saw that question. Oh shoot. Literally, she thought I was online. Oh god. Man, I cannot. It's a bit so. I don't like, asking doubts to teacher online. Like, it looks so. No, I don't like doing that at all. Like, it looks so very weird, man. I mean, I'd like doing it in coaching because it's like, you're having more privacy there. But of course, here there is a lot of privacy, but the teacher literally knows you. I don't like that actually, if I'm being honest. I want to be less known by the teacher so that I can like easily and nicely study. There is a problem with when teacher knows you very well. Like there's a specific problem with that. Like I know what happens with that. Like bro, that's not good at all. Creating awareness. Like I just saw the read receipt switching on, like blue color, and I was like, yeah, she's reading it. Let's just go offline. I don't want to mess it up. You may hear that notification sooner or later. Okay, someone's also watching me. I don't know that. I don't know who is it. Creating awareness about people, for the people about various reproductive aspects, providing facilities and support for building up a reproductively healthy society. To create awareness among the people about reproduction related aspects, audio, visual, and the print media, parents, relatives, teachers, and friends. Man, okay, fine. Okay, teachers, let's say fine. Re- Discussing it with relatives is diabolical, and then friends, that's even worse. Information about reproductive. Okay, let's be serious. For school, information about reproductive organs, adolescents, and related changes, STDs. That's the venereal diseases, or we call it as there are many names to this actually. STDs, venereal diseases. That's the name. Then we call it as yeah STI. STI also. Yeah. Then there is another name which is not popular. It is very less known. But yeah. And oh yeah, hi there, man. How are you doing, man? I can literally see a person literally watching me. I mean, don't worry, I cannot see you through the camera, but I just know from the control panel of my YouTube channel that there's someone watching me, and I highly appreciate you, man. Thank you very much. Also, care of pregnant mothers, postnatal and prenatal care, then equal opportunities for both the male and female. That is gender equality. Statutory ban on amniocentesis. She just read the question. I think she is reading the question. The question. I saw the read receipts turning blue, but she didn't respond yet. Respond yet. I will reach it. I'll reach out to you. Out to you. Bro, what happened, bro? Oh god, let me go. Yeah, literally, she's typing right now. She is typing it out. Okay. How can I like send this? Said about this. I know I cannot show my WhatsApp. I'm so sorry, guys. Like literally, I'm so sorry. But trust me, uh, because I cannot leak any number. I have some non-con like no some numbers which have not saved in contacts. So according, okay, he's reading it now. Like the person I just sent to. Man, I gotta go. Bit busy. As soon as possible. Alright. Sorry, I have to go now. I'm a bit busy. Respond as soon as you. Let me just like edit this. I need to edit it. It should say no. Okay, now that's fine. Statutory ban. Finally, man. I feel like the timer has already ended. Yeah, bro. Come on, bro. Like what? Ah, man. It's already 3 hours now. Looks like this will take a lot of time, guys. This is not gonna end so easily. Alright, good luck. Same to you. Same to you. Statutory ban on amnesia sentences if fetals. I think this is gonna be my weakness here. Like I feel like this chapter is gonna be my weakness. I don't know why. Incentives given to couples with small families, statutory raising of marriageable age of the female to 18 years and that of males to 21 years. Creating awareness about the use of various contraceptive methods. I see. Medical termination, pregnancy induced abortions. Government of India legalized MTP in 1971 with some strict conditions to avoid its misuse. Some restrictions are all the more important to check the indiscriminate and illegal female feticides. All right, okay, okay.
Let me get this out. I think we'll get enough time management for biology. That's what I know. Legalized MTP in 1971. To inject indiscriminate and illegal female feticides because it is considered safe during the first trimester, but it is a bit risky in the second one. That's what I remember. Oh yeah, this one I remember. People make jokes about this. The majority of the MTPs are performed illegally by unqualified quacks, which are not only unsafe but could be fatal too. Misuse of amnio, but yeah, this is a serious matter by the way, just in case if you don't know. Venereal diseases or reproductive. Okay, wait, why am I skipping that? Misuse of amniocentesis to determine the gender of the unborn child. If found to be female, it is followed by MTP, which is dangerous for both the young mother and the fetus. Yep, that's one thing. Based on chromosomal pattern in the amniotic fluid surrounding the development. Based on the crown. Okay, so amniocentesis, amniocentesis, which is a fetal sex determination test, where the amniotic fluid, which is seen through the allosomes or test based on the chromosomal pattern in the amniotic fluid surrounding the developing embryo, for to legally check increasing. Uh, Massive child immunization. Okay, which has helped in okay awareness of problems due to uncontrolled population growth. Audio, visual, and print media. Then discussing it to parents, relatives, teachers, and friends. Then non-governmental agencies also talking and taking awareness about this. Then what not only that, but the taking postnatal care of the mother and also for the fetus. Then available birth control options. Yes, okay. Then equal opportunities for both for the gender opportunities for the child. Statutory ban on the amniocentesis, a 6 fetal determination test where the amniotic fluid is taken to test the chromosomal pattern around the developing embryo. Then the awareness of problems due to uncontrolled population growth. Then increased health facilities with better living conditions, a rapid decline in death rate. Yeah, this one. Population explosion, a rapid increase. Increased health facilities, rapid decline in death rate, decrease in mother mortality rate and infant mortality rate, and what else? That's it, right? Along with better living conditions, was it there in the textbook? I don't remember. Uh, okay, so nobody's here, and that's fine. Okay, so great awareness about the various contraceptive methods, statutory raising of marriageable age of the female to 18 years and that of males to 21 years, incentives given to couples with small families, medical termination of pregnancy, MTP, induced abortions, Government of India legalizing MTP in 1971 with some strict conditions to avoid its misuse. Some restrictions are all the more important to just indiscriminate and illegal female feticides. Guys, when do you want me to go live tomorrow? Like tomorrow, I will come early, like after school, and maybe at 11, no, 12 o'clock. So let's go live at 12:30. Is that fine? If the bus comes late, then it will be even more late. That's the problem, because the problem is that I'm a bus going student. So what happens is that whenever I like come back from, whenever like I search for the bus in school, it is not even there. Like it's not just there. Like I have to like wait for a while, then the bus comes in, and then I have to come here back. So it's like time taking. It does take a lot of time. So and sometimes we get so late that I don't know when I will be starting the stream that time. But yes, we'll get some booster, we'll get some more time to study for physics tomorrow, because physics is our next exam and we have to lock in for it. I feel like completing current electricity, because current electricity is something which is pretty much easy and new to me. And I don't want to give more stress whenever I complete after an exam after a day, okay, because that's one thing. A majority of the MTPs are performed illegally by unqualified quacks, which could be which could only not be unsafe but fatal. Two, misuse of could be fatal, because there is a possibility, weak possibility. Misuse of amniocentesis to determine the sex of the unborn child. If found to be female, it is followed by MTP. It is dangerous for both the end. So misuse of amniocentesis for the fetal sex determination. If found female, then the female feticide would be taken. It is followed by MTP, which is pretty much dangerous for both the mother and the fetus. I don't know why people actually like mess this up. Like, why do you want to do this, man? Like, you just because it is a female, you doing MTP. That is crazy, bro. Uh, then we got STDs: gonorrhea, syphilis, genital herpes, genital warts, hepatitis B. Then what else? There is a disease I remember. Yeah, trichomoniasis. That is the what is called? Wait a minute. Protozoan disease. Then this one, chlamydiasis, gonorrhea, syphilis. These are bacterial ones. This is a viral disease. This one is also a viral disease. AIDS. How can I forget AIDS? Except for hepatitis B, genital herpes, and HIV infections, which are all basically these all are, viral diseases, and yeah, except for genital warts, because genital warts is also a viral disease, but it is curable. But the thing about this is that these three are non-curable; they cannot be cured. They can be completely curable if detected early and treated properly. And what is this? Which yeah, okay, this one. Yeah. The symptoms. And the proper treatment, if not given, could lead to complications later, which include uh, PID, pelvic infectious diseases, pelvic inflammatory diseases, abortions. Oh, dang, that's the MTP there is basically stillbirths. Okay, minute. I still remember. Oh yeah, like that child is born. Yeah, I remember that. Infertility, literally cannot fertilize. Even cancer of reproductive tract. That's crazy. Ectopic pregnancy. This one is a bit crazy actually. What can cause a lot of problem in ectopic pregnancy is that usually these blastocysts, when it's fully developed, like after the zygote it goes to morula, then blastocyst, after the plastrum is divide, when it comes to the uterus, and then it gets implanted on it, so that the whole development, the gestation period starts. But here the gestation period, or the whole embryonic development, just starts at the fallopian tube, which is crazy, bro. So this is one such idea I remember. Then abortions, basically MTB, I guess say RTI, reproductive tract infections. Oh yeah, that one is also there. So STDs, STIs, venereal diseases, reproductive tract diseases, like tract infections. What kind? Oh, there's so many names for this thing, bro. Like four names. So gonorrhea, syphilis, chlamydiasis. Then what is that? Trichomoniasis, genital herpes, genital warts, uh, hepatitis B, and then what is that last one? HIV. How am I forgetting HIV? It's literally human immunodeficiency virus. Uh, yeah. There's no D though. That's one thing. Okay, so it can lead to pelvic inflammatory disease, ectopic pregnancy, stillbirth. Then what else? It can lead to abortions. Then what else did I mention? Yeah, cancer of the reproductive tract. Okay, we did mention all. No, infertility. I did not mention infertility. Yeah, I have to also talk about sterility. Sterility, where's sterility? What? Sterility is not given. This is induced abortions. Yep. Okay, so this one is there. Okay, I have to go through this part. Then I don't know. They didn't give me the value here. They didn't give me the value. I have to check through the textbook. The textbook has the values. Like I remember it. No, not I don't remember the values, but I remember that there are values given in the textbook. So I have to check that out. Yes, here it is. So, man, okay. So the world population was 2 billion in 1900s. Crazy. That time it was 350 million, I believe. Then 6 billion in 2000s. That was how much? How much? 6 billion. Okay, so it's 1 billion by 2000, and 1.2 for 7.2 billion. It's interesting actually. If you notice closely, there like there is a if you like divide the population with world and the Indian population, you're actually getting a factor of 6. Like I just noticed that. Like let's say okay, 350 by 2 is sorry, 2 billion by 350. It's not exactly 6, but it is somewhere around 6, like more than 6. No, my bad, less than 6. But then if you like divide 2 billion, it is with the Indian pop 2 billion. Okay, that 2 6 sorry 6 billion. With the Indian population in 2000, that was 1 billion. That is 6 again. That's 6, literally 6. Then it's closer to 6. It's actually lesser than 6. That's what I've noticed, the factor. But yeah, it's like some like varying lesser than 6. Like it's somewhere around 6. You can say. Then 7.2 billion in world population, 1.2 billion in this. So that's interesting actually, if I'm being honest. So 1900s, it was literally 2 billion. Then that was 350 million. Then 1.21 billion close in India during the 2000s and for Indian population, and the world population was 6 billion. That was a really crazy height. And then that was a crazy population increase because of increased health facilities. Yeah, I'm going out of context. No, I'm not. Then decrease in death rate. Then decrease in both the MMR and IMR. That is mother mortality rate and infant mortality rate. What else? What else? What else? Man. Oh yeah, 7.2 billion in 2000, the 2011 census, and for India it was like 1.2 billion. So right now people say that it's 1.4 billion. I'm not sure about that. The population growth was less than 2%. So according to the 2011 census report, the population growth rate was less than 2%, but that is not a good thing actually. Like it's like in fact it's great, it's fine, but it's fine. Let's say it's fine, but it's not that good. Create awareness about the use of contraceptive methods, statutory raising of marriageable age limits from 8 to of females to 18 years and 21 years for males. Then incentives given to couples with small families, medical termination, MTP induced abortion to check indiscriminate and illegal female feticides. Let me just keep my like that. Okay, now I have to just check the disturbing trends. And that's it, guys. This part is done. I know I'm taking so much time, but this is general, man. This is too general. Like professionalism is expertise, professional expertise, then good health facilities. Like there was a before I remember. I just know. I don't know which one was it. Yeah, strong in fact structural facilities, professional expertise, material support. Okay. Misuse of amniocentesis to determine the of unborn child if found to be female. It is followed by MTP. It's dangerous for both the young mother and the fetus. Nobody's watching. Okay, that's fine. I like it. That's fine. It's great. Abortion, stillbirths. Okay, PIDs, that is pelvic inflammatory disease, abortion, stillbirth, ectopic pregnancies, infertility, cancer to the reproductive tract, and beyond cancer. The 1, 2, 3, 4, 5, 6, 6. Yeah, that's what I wanted to know. Gonorrhea, syphilis, chlamydiasis. I think that's correct. Yeah, that's correct. That's correct. That's correct. Yeah, chlamydiasis. Then genital herpes, genital warts, HIV, AIDS, basically. But what else? What else? Oh. Hepatitis B. And the last one. Last one. What is that last one? Come on. Come on. Come on. Tell me. Yeah, the trichomoniasis. That is the protozoan disease. I forgot. Then timely detection. Yeah, that we discussed. I think we are confident in this. Such restrictions are all important to check and discriminate. Uh, with this one. Uh, a majority of the MTP is being performed by the unqualified quacks, which are not only unsafe but could be fatal too. Misuse of the amniocentesis could determine the sex of the unborn child. If found to be female, it is followed by MTP. It is dangerous for both younger mother and fetus. Yeah. Okay, so finally, bro, this took me so much time. Let's move on. You have already completed this part. Now let's go on to these parts. I think this part is strong, so we can do this. Periodic abstinence, okay. Oh yeah. I remember this now. Okay, lactational amenorrhea. There is another one called as coitus interruptus. I don't know why it's not mentioned. There is another one called coitus interruptus actually, where you basically withdraw the bins out of the vagina so that the ejaculation doesn't take place, and then basically insemination shouldn't be taking place. For those who don't know what insemination is, it is transport of gametes from the male external genitalia to female reproductive tract. Then we got periodic abstinence. Yeah, I wanted to know about this. I forgot about it. That's why. So revision is very important, man. Okay, so this shouldn't be taking much time. We can do this. Lipis loop, non-medicated IUD, copper releasing ID is CUT, CU7, and Amultilode 375. Progestasert and LNG20 hormone releasing IUD. I want to know which one is. Okay, this is not mentioned in deep, so I'll go through the textbook for this one. Diagram, cervical caps, and vaults, and females, not families, females. Barrier methods, yeah, that is a really one of the ideal methods, they say. Because it can literally prevent from physical meeting of coitus, with the help of the barriers. It's like, you can use it by yourself. It is easily available. It is, not that it is for this one, it is not reusable for the condoms, but for the females it is reusable, so that's one thing. Another thing about this is that it is you can be done in privacy, like like you don't need the doctor's instruction to do this. It's very easy to do, like keep on. That's what I remember. And then what else do they mention about this? Yeah, New Road, a very famous brand of this. I think it's a governmental brand, uh, which was like giving products of condoms for males. Okay, then yeah, diaphragms, cervical caps, and walls in females. That's a barrier for the females, but the problem with this is that these are not capable of protecting the females from STDs. But this condoms can prevent the STDs for males, because the vagina is exposed. That's why it is said like that. That's why it is mentioned as, yeah, you cannot like actually do this. That's why one thing.
Non-medicated IUDs, yes. Okay, wait. Let me just learn the natural methods. From day to 17, day from day 10 to day 17 of menstrual cycle of female and females, the coitus should be avoided as the ovulation of the chances of fertilization are very high during this period. It is called a fertile period. I didn't know that. I didn't know that it was called as fertile period. That's something new. Absence of menstruation method is based on the fact that ovulation does not occur during the period of intense lactation following parturition. Okay, intrauterine devices, IUDs. It is said that it is the mostly used type of contraceptive device in India, and you know these three are the types: non-medicated IUDs, lipus loops. I think it helps in phagocytosis, like there are some types of cells which will engulf those types of gametes, like digesting, that's how it is done. But then copper releasing IUDs like CUT, CU7, multilo 375. What do these do? Basically, they release copper ions, and these copper ions are capable of suppressing motility of sperms and the fertilizing capacity of the sperms too. Then, yeah, then I want to know about this one too. Hormone releasing IUDs, progesterone and LNG20. If you don't know, it makes the uterus unsuitable for implantation and the cervix hostile to the sperms, like through the help of the ovarian hormones. Like that's what I remember about the hormone releasing IUD. Like that's one thing about this. And LNG20 progesterone here, and then CUT-CU7 multiload 375. Just remember the numbers nicely. Lipus loop, okay. Pills, oral administration of small doses of either progestogens or estrogen progesterone. Okay, it's progesterone estrogen combinations is another contraceptive method used by the female. Pills are to be taken daily for a period of 21 days, starting preferably with the first 5 days of the menstrual cycle. After a gap of 7 days, okay, this has a lot of side effects if you don't know. Then we got the Saheli. This one is very important. Like, they say that what is this, that non-steroidal oral contraceptive pill that was released by CDRI, or what is Saheli? Who released Saheli? Like, that's one thing about it. And then, if anyone talks about it, just remember that it is basically a non-steroidal. I mean, of course, you guys are not going to study about this, but I'm just telling using Feynman technique. It is a non-steroidal pill or oral pill, which is a once in a week pill. This type of Saheli pill, if you don't know, is basically having very few side effects and can be used and has a high contraceptive value. Yeah. What else is it used? Yeah. Then we got the implants and injections. This one has very less points in it. Basically, it is placed under the skin, and it can actually help it can help preventing conception. Is it called conception? Yeah, conception. I guess it's called as conception. Well, basically pregnancy for like a very large amount of time, for a long period of time. That's what I remember. I think it's 4 or 5 years. That's the major point I remember about this. You know, injections or implants under the skin. They're implanted under the skin. Like the silicone shells are there. Basically, those are the ones. It's not there in the textbook, I guess, as far as I remember. Uh, okay, tell you. Also, I noticed that there are no questions in this. Like, what? Maybe this chapter is not that important. The genetics chapter is important, though, guys. We need to know. Maybe we'll practice crosses there. That's what I know. We can do this, guys. We cannot give up. Then we'll do textbook questions. We'll use the help of AI with this because that's the fastest method I can do. Let's use AI. What happens with the use of it? Okay, I'm not like cheating or anything, but this will give us the idea of what kind of questions can come in the exam, and this can help us. So the textbook questions we'll be doing. Reproductive health. Now this chapter is the only chapter that I didn't study that nicely, that great. Like I studied the other chapters well. Genetics, yeah, there are some concepts which are a bit weak. We'll go through that, like sickle cell anemia, thalassemia. We'll study that nicely. We'll study that very well, so that we don't prevent the confusions. We'll type. That's the only thing which will be taking more time for. But yes, you know what? We are completing the whole thing.
it progesterone and estrogen? Yeah, or in combination with estrogen, can also be used by females as injection or implants under the skin. Hmm. Blocks gamete. Okay, this one. This is a permanent method where the recovering of to recover is very very difficult. You cannot actually like recover this. This basically what happens is that there are two types of surgery taking place for each different gender. For males, it is basically vasectomy, where the vas difference I believe is cut it out and then it is tied, so that there is no transport of gamete, and then for that sperms, and then tubectomy, where the fallopian tube is cut and tied for each the pair of these both. So remember that. So a small part of the vas difference, okay, small part is removed or tied up, whereas tubectomy, whereas in tubectomy, a small part of fallopian tube is removed. But thinking about it, I mean, I don't know about this, but what do you guys say? Like sperms, would they get collected in the vasectomy and then will they like do that? I mean, I'm not sure, but like the thing about this is that they do degenerate, so that's one thing. So okay, but tubectomy, like you can say that only one ova is released, so it's not like every time there are a lot of ovas coming up. So plus, if there are like they will also like get degenerated as far as I know. Maybe I'm not sure. This is my theory. Like I have no idea about it. I have a high chance of being wrong here. So yes, which one was the part I feel weak? It's periodic abstinence, where from day 10 to day 17, the coitus should be avoided, as it is those days in between where the high fertilization can take place. It is also known as the fertile period. Lactational amenorrhea. This one is the absence of menstruation after the parturition taking place. The intense lactation takes place, through which it basically prevents ovulation for a long period of time. Um, it does not occur during the period of intense lactation following parturition. So that's one thing, lactational amenorrhea. Then we got another one known as coitus interruptus, which has the highest failure rate among all these contraceptives. So that is, just withdrawing of the male genital, external male genital, which is elongated, and that is, basically withdrawn from the vagina, so that the sperms don't reach the ova. Then barrier methods. Yes, basically the males can have wear condoms, which are you know what, that was very weird to say. Okay, but still, these barrier methods, and sperms are prevented from physically meeting with the help of barriers, such that the condoms in males, the diaphragm, cervical caps, and valves and phylas acts as a barrier, so the blocking the gamete transport. Like the gametes, like the insemination is literally blocked here. It doesn't take place at all. So that's one thing. Then non-medicated IUDs, lipis loop. Yes, CUT, CU7, and multilode 375. LNG20 progestasert. What is this? What did they send me? Oh god. Oh, like really, Jason? But wait a minute. Sub horse. Sub horse 68. Sorry, I didn't see your message. But wait, what? What? Man, I thought I was gonna leak my number here. I was gonna leak my own number here. I thought, like, bro. Like someone literally have my contact not saved, and then like, bro, what? Sup horse XD. Sorry, man. I will just reply to you in WhatsApp. But wait a minute. Right now, I'm locked in. Periodic abstinence. Yep, that's done. Okay, the course one is important. Ovulation and implantation. Okay, I want to know them. This one is important. These two important. Here they're just avoiding the implantation and then the cervix hostile response. Okay, so wait. This will take time, guys. But at least we are studying all the contraceptives, and just one last lecture, and this chapter is also finished. Hooray, guys! Say hooray because we are doing great. This chapter is small, by the way. That's why. So that's one thing. I think the next one will be in vitro and in vivo fertilization. It wasn't covered, so the next one will be the that one topic, and then we'll complete it. This chapter is not that weak overall, but the problem is, it's very general, like very, very general. So I might have a weak spot there. Like, if any question comes from a general topic, then I might get cooked there. Because, mainly I don't like the thing about this is that I don't like general topics sometimes. I mean, even though it's interesting to see it and then this, but I don't like studying them. Like, I don't want this to come in exam, and it feels like you're literally studying social science. That doesn't mean I hate social science, but I studied a lot of social science and I have suffered a lot, so yeah. But social science is fine overall. It's okay. It inhib Okay, so wait a minute. Let me just think. So this each really, bro, he made a sticker out of it. Like, really? Literally no break, guys. We are doing great. Alhamdulillah. Yeah, I just went for a toilet. That's it, and then I might have to go for prayer too soon. You know what? Let's just complete this whole thing, man. We'll not give up. Sad part about this is that I'm not gonna get enough time for full biology work. Like mathematics is one thing. I just don't know why teacher wanted to do this thing. Like, why did she even wanted to keep this during exam days? Like, why can't she find another time to do this? Like, everyone is so busy, and you want to do this. Like, that is very dumb, man. I mean, I just don't like it. Inhibit ovulation and implantations, alter the quality of cervical mucus to prevent retarded entry of sperms. They inhibit ovulation and implantation.
Okay, so pills will inhibit ovulation and implantation. They will retard the entry of sperms. They will, oh, alter the quality of. Sorry. Sorry. Ouch. What's going on? My allergies. Okay, IUDs. Phagocytosis. No. Sure. Yeah, phagocytosis of sperms. Remember that. Don't try just phagocytosis. Phagocytosis of sperm in the uterus. Then medicated IUDs or hormonal IUDs. These will inhibit implantation, make the cervix hostile to the sperms. Then what else? Let me think, man. Let me think. Let me think. Let me think. Let me think. Then yeah, the last one, the copper one, suppresses motility and the fertilizing capacity of the sperms. Oral pills now. Oral pills. So they're taken during daily for a period of 21 days, starting preferably at the first 5 days. For 21 days. After a gap of 10 days. There are not side effects given here. I think we have to go through the textbooks and effects. I don't remember there were like side effects given. I don't know where it is. Maybe it was in neat level. Oh yeah, what about the emergency? Oh wait, this does not even mention the emergency contraceptives. Oh, did not notice that. Okay, I think this one is done. Let's move on to the last part, and this chapter is also completed. Okay, so there are a lot of highlightings, and there's this. Okay, yes, this ban is necessary to prevent female feticides, bro. And then because first of all, it is a fetal sex determination test where the amniotic fluid is taken, where the dissolved fluids and the fetal cells are checked, and where we find that the embryo, around the embryo, developing embryo, where the amniotic fluid checks the chromosomal pattern. You know that thing, like suggests some advantages. Very few oral, very few side effects with very high contraception. Then what is the principle on which the battery? Literally, I think it's literally blocking the insemination. Literally, the barriers are literally blocking it. Let's just check if just did I miss anything? Yeah, prevents the from physically meeting with the help of the barriers. Yeah. Gift, gamete fallopian transfer, and this is zygote intrafallopian transfer. Okay, name some hormones releasing IUDs. Okay, this one is important. Wait, I Okay, 1 minute. IUDs. Hold on. Okay, IUT and IUR. Okay, then that's fine. That's IUDs only. Let's just check this in Google. Hormones like what? Like what? Progesterone, estrogens? No, that's the ovarian pill. What? Name some hormones. I want to know actually if I missed anything. That release IUDs. Let's go. Progestin. I have no idea, guys. What in the world? I have no idea what this progestin means. Levonorgestrel. 1 minute. Progesterone. Okay, let me just mention that. I've not heard of it. This is not there in the textbook, man. Okay, wait. Let me just check if there are. No, it's not there. Okay, it's not there actually. Why am I writing it here? Progestin and what is that name? Levonorgestrel. Levonorgestrel. I'm gonna write those two. No, wait. Okay, wait. Okay, that's the IUD name. Okay. Okay, wait. So levonorgestrel is another type of. I'm just gonna write progestin. This progestin. Yep. Implants. No plant. Okay. We didn't study emergency contraceptives. I don't know why it's not there. It's important, guys. We have completed biology nicely, but I have a doubt in genetics, so we'll also complete that nicely. Guys, I know that I'm sure that I can actually like crack the code and all that stuff, but the thing is, we have to be really careful. Thank God I revised biology nicely previously. Like I can do it fast. Okay. So this one will take some time. This one will take some time. And bro, what? Okay, the notes does not contain emergency contraceptives, not infertility, and not sterility part. Like, infertility means by the means like there is a definition. Why do we people at what moment or why do we advise that people are infertile in some conditions? We need to like know those conditions, right? So no, it's not there. I'm pretty sure that they can have a chance of asking those questions. So I'm going through that.
growth pathways. Okay. Sterility. Okay, so first of all, we'll go through this, then we'll go through those topics which we have not gone through, like uh, sterilization. Sorry, not sterility, not sterilization. That is surgical method or terminal method. Infertility and emergency contraceptives. Okay, then we're gonna study art, not the art that you're thinking of. It's assisted reproductive technologies. The couples could be assisted to have children through special techniques commonly known as. Now, this thing is important. We do know that with some techniques to actually like prevent conceptions, that is, birth, that is through contraceptives. Now let's talk about how can we make conception possible, even though the person is not like let's say that we need to know that there are some chances that person desires to have a baby, right? So but the problem that he faces is that he might have some problems with the contraception. I mean, let's not say even contraception, let's say sterility or infertility. So these are some techniques that helps out a person in such way. Okay, so first of all we have the in our okay art we have the in vitro fertilization and in vivo fertilization. Where is in vivo fertilization? It's not given. That's weird. Okay. So in vitro fertilization is the fertilization that is taken outside the body in almost similar condition as that in the body by embryo transfer in one such method, that is test tube baby program. Like I think I remember this. Like the one thing about this is that this test tube baby is, I don't know why this is just too overrated. Like I still remember studying in vitro fertilization, and that was back in 8th standard. Like when we started studying reproduction for the first time, like human reproduction and all. So that was the first time we studied about this. And we just studied about this. We didn't study about ZIFT, IUD, GIFT, AI, and then ICSI, and in vivo also, in vivo fertilization. Even in vivo fertilization contains GIFT and AI if you don't know. Uh, Here, the zygote, or yeah, okay. This one is a bit type of topic which I didn't study much about. So let's study nicely. So, an ova from the wife or donor and sperms from the husband donor are collected that are induced to form zygote in the laboratory itself. The zygote are early embryos with up to 8 blastomeres could then be transformed into the fallopian tube. This is known as ZIFT, zygote intrafallopian transfer. So this one thing is there. However, if the embryo has more than 8 blastomeres, then it is considered to transfer it to uterus, and that's why it is known as intrauterine transfer to complete its further development. And yeah, that's just about the in vitro. However, there is another one that is talking about in vitro, that is ICSI, a sperm that is directly injected into the ovum. Infertility. Okay, hold on. Robot plus check. Let's use this. Let's say that I want to study about the topic in vitro fertilization and in vivo fertilization. I have studied 4 topics: zygote intrafallopian transfer, ZIFT. Then I studied about the topic ICSI, intracytoplasmic sperm injection. And then I also studied about the topic GIFT, gamete intrafallopian transfer. And the last topic as artificial insemination, AI. All these topics, could you please sort them into the in vivo fertilization and in vitro fertilization?
Absolutely. So let's break it down. In vitro fertilization, IVF, is when fertilization happens outside the body in a lab dish. Meanwhile, in vivo fertilization happens inside the body. So for the IVF in vitro group, you'd have ZIFT, zygote intrafallopian transfer, ICSI, intracytoplasmic sperm injection, GIFT, gamete intrafallopian transfer. Technically, it's inside the body, but gametes are placed in the fallopian tube before fertilization, so it's a hybrid. For in vivo, you'd have artificial insemination, AI. In short, AI is purely in vivo, while ZIFT, ICSI, and GIFT all involve lab based steps, so they fall under in vitro.
Thank you, ChatGPT. That's useful. Actually, what I've studied so far from teachers is that gift should be there in vivo, not an in vitro. So ZIFT. Okay, that's interesting. Actually, if I'm being honest, that could be a tough question if anyone asks about it. But I would say gift. ZIFT, zygote intrafallopian transfer, because gamete intrafallopian transfer is inside the body, but gametes are placed in the fallopian tube before fertilization. Fertilization, so it's hybrid. Hmm. Mostly it should be the female one. Of another female who cannot produce one but can provide suitable environment for fertilization and further development. So it's like a female donor which can actually like capable of ovulation. So they can do the gamete intrafallopian transfer of another female who cannot produce, but one can provide suitable environment for fertilization. That is like implantation, and it can be suitable for gestation and even parturition, and that's further development. Yeah. Uh, that is used for such. Okay, it's given here in vivo fertilization. Okay, my bad. Within the female, fusion of gametes take place in within the females. That's this one. Also could be used for such transfers. Now, in vitro is actually outside, but in vivo is inside. Remember that. I don't know why ChatGPT said it's hybrid, because the fact that the main type of fusion is taking place inside, not outside. So that's one thing. And this one, yeah, this one is done outside, I believe. That's what my teachers told me about this. So yeah, this one is in vitro. Case is either due to inability. Oh wait, yeah, that's yeah. Name some hormone. It just says hormone, so it's progesterone. It's an outside topic, right? Like it's not there in the textbook. That's one thing. Adoption, emotional, religious, social factors. There are other topics. So like adoption is one such topic. If this is not working out, adoption is also a topic. Like you can literally adopt an orphan, or a kid that is going in a very bad time. So that they can actually like take them as their child and adopt them. That is also possible. Adoption. Emotional, religious, and social factors. I don't know what this thing means actually. Maybe is it like influential, like something, but not related to. I'm not sure about this. This is general, by the way. This is general. This is not scientific. This one is scientific, like full. So yeah, damn, we are giving a lot of time to this thing. I'm sure that I can complete this full thing. Just the only thing that scares me is a bit of, I don't know whether I can even complete this. It's the last one, genetics. Thalassemia and sickle cell anemia. Those are the weakest ones. Trust me. Like I have weakness there. If I can strengthen that topic, then that's really fine, because I feel like they will ask us to like explain this topic. So yeah, I- if I'm being honest, if anyone gives an or question to explain sickle cell anemia or thalassemia, I'll go for sickle cell anemia, because it's much easier than thalassemia. Thalassemia looks even more complex. In fact, I feel like I can explain sickle cell anemia, but thalassemia is weak, very weak. I don't have much idea about it. Also, MCQ questions arise from there, so that's one thing. I feel like one sure question will come from there. Also practice crosses alternate these nodes at them, and then you'll also do some idea other topics like emergency contraception, then sterility and infertility. Okay. Okay, so intracytoplasmic sperm injection. Sperm is directly injected into the ova in fertility cases due to inability of the male. Now, I don't know why some people say that they actually take the haploid nucleus of the sperm and then they just take that and then just inject it to ova. I don't know whether that's true or not. No, that's like some people say that. That could be true, but then but mainly it just says that a sperm is directly injected into the ova, like literal sperm, not like haploid nucleus of the sperm. Infertility cases either okay, this could be the one thing: inability of the male partner to inseminate, very low sperm count, erectile dysfunction, and what else was there? Um, there were more actually. I think it's there in the textbook. We can study those infertility cases. Like I have some very like not only main textbook points, but I also have like very outside of the textbook points too. Okay, low sperm count, no gamete production in others one. Okay, failure of fimbriac collection, erectile dysfunction, sperm motility problem, endometrium not thickening, no thickening of endometrium. So no thickening of endometrium, erectile. So let's say for females, no endometrium thickening. There is problem with the this one. I don't know how I did that like that. Oh my god, I'm kind of tired, but let's not give up, guys. This is what we need to do. We need to study. Also, I believe that my screen is. What? Wait, I did not show my screen. What? Oh, my bad. Oh, I'm so sorry. I did not see that coming. Here we go. Also, there is one viewer watching. Hi there, bro. How are you doing? I did not notice that. Transference ones of methods. IoT. Okay. IoT and the IUI. Is that the only thing? ET is the embryo transfer. This one should be taking inside. No, outside, outside. No, inside is in vivo. My bad. So in vivo fertilization gift. So basically, transfer of an ovum collected from a donor to into the fallopian tube of another female who cannot produce, but one can provide a suitable environment for fertilization and further development. If that's the case, then gift is the option here. For intracytoplasmic sperm injection, which is an in vitro fertilization, a sperm is directly injected into the ova, and then through that we can actually like literally the fertilization will take place there itself inside the fallopian tube, uh, because it's in vitro. Sorry, it will the fertilization take will place outside, then it will be transferred again inside. That's what I know for sure. I don't know what's the difference between ZIFT, like ZIFT and intracytoplasmic sperm injection. It's just literally same, right? And then gift, yeah, this one is like a female of a donor which would a female donor which is available to like provide an ova for another female who's infertile to release ova but has a suitable conditions for for implantation, gestation period, and parturition, that is after the aftermath, or let's further development after the fertilization. Then we have Yeah, artificial insemination, not AI. AI is artificial intelligence, right, guys? So artificial insemination. Uh, the semen collected either from the husband or a healthy donor is artificially introduced by either into the vagina or into the uterus, IUI, intrauterine insemination of the female. Like, bro, the technology is crazy. I did not know that this existed. But then I again have no idea. But then I have no idea why is this useful. Like, Gift has a reason. Zift also has a reason. This one also has a reason. But what? Okay, wait. Maybe there is a problem with erectile dysfunction. Okay, so yeah, erectile dysfunction could be the reason for this one. That's one thing. Okay, insemination, and this is intrauterine transfer. Gamete intrafallopian transfer, intracytoplasmic sperm injection. Then that is zygote intrafallopian transfer. That is in vitro fertilization. That is embryo transfer, assisted reproductive technology. Yeah. Then test tube baby programs. That's one thing. Insane. Yeah. Then this one should be taking place inside the body. Where's the in vivo? In vivo fertilization. IUI intrauterine insemination. Emotional, religious, or social factors adoption. It's just in points. What? To this, we come to the end of this chapter. Let's go. We are progressing great, guys. Don't worry. Now we are at 12 o'clock. I know we are doing a bit slow, but I think we are doing great. All right. 1 minute. Let's go. So, emotional, religious, and social factors are also deterrents in the adoption of these methods. True. Okay, let's read this. Though options are many, all these techniques require extremely high precision handling by specialized professionals and expensive instrumentation. So this is expensive, and also really, very it should be done precisely by the highly expertise professionals, and then obviously their benefits is affordable. Therefore, those facilities are present available only in very few centers in the country. Obviously, their benefits is affordable to only a limited number of people. Emotional, religious, and social factor. Yeah, so I think this is very less used. What do you guys say? In vitro fertilization should be used very well. Yes.
118 subs. Yo, thank you, man. Whoa, when did that happen? I did not notice that. Oh, wow, thank you very much, guys. 1 minute. Alt. Yeah, that's much better. 118 subs. Wow, thank you very much, guys. Wow, thank you, thank you, thank you. Thank you so much. 118 subs is a lot for me, man. One sub. That is one sub crazy, bro. I don't know who subbed. Let me just check. I think I'll get to know a person who subbed to me. Uh, let me just check who subbed to me.
I don't know the notification. What about YouTube Studio? What does YouTube Studio has to say about this? Come on. No notifications? What? Um, Alright, so 118 subs. Wow, thank you very much for that, guys. Like, that's great, guys. Thank you very much. Oh man, thank you. Okay, emotional reasons, social factors also. Since religious, social, and emotional factors, since the ultimate aim of all these procedures is to have children, in India we have so many orphaned and destitute children who would probably not survive uh, till maturity unless taken care of. Our laws permit illegal adoption, and it is, yeah, as yet one of the best methods for couples looking for parenthood. Okay, one of the best methods for the couplehood for looking for parenthood. That's great. Okay, okay, but there is one topic about sterility I want to share. Where is it? But sterility. Okay, have a minute. A discussion on reproductive health is incomplete without a mention of infertility. A large number of couples all over the world, including India, are infertile. They are unable to produce children in spite of unprotected sexual cohabitation. The reasons for this could be many: physical, congenital diseases, drugs, immunological, or even psychological. In India, often the female is blamed for the couple being childless, but more often than not, the problem lies in the male partner. Specialized healthcare units and fertility clinics could help in diagnosis. Could help infertility clinics could help in diagnosis and corrective treatment of some of these disorders and enable these couples to have children. Wow! Thank you for the 118 subscribers, guys. Okay, wait. Why is it not given here? Okay. 4 hours 36 minutes. Wow, that's a good progress. We literally are right now having the landmark of 4 hours 36 minutes. What do you guys say? Like, okay, wait, 7 hours more to go, right? So that will be 11 hours 36 minutes. So 7 hours. So at night 7 o'clock. will be literally able to like hit 11 hours. That's crazy. I mean, that's fine because we can do that. There's no problem in that. Then the emergency contraceptives. Longer. Like I think it was 4 to 5 years. As far as I remember, I studied. It was 4 to 5 years. 4 to 5 years. Yep, that's what I know. Realization. Mental, physical, drugs, immunological, mental. What about mental? Is it not mental? No. Diseases. Psychological. Okay, it's not mental but psychological. Okay, so I think yeah, this is the one doubt I have. Inability to achieve. Oh yeah, this is the NCRD one. Inability to achieve clinical pregnancy. Such that the yes, I think that's it. Yeah. Oh my god.
Phew, that was long time. Okay, wait. Oh man, I didn't complete that emergency one. Contraceptives. Okay. Uh, contraceptives. This is basically oral pills, or the effect of the contraceptives to prevent the pregnancies from under the 72 hours of coitus, 72 hours of coitus, due to unprotected or unprotected or casual copulation or rape. That is a bad thing actually. So yeah, social levels like sexual abuse or rape, these are really bad. I'm. Why am I even saying that word? R word. Like that's really bad word. I feel like that's a bad word. I shouldn't be saying that. Yeah. Progestrogens or estrogen, progesterone, estrogen. Never mind. Progestrogens or progestrogen, estrogen. Okay. Or IUD and IUDs are effective under this are effective contraceptives that can be used under 72 hours of coitus to prevent uncasual, sorry, not uncasual, irregular, unprotected, casual, or social evils like social abuse or R word. Okay, I'm not gonna say that word again. My bad. I shouldn't be saying those words. Okay, finally. Oh, we did it. We did it. That took us 5 hours, by the way, to like complete this whole thing. Almost 5 hours. Just imagine what about the next part. Imagine what the next part will take much, man. The next part will be the final boss of this thing. So we have 9 lectures to go through for this 4.1. Let's go for this. Let's go. We're gonna speed run. We are not gonna waste time, okay? Unless and until if we have to study laws. That's one thing. Just check how many. Okay, so genes, the basic unit of inheritance. What was so new and innovative in this? He used statistical analytics and mathematical logistics in biology. And then he he used large sapling size. That one thing I knew 2 reasons. Then explain the law of dominance using a monohybrid cross. First of all, we'll write the law of dominance that the characters are controlled by discrete units called factors. Factors are affected. We'll understand and learn the explanation just in case, who knows what will happen. Then uh, differentiate between the following: dominance and recessive. Dominance is a type of a trait where it can show the effect of the character even in the presence of the recessive trait. Uh, uh, Homozygous and heterozygous, where homozygous are the pair of alleles which are there are pair of alleles which are identical. Heterozygous are a pair of dissimilar alleles. Monohybrid is a cross of in which one character or trait is considered at a time. A dihybrid cross is a cross where two characters or traits are considered at a time. Uh, we mentioned the advantages of selecting pea plant for experiment by mental, because it has a short span of lifetime. It can produce a large amount of pea plants. It is basically having many characters and control characters or traits. And then it is what else can we say about this? These questions are yeah, I was able to answer some of these because we have studied this in 10th also. Then explain the law of dominance using monohybrid. Okay, law of dominance factors occurs in discrete. Yeah,
We need to know the law of dominance, guys. The law of dominance. We're cooked basically. Like, what? This is gonna take so much time. This chapter is gonna take so much time. Let's lock in, guys. We'll try to complete this under 2 hours for this chapter because this chapter is very lengthy, big chapter. Took me so much time last time, if I remember. Like, especially the laws. We need to know the laws. The laws are also important. So what we're gonna do? Let's fast. Okay, we'll read. He conducted hybridization experiments. Okay, the date years 1856 to 1863, and proposed the law of inheritance in 1865 of in living organisms. Statistical analysis and mathematical logics were applied to problems in biology. His experiments had a large sample size, which gave great credibility to the data. Mendel investigated characters in the garden pea plant that existed as two opposing traits. This allowed him to set up a basic framework of rules governing inheritance. Okay, basically dominant and recessive. 1 minute. Let me just remember this. Stem height. Then that is stem height, flower color, position of the flower. Then what else? The color of the seed, shape of the seed, color of the pod. The last one. What's the last one? The last one. Yes, shape of the pod. So this one will be tall, dwarf. This one is violet, white. This one is axial, terminal. This one is inflated, constructed. Then this one is green, yellow. This one is yellow. No, this one is round, wrinkled, and this is yellow green. Okay. Then yeah, the confirmation of his experiments of successive generation of test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Are these 2 points necessary? Like, I have no idea because like mainly we studied about only these 2 points as far as I remember. Yeah, he proposed to call these things as factors, which are nowadays known as genes. No, genes together, therefore, are the units of inheritance. They contain the information that is required to express a particular unit. Okay, so that's one idea. Genes which code for a pair of contrasting traits are known as alleles. They are slightly different forms of the same gene. So that's one thing about this thing. Then we got the monohybrid cross. Homozygous, same identical pair of alleles which are identical to each other. Pair of dissimilar alleles is heterozygous. Capital T, small t are the genotype are called the genotypes of the plant, representing the genotypic forms. That are like writing in letters. Alleles are written in letter form. That is genotypic form. While the descriptive terms or the observable characters or traits are seen, the way of writing the characters in an observable way is known as phenotypic characters, like tall. Using this, the way of the application of descriptive terms by observation is like tall and dwarf are the phenotype. Then dominant recessive Punnett square, which was introduced by Reginald C. Punnett, which is a British geneticist. He used this as a graphical representation to calculate the probability of all possible genotypes of offspring in genetic cross. To be honest, Punnett square is very useful, very useful. So W. Reginald C. Punnett, very useful. Like, bro, I don't know how they did the genetics before Punnett square. Like, I'm so confused. Like, what did they actually use? That's what I'm saying. Like, this is the Punnett square if you don't know. Then emasculation, like removing of the stamen so that self pollination doesn't take place in the parents. That is known as emasculation, as far as I remember. Sperms. What did I say? Stamen. Yes, stamen, not sperm. Did I say sperm? I don't know. I don't know. I don't remember. I just don't know. Maybe it's my mind playing with me. Maybe not. Maybe no. I know I'm kind of tired, but let's not give up. Our goal is to complete this under 2 hours, guys. We can do this. We can lock in. Like this chapter is big, I know, but once this done, we'll do this in the 1 hour. Like we'll complete the biology textbook questions in the 1 hour. Then we will do diagram practice. Oh, sure, diagram practice is also important. Then what do you guys see? Like if I can start at 7 o'clock, then that's fine. But then the problem is, okay, let's challenge myself. Okay, I'll challenge myself at 6 o'clock. I'll complete full biology, full work. What do you guys say? And then after that, I will do full maths, like only mathematics, no biology at all. But then that is not easy at all. Then, this is not easy, bro. This is gonna be tough. We're not gonna give up. Okay. I don't know what's going on with me right now. Okay. So, the confirmation of his inferences. It's kind of crazy that we are just continuously studying for 4 hours. That's fine. I don't know why I'm getting tired. I'm not supposed to be like that. Let's log in. Bro, how am I gonna write this thing? Not. This one is done. Genes, basically, these are the ones that contain an information to express a particular trait. Genes are basic unit of inheritance that contains the information to express a particular trait, such that it gets passed down to another, to the other generation, to the progeny, through that is inheritance. Genes which code for a pair of contrasting traits are known as alleles. They're slightly different forms of genes. T and T are alleles of each other. Uh, Monohybrid cross, inheritance of one gene. Inheritance of one gene is known as monohybrid. Inheritance of one gene, or a hybrid cross in which one trait is considered at a time, is known as a monohybrid cross. And a dihybrid cross is basically inheritance of two genes, or or a cross where two genes are considered at a time. That's how we can actually mention this. And then homozygous, capital T, small capital T, pair of identical alleles, a pair of alleles which are identical to each other, and pair of dissimilar alleles are known as heterozygous. Capital T, small t are called genotypes of the plant. There is a way to write this. The genetical representation of alleles is known as genotype, and to write the observable characteristics in descriptive terms is known as phenotype, like tall. And to write the observable characteristics in descriptive terms is known so to write the observable characteristics in descriptive terms is known as phenotypic characters, dominant, recessive. Then Punnett square, like introduced by or developed by Reginald C. Punnett. Then characters are controlled by discrete units called factors. Factors are pairs and pairs, and it's similar. Yeah, the law of dominance is easy. I can like easily learn this. This one will take time. This one will take time, and then yeah, this one. Okay, we are actually fast with this, and wait. We'll look at what do we need to look up? We need to look up this thing. Where is it? Where was that? No, where is it, bro? Yeah, this one, the first one. Like why P plant? That's one thing. So I'll copy paste this and then we'll check this out because we need to know the exact reason. We need to know this because it's not given in this book, right? Or this PDF. The confirmation of his inferences from experiments on successive generations of his test plants. The confirmation of inferences were proved by the that these results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in Queen Garden Pea plant that existed as two opposing traits. This allowed him to set up a basic framework of rules governing inheritance. This one can be ignored because this is just like a binomial expression. where basically a plus b the whole square is equal to a square plus b square plus 2ab, and that's how you get the value. You know, basically what happens here is that this is the genotypes of each character of each trait given, and here this 1 by 4, 1 by 2, 1 by 4 are the probabilities of that genotype. Like that's what it says. Okay, law of dominance, law of segregation, then this one. The confirmation of inferences of the inheritance of the traits proved the pointed out proved that his results pointed to general rules of inheritance. Okay, you know what? I'm not that good in English, but I want to know what do you mean by unsubstantiated. Also, 1 minute. I think you guys can see it now. Yeah. What is unsubstantiated? Un unsubstantiated, not supported or proven by evidence. Okay. Hmm. I see. You're using some good English here. Characters are controlled by discrete units called factors. Factors occurs in pairs, and in a disciplinal pair of factors, one pair dominates the other. Rest is here. Okay. This law is based on the fact that alleles do not show any blending, and that both the characters are recovered as such in the after generation. The parents contain two alleles during gamete formation. The factors or alleles of two pairs segregate from each other, such that a gamete receives only one of the two factors. A homozygous parent produces all. We have 6 hours, guys. Only like 6:30 or 6 o'clock. That time we need to like fully complete it. Uh, wait, I just can't remember. It's the confirmation of his inferences of the experiments proved that his proved uh, of his inferences were proved or confirmed led to point out his general rules of inheritance. Mendel investigated characters in the garden plant that existed as two opposing traits. This allowed him to set up a basic framework of rules governing inheritance. So it have invest ex. His 6 years of work has really formed another branch in the world, which is pretty important. Like, Mendel deserves it. Like, sadly, bro died without, like, I cannot even call him as bro, but still, he died without even knowing that it's gonna be so influential to the world. Like, wow, feel bad for him though. Okay, so the Mendel investigated that the characters in the Garden P that existed as two opposing traits has existed the two as two opposing traits, which give which gave him a basic framework setup of rules governing inheritance. Man, this this is not going into my brain for some reason. Like this last one, this one has got me idea. Uh, okay, let me just check. Can I write it at somewhere? Yeah, okay, let's try this on the practice book. Do we need to wait? So let me just recall what I studied for this one. So basically, he applied mathematical logics and statistical analysis for the problems in biology, in biology for problems, and not only that, he he his experiment contained large sampling size, which gave him great credibility of data. Then the third point would say that the confirmation of his inferences from the experiment proved and pointed out his rules of general rules of inheritance. Then the last point that says is the you know the Mendel investigated that in the garden pea plant that there were two opposing traits in a pair, as in a pair, and which gave him which set up him a basic framework for the laws of inheritance. So laws of inheritance is something which is a big discovery. So each law that he proposed, there were three laws. Okay, so law of dominance, law of segregation, and law of independent assortment. But for now, we are talking about monohybrid cross, so law of dominance. Characters which are controlled by discrete units are called as are called as factors. Factors occurs in pairs, and in a pair of factors, the uh, one member dominates over the other. Uh, Dominates the other. In a dissimilar pair of factors. My bad. Dissimilar pair of factors. The one member dominates the other. That's the member of the pair dominates the other. This law is based on the fact that the gene factors do not show any blending, and that it is recorded as such in the after generation. Then what does it say? The parent contains two allele during gamete formation. The parent contains two alleles during gamete formation. The factor or alleles of a pair segregate from each other. Okay. Each other such that a gamete receives only one of the two factors. This will take time. This chapter will take time. Literally, I'm taking half an hour for this one PDF. Oh my god. Okay. Then one thing about this is that Mendel investigated characters in the garden pea plant that has existed as two opposing traits. That this allowed him to set up a basic framework for the laws of inheritance or general for the governing inheritance. Okay, characters which are control. Now these two laws will disturb me a lot. Okay. Oh, you know what? Let me just open another file just to check. 4.2 here itself. I'm gonna open these. Okay, so we're gonna talk about test cross, incomplete dominance, codominance, multiple allelism, and then questions. Okay, this time we're gonna do crosses. Nice. This time. Okay, then I'm gonna open 4.3. Okay, so we got pleiotropy, polygenic inheritance, law of independent assortment, what is given here? Chromosomal theory of inheritance, Th. Morgan work. Okay, only one page. What? Okay, this one is also one page work. Then, no, this is gonna take so much time, guys. 4.6. Okay, 6 determination. Only one page for that. That's crazy. 4.7. Uh, mutation. Okay, gene mutation. That's important. Okay, this one is a bit lengthy, so it will take time. Then 4.8. Okay, this is where this will take a lot of time. Sickle cell anemia and color blindness. And last but not the least, 4.9. The last, thalassemia, and the chromosomal. Okay, pedigree analysis is also given. Okay. Oh, look at that. These are those same questions which came in exam too. Hmm. You think? Are you thinking what I'm thinking? I mean, basically these are PYQ, so yeah, that's understandable. Oh shoot, this question is gonna be a really crazy one. We'll have to search that up. This one, sickle cell anemia and thalassemia. Okay, so this one is a bit okay, lengthy. You see what I mean? Like, look at this. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. Like, what? Oh my god, this is gonna be so much time taking. Like, I don't even know whether I can even complete this in 2 hours, bro. Like, that's so many of them. Like, 9 notes. Can you imagine that? Let me count the pages. Wait. 1, 2, 3. What about this? 2, 2. That is. How much is this? 1. Okay, so this is 3, 2, 2, 1. So 5, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay. 15. Like, I'm not sure, but that's too much, dude. At least this will give me enough practice for this chapter. Let's move on, guys. Okay. This law is based on the fact that alleles do not show. Okay, we'll speedrun this second one, third one. Let's see what else things we can speedrun. This second one, third one will take some time. Third one will take some time. But yeah, I think this will be also done faster than the first one. Then what is this? Yeah, this one also. Oh shoot, what is this? Okay, this will take some time. This will take time. This one shouldn't be taking time. We just need to read and understand and learn those points. So this will take a bit less time. This one. This shouldn't be taking us. This should take us very less time. This one. This should take us less time too, actually. This one. This will take some time. And this one should take a lot of time. No, less time. No, mid. But this one is the largest for me. Like, what? Why is this so large? Because there are two laws and one question to learn. Interesting. This law is based on the fact that alleles do not show any blending, and that they can be recovered as such in the afternoon. and that both the characters are recorded as such in the F2 generation. The parents contain two alleles during gamete formation. The factor alleles of a pair from each other. s- segre- the the parent contains two alleles during gamete formation. Uh, the factors or alleles of if pairs segregate from each other such that a gamete receives only one of the two factors. Homozygous parents produce all types of all gametes that are similar, while a heterozygous parent would produce two types of gametes, two kinds of gametes. Okay, this one is easy. This one will take some time. This one is easy. Characters are controlled by, because I've already learned it. Characters are controlled by discrete unit called factors. Factors occurs in pairs, and then that last one is that in a in a pair of dissimilar factors, the one member of the pair dominates the other. So one member of the pair dominant, the other recessive. Okay, then law of segregation. This law is based on the fact that alleles do not show blending and that both the characters are recovered as such in the F2 generation. The parent contains two alleles during gamete formation which would segregate from each other such that a gamete receives only one of the factors such that a gamete only receives one of the factors the one of the two factors. A homozygous parent produces all these gametes of similar kind, whereas the heterozygous parent would produce two kinds of gametes. And another thing about this last time about the Mendel's part, why is it what innovated it? Basically, the thing is about this case or situation is that uh, Mendel's he applied mathematical logic and statistical analytics in biology for problems for biology, uh, and he hell he no no he in his experiment required a large his experiment contained a large sampling size where which gave him a large credibility to his data, and then uh, there were the confirmation of his inferences from the experiments proved and pointed out his general laws of inheritance. And then Mendel, which was able to investigate the opposite factors existing in the garden pea plant, which allowed him to set up a basic framework for the governing of inheritance. Okay, that's one thing we learned about 4 points. Okay, we have to keep revising this because this one is something which is new. And then law of dominance. Now I think we know this. Factors which are controlled by a discrete unit. Characters which are controlled by discrete units called factors. What is that? Okay, uh, I don't know. Uh, let's log in, guys. So 2:30 at that time we'll be completing this chapter. I hope so. That time we can do this. We'll complete the diagrams and all and textbook. Then we'll do the NCRT punch objective. And at the last we'll do maths. I'm scared, bro. Kind of scared for maths. I'm not scared for biology, if I'm being honest. So I'm just scared for maths because maths work is also there. Like I have to submit assignments. I'm not scared of biology actually. Like tomorrow's biology exam, by the way, if you don't know. But I have to submit some maths assignments, so I have to do the maths work today. Man, tomorrow I feel like I'm not gonna do any maths work. I'll just do full physics. No, like I do not want to interrupt my physics. I will master physics nicely, and I will also be live streaming so that we can fully focus on it. Then we'll have fun. Then about this, after that we'll do physics first chapter, current electricity. Then the last two chapters of electrostatics. We'll complete that, and then finally, at last, we will do chemistry. Chemistry have a lot of advantage, since we have completed it. We just need to read textbook and all. Then maths, physics, sorry, English. We also have another advantage. We did study literature. We will be going through the textbook of English literature, and then we will study that nicely along with the notes so that we get the ideas of the questions that will be asked, which is great. I like it. Then we have writing. That is the only part which I haven't done yet. So for English, that's the only part, and that will take some time for that too. So that's one thing. Another thing that I've noticed is that maths. If I complete it today, then it will help me for that next exam, because it will give me a lot of advantage for the next maths exam that was gonna come. So yeah, one thing. I'm not taking breaks, guys. Trust me. I'm like just talking with you and just studying along with it, like balancing. So this law is based on the fact that alleles do not show any. Yeah, wait. Did I say this? In a dissimilar pair of factors, one member of the pair dominates the other. That is dominant and then recessive. Then in the law of segregation, it's yeah, this law is based on the fact that the alleles do not show any blending, and the characters are and that both the characters are recovered as such in the after generation. The parent contains two alleles during gamete formation. The alleles are would segregate from each other during and that the gamete would receive only the sorry gamete or factors of a pair would segregate from each other such that the sperm would receive of only one of the two factors. Okay, this statement is easy to say, but then the concept is a bit too deep, and this is the main statement, by the way. A heterozygous parent, no, sorry, a homozygous parent would produce would produce all gametes of the same kind, whereas the heterozygous parent, well, a heterozygous parent would would produce gametes of two kinds. Okay, would produce heterozygous one produces. Okay, it's while a heterozygous one produces two kinds of gametes. Okay, what does this say? What do you want to say, bro? Okay, so test cross an organism showing a dominant phenotype and whose genotype is to be determined is crossed with a recessive parent. An organism who has where the dominant phenotype, an organism showing a dominant phenotype but the genotype to be determined and is crossed with a recessive parent, and this is how what we get to know. If it is like WW, then this is fine. Can I zoom this? Can I zoom to this? Like anyway, if I can zoom. Uh, I cannot zoom it. I cannot zoom this thing. But how can I zoom? Nah, sorry. Print? No, I don't need to print. Never mind. Let's just move on. Let me just get comfortable and just study nicely like this, because I don't know what's going on. Okay, an organism showing dominant phenotype but the genotype to be determined uh, is crossed with the parent of with the recessive parent. Incomplete dominance. The inheritance of flower color in the dog flower, snapdragon, or antirrhinium, antirrhinium, or dog flower, or like we say snapdragon. It seem to be no. This is not an example of blending. So we have three crosses to practice. I'm gonna use my practice book on that. Oh my god, bro. Phenomenon in which more than two alleles govern the same character is known as multiple alleles. Let's say if this is crossed with this one, this will be the phenotypic ratio of 3 is to 1 and the genotypic ratio of 1 is to 2 is to 1. But this one, 1 is to 1 for both phenotypic and genotypic ratio because test draws. Mm. A minute. Okay, this one is done. We'll have to practice crosses, that's it. Let's do that, okay? Yeah, protrudes from the surface and there, this is going to look like this. The codominants where the This law is based on the fact that alleles do not show any segregation and that both the characters are recovered as such in the after generation. The parents contains two alleles during gamete formation, and that the two factors or and that the factors or alleles segregate of a pair segregates from each other such that such that a gamete would receive only one of the two factors. And then a homozygous parent produces all gametes of the same kind, while a heterozygous one would produce gametes of the two kinds. Would produce One produces two kinds of gametes. Yo, super VFX, bros cooking. Thank you very much, man. I'm gonna subscribe to you. Yes, thank you very much. Uh, Sup, man, sup. Sorry, you guys. By the way, still here. Don't worry. I've been practicing this, like going through each and every work. I've been busy, but don't worry. I'm so sorry that I'm not reading the chats because I am actually like going through the notes, and that's why I'm busy. Okay, so let's move on. Yeah. Okay, so right now this is the one. Let's go. Let's practice this one.
Biology work is done. Let's go. And I need to use a pencil. I don't know what's going on with the pen, so let's go.
I'm here, guys. Nothing is going on. I'm just crossing out. Okay, I'm like practicing crosses because those are important. Mm. I like practicing some types of mathematics like this in bio. I'm telling you, man. This chapter will take more time. This is the boss chapter. Like molecular basis of inheritance is even more bigger than this. I'm sure about it, but we don't have full chapter. That's why we are fine with it. So, and plus I even completed it already. This is the boss chapter. This will take so much time. Trust me. This will take so much time. Once the crosses are done, we can move on to the third part. This one. This one is done. Like we have learned the laws and all that stuff. Oh yeah, we need to check this one, right? Okay, wait. Once this done, we'll check this one, first one, so that we know all the reasons. Because we don't need to forget all of these, right? Ouch, ouch, ouch. Oh, okay. Phenotype. Eh. Right, we have completed this cross too. We're doing great. Okay. Minute. We need to practice as much as we can, guys, because these crosses are important. Now this is full monohybrid cross, so after once we reach dihybrid, we'll be doing dihybrid ones. Those will take some more time because the Punnett square was the one that takes more time. Then Punnett square, finally, and here. Punnett square will help us here. Okay. Oh, I didn't write gamete formation. I should do that. Hmm, the gametes. Gametes. What are the gametes? Anyone live watching me? 1 minute. Dang, 13 people were watching me. That's crazy, man. I keep missing out people watching me. Also, I'm so sorry, Super VFX. Like, I didn't mean to video comment. I'm screen sharing, right? So yeah. Like, is it even readable? Like, I don't want to waste my time, but I will just check once. To my channel, capital T. Thank you for one subscriber, guys. Thank you very much. I don't know who is it, but hey, bro, I'm happy for that. Like, okay, it is readable, but it is a bit, blurry. I can see that. I can see that. But it can be readable. Hmm, what do I need to do? That's my lesson, Majestic, I guess. Till then, I'll move on to this. And yes, there is one question I have to check. Here we go. The third part. Okay. Okay, so pleiotropism, polygenic inheritance. These are easy. We can do this fast. This one, dihybrid cross, will take some time. How many questions? We have two questions to face. We'll go through this, how it's going on, okay? And then we'll do these two questions. We have to do this. So this will take as much time as the second one, and then the fourth one. This shouldn't be taking much time. Come on, guys. Fifth one should be taking more time too. Uh, yeah, I know it is a bit time taking, but still. Sixth one should be fast. Should be done fast. Seventh, okay, normal. Eighth, fast. We can do this fast, right? Yes, we can do this fast. This one, yeah, this one will be the tough part, I guess, because thalassemia is one thing which takes so much time. Okay, wait, this point is better for differentiating, I think. Thalassemia is quantitative disorder, while sickle cell anemia is a qualitative one. Uh, yeah, both are autosomal recessive. That's what I know for sure. They're, this the differentiating. This question will be crazy. I need to learn this. Like, I think there's a chance that they will ask this, and I need to be prepared for that. Like, bro, there's so many questions from for each notes. Like, you can just check this out. Five, three, then this one like 8, right? Then this is like 10. And then this one, come on, go down. 10, 13, 16, 18. There's no question here. No, okay, 19 and 20. No question here. No question again. No, wait. No, there are supposed to be questions. What? Oh, yeah. 25. I have it out here. I have to check. 25. 25 questions, guys. 25 questions. Oh my god. 25 questions is a lot. Say. What is that? What the hell, man? What the hell, man? What the hell? Wait, actually, can you see WhatsApp? I was just wondering about that. WhatsApp. But can you guys literally see that? Easy, bro, if anyone saw that. Like, really. Window display capture helps a lot. Window display helps a lot, man. Like, display capture takes your entire screen. It like shows your taskbar, then it shows your your notifications. Sometimes it can leak a number. Like, bro, that is crazy. 1 minute. Yeah. I don't know why there were some patches of light or like incident on my face. Let's go. We completed this. Wait 1 minute. We'll open Chrome, and from Chrome we can check this out. Right. Yes. Okay. So technically, what I meant here is that let's just check this one thing out. What was it? Why did Mendel use pea plant for his experiment? This one is a famous question. Short life cycle, abundant seeds. Okay, yeah, abundant seeds, then easily observable contrasting traits. Okay. Okay, this one is the one I needed to know. Easily manipulation. What does this mean? Naturally self-pollinating, which helps Mendel cross-pollinate by transferring pollen to different plants while preventing accidental wind or insect pollination. Okay, I have never studied about this, but I know that they're like they have short lifespan, higher yield, distinct traits. Like you can easily distinguish them. Then what? Because the petals enclose their reproductive organs, the flowers naturally self-fertilize. This enables Mendel to breed true breeding lines, ensuring parental traits remain consistent before conducting his process. So he did control pollination, that is selfing, through the help of these because pea plants are bisexual. That's why, and which means that they consider that both the stamens and the ovary, style stigma and all that stuff, both are present. So yeah, you know what I mean. What is going on? 1 minute. Huh? Okay. Okay. So this is what I wanted. Like you can literally see what I'm doing, and if I can switch like this, you can check what I'm doing. But literally queen switch like that. I like it. Yeah. I mean, I wait. I'm confused. How did he use Punnett squares for his crosses? Because technically, the person who invented Punnett square was after the discovery of the after discovering the laws, right? What? Nah, that is something which is very weird to ask. That's confusing. Okay, I know the seven specific traits, so no need about that. Okay, easily manipulation. Their close structure makes them naturally self-pollinating, which help manually cross-pollinate by transferring pollen to different plants while preventing accidental vent or insect pollination. So them naturally. Okay, so their close structure makes them naturally self-pollinating, which manually cross-pollinate by transferring pollen to different plants. Okay. So their close structure helped them naturally pollinating, which helped Mendel manually to pollinate. Okay, that's interesting. That's interesting. I see. Like I get it. So if I switch this and I switch this off, now there is no more. I hope you can see what I'm doing. What was it? For helps in control pollination and self pollination is also okay. Here we are to the third part. Can you guys see? Yes, you guys can see. Like I really want to take a break. I don't know why, but I cannot. How many hours has been passed by? 6 hours. That's crazy. Single gene product may produce more than one effect. That is known as pleiotropism. Starch synthesis in pea seeds control is controlled by one gene. Okay, this one is a bit different case. Bro, yeah. So pleiotropy, and then what? Okay, it has two alleles B and B. Starch is synthesized effectively by capital B homozygotes, and therefore large charge grains are produced. Baby homozygotes, BB, small BB. Okay. What is homozygotes? Do you mean by alleles or something? And therefore large starch grains are produced. Yeah, phenylalanine to tyrosine with the help of phenylalanine hydroxylase. If this does not take place, so it will convert into phenylpyruvic acid, uh, and then other derivatives too, which will cause mental retardation, skin pigmentation, and other symptoms. This is phenylketonuria, example of pleiotropy, where one gene can produce more than one effect. One gene influencing more than one effect is known as pleiotropism. More genes used in expressing it's the same characters. More alleles used in expressing of same characters is known as multiple allelism, right? Okay, more than two multiple allelism. If in a phenomenon in which more than two alleles would govern the characters are known as what? Multiple allelism. Example is ABO blood grouping for that, and then codominance is also example for ABO blood grouping. I'm sorry, ABO blood grouping is also an example of codominance. And then we have pleiotropism, where the starch of the grain and that one and phenylketonuria both are the examples of pleiotropism. For polygenic inheritance, the example for this is the skin color and the hair color. Is hair color included? Let me check. Let me check. I'm not sure. Quantitative. Polyluminant skin color. Very dark AABBCC to very light AABBCC, with many intermediate shades in between. Yeah, I think they will abuse this thing, and then they'll create a lot of questions. Like I know for sure. Like I have seen some crazy questions coming out from this thing. Like I want to also check the NCRT punch of this chapter, because this chapter's crosses are crazy, bro. They're not easy. Like they are confusing. Trust me. When I'm saying they are confusing, they are. Then we have to write the karyotype. The karyotype. We'll deal with that later, even though it's easy. Chromosomal disorders are easy, but then we need to be really careful. We need to understand what all types of questions we will be doing. I will be helping out others today. This is gonna be like a full effort type of thingy. Thank God there is not much into depth into it. I like that it's only 3 lines. It's not like the law of segregation, bro. It took me so much time. These are generally controlled by 3 or more genes with phenotype reflecting the contribution of each allele. It is controlled by 3 or more genes, in which it is which is reflected on the phenotype, where the genotype would consist of more than two alleles. Genotype. I'm not meaning by governing the same character, because that's the case of multiple allelism, and this is the polygenic inheritance. Example is given. You need to understand this inborn error of metabolism is also inherited as the autosomal recessive trait. So this is an inborn error of metabolism. We should remember that. And then it is also an autosomal recessive trait. Okay, accumulation of these in brain results in material retardation, reduction in hair and skin pigmentation, hair reduction and skin production. Sorry, skin production. No, Human skin pigmentation, then mental retardation. Uh, okay, okay, we got this. Single gene may influence or produce more than one effect, multiple effect of a gene, or control cellular phenotypes depending on its position. Okay, yeah. Then polygenic inheritance, three or more genes, which with a phenotype reflecting the contribution of each allele. Yeah, that's one thing. Like that's interesting because that's why we see many types of colors in human skin, of human skin. Like that's interesting. Like that's gonna be fun to watch. Like that's gonna be fun to understand. Okay, guys, do you think I should be taking a break right now? Like I don't know, man. I don't know. What, there is nothing going into my mind. It's going crazy. But let's not give up. Let's not give up. Let's complete this first. This is easy. This is easy. Only two questions. Law of independent assortment, and these all are easy. Just go through this part. Uh, yeah. Yes. I want to know the B capital B small B one. Okay. More three or more alleles that are okay. I know this is not from this topic, but still, small revision. Three or more alleles that are quantitatively affecting the which are affecting the phenotype. Each time is known phenotype is known as the polygenic inheritance. Example: the human skin color. It can range from very dark AA, BB, CC to very light small a, small a, small a, small a, small c, small c. Let's go. Also, thank you for the views, guys. Like W, you're doing great. Today we'll hit 11 hours easily. I know that for sure. I just don't want the stream to like end just because of some very stupid factors. But yeah, 81%. Let's go. That's great. Hmm. The alleles show incomplete dominance. 1 minute. In that case, let me just, yeah. Clear trouble. Let's go. Should be. Let's go. Hmm. It because it kind of affects not only the size of the seed, like not only intermediate, it also affects the overall start synthesis. It's like very less like seed shape. Yeah, let's go. We did it. Okay, so this one is done, I believe. There's yeah, they have lesser efficiency. The capital, okay, I'm gonna call them homozygotes. Okay, that's for now for this one. So capital B has has a higher amount of start synthesis taking place, and round shaped seeds. But here this one maybe will have smaller shaped seeds which are wrinkled, or less, and then they have very less start synthesis, lesser efficiency of start synthesis. Uh, but homozygotes, right? That's why. And then talking about the heterozygous condition, that is the capital B and small b, they will give intermediate size, and this intermediate size that they exhibit would actually, no, this is like, okay, no, no, more than three. Sorry, affects the size of these the that p, no, p. What does it called? The seed, say grain seeds. Yeah, the starch grain size is a bit like intermediate, and the starch synthesis is also not that strong, like not that efficient. I mean, it's like lesser efficient than capital B. Capital B is homozygote, but this heterozygote capital B small b is more efficient than small b homozygote. Okay, let's just check this out. This is easy. We can like just go through this. Yes, okay. Okay, now this is important. This is where we need to log in. People can do this, but this is where we need to log in. The Pinhead score part. Oh, what is that? Clean my screen, I guess. It's a bit dusty. I just noticed something there. I don't know what is that. Do I have a tissue box? No. That's a bummer. No, wait, there is one. Let's complete this first. Okay. Yeah, this is all taller and round seeds. On selfing, we get these gametes, and then these gametes arranged like this. Okay. They should be giving us this. Then this is the phenotypic ratio. I still remember the genotypic ratio. Is it this? 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. I hope so. I think that's the genotypic ratio. Like it has a pattern. That's why I was able to like easily memorize it. You know, it has a pattern like 1 is to 2 is to 1. That one genotypic ratio. Then you just multiply it by 2. 2 is to 4 is to 2. Then again 1 is to 2 is to 1. So 1 is to 2 is to 1. 1 2 is to 4 is to 2 is to 1 is to 2 is to 1. I hope so. That's the correct genotypic ratio I'm saying. Let's check. Genotypic ratio. I'm asking so many questions today, aren't I? Dihybrid cross. Dihybrid cross. Yeah, see, 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. That's crazy. It has a pattern, that's why. Okay, I should remember that I have to like also mention the phenotypic ratios characters like this and for genotypic ratio. Yeah, I'll note that down. Hmm. Okay, now it's time for learning this a law. Then we'll do questions and then we'll move on to the fourth one. Let me just get this. Please, please, 1 minute, guys. Chill, 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 chill. It's their dust. Mm, I barely can even see anything. Like I think there's no such dust here. But yeah, the law states that when two pairs of traits are combined in a hybrid, segregation. Okay, this one looks a bit complex too, but it is easy. Mm. Okay, okay. Segregation of one pair of characters is independent of. The law states that when the pairs of the characters are joined, or is it combined in a hybrid? Okay, just when two pairs of traits, when two pairs of traits are combined in a hybrid, then the segregation of one pair of the traits, or it's just one pair of characters, is independent of the other pair of characters. Now the law is easy actually, but you have to be really careful. So first of all, we'll do law of independent assortment, and yeah, we'll go quick revision for this one too. I just don't know why, bro. Like, why is it so like difficult for me to do this? Like, there's one thing I noticed. Uh, like every time, whenever I like learn something new, like this thing, like I just learned this. I just easily forget it. I think yeah, it's because of the less revision. But even though when I do the revision, it's just a problem thing. I don't know why. Like I sometimes apply those 1, 2, 4 techniques. Like revise 1 day, 2 days. Sometimes, not every time. Very less. I did that, but still I forget some things. I don't know why. That's not good. I have to get a really good retention way. The mouse is kind of helping, by the way. It's helping. Okay, so the law states that when two pairs of traits are combined in a hybrid, then the segregation. Sorry, segregation of one pair of characters is independent of the segregation of the other. Is independent of the other pair of characters. Do you think I should be writing down the law of dominance, the law of segregation, law of independent assortment? The next law that we are studying is pretty easy. It's not hard. It's not hard actually at all. It's this law, chromosomal theory of inheritance. You just need to study this law. That's it. This law, and then sorry, not this one. This one, pairing and segregation. This one is the chromosomal theory of inheritance. Then this one, yeah, I know I have an idea about this one, and then I have an idea about this one, but this is the law that we have to memorize nicely. And law of independent assortment is the one I will be memorizing nicely. This one, law of segregation of law of dominance. Here, I think there is not such things to there's no such thing to memorize nicely. Uh, I don't need to memorize this. I just can write this in my own words. It's just law of dominance and law of segregation that I have to memorize. Then law of independent assortment, and yeah, again last time this thing. So four things to memorize nicely. Rest all I can just say it on my own words. That's the thing. Okay, let's put that. Okay, so law of independent assortment. Just let me write that down. I know it's taking me a lot of time, but trust me, bro, this will be worth it. This will be worth it. We have already passed 1 hour, and we just completed almost three. Bro, come on, bro. Okay, let's do it. No worries because some parts are very small, like only one page, so we can complete them as fast as possible. Like this one. This one? Yeah. You know what I mean. Let's deal with these two later. Segregation of one pair of law of independent assortment. Let's do this across now. We'll have fun. Perform a cross between a pea plant that has seeds with yellow color and round shape, and one that has seeds that had seeds of green color and wrinkled shape. Okay, this is gonna be easy. This one will take. Okay, let's do this fast. Why is there a clue though? I'm confused. Is this because we're doing this as the first time? I mean, even though we had done this like previously, like in 10th, so I don't think it's required. But anyways, let's just move on. Let's just do it. Parents. Let's say that the male parent is yellow color. Okay, how did they start? Did they start with phenotype? No, they just parents phenotype. Let me just write it as phenotype here. In this case, we can change it, and that's not a problem. I mean, yellow and round. Let's say this is a male crossing with. Ouch. Sorry, my bro. I don't know. It's just fun doing this. Okay, green and wrinkled. Then genotype. Uh, wait. Capital Y, capital R, capital R. Crossing with small y, small r, small r. Gamete formation. YR crossed with small y small r. Okay. Now here F1 progeny, that is YY R. 1 minute. Someone's coming. Someone just came in. Someone also sent me a notification. What? जल्दी करना ओके ओके
Finally, I'm back. Let's go. I know I'm sorry. You may be thinking where I had went to. I was basically, like praying, and also, my sister came in, so she had to change clothes. So I had to go out. My bad. This is because this is also a dressing room. If you don't know, 16 weaves. Oh, two weavers already saw me without being here. My bad, guys. I'm sorry. Anyways, let's move on. Where was I? Here we go. Okay. Selfing. Write the phenotype. Let me just write the phenotype first. Genotype and phenotype. I'm practicing the dihybrid cross now. I think this is enough, right? We have done a lot of crosses. I will be doing another one. Don't worry. But I just want to tell about this. Okay, wait. Selfing and then what else? Gamete formation, of course. I forgot about that. Thank God. I got to know. So that is YR. 1 minute. Yeah. Gamete formation. Now F2 because we are about to cook with a Punnett square. Because we're gonna we're gonna do the cooking with the Punnett square or Punnett square. I don't know how you exactly pronounce this thing. Some people call it as Punnett, some people call it as Punnett. Like what? Okay. Yeah, that's enough. Then Y R Y smaller units of Y R. Similarly, YR. Hmm. There are so many genotypes to cover, so I'm taking that out.
No. Oh, this is smaller, smaller. Right, I think I wrote all the genotypes. Phenotypic ratio. Mm-hmm. Genotypic ratio. Not needed, but let's still write it out. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Okay. They even gave the clue as this Dan. Okay, another one. This is a bit time taking, but oh, it's fine. This is worth it. Hmm. Okay. Phenotype and genotype of F1 and F2 generation. Give the phenotypic ratio of F2 generation. List the Mendel's generalizations which can be derived from the above clause. You mean law of independent assortment? That the law states that when two pairs of traits are combined in a hybrid, the segregation of one pair of traits is independent of the other pair of traits or characters. Okay, characters. Okay, that's one thing. We'll also check. You know what? We'll copy and do this question. Copy paste this. Control C. Use this. Uh, okay, you guys can see it. Never mind. That's great. Control V. I have already copy pasted it. Once it's done, we'll do that. Okay, so let's do it fast now. I'm not gonna write steps because I want to save time. So let's do that fast. Okay. G GF. Oh, that is gamete formation, not girlfriend. Okay, guys. Um, GF is gamete formation. AVO. I didn't, bro. How didn't I notice that? That's crazy. It's so. This is the F1 progeny, by the way. Whoa. Gamete formation. AVO. F1. I'm skipping some steps, guys. I want to save my time. self s. I'm gonna write it as this. Okay. Now the punit job, I'm gonna do that. But let's first, write the gametes so that we don't confuse. Oh my bad. All right. We are gonna complete biology. I know that for sure. But we'll just have to do some remaining work like biology textbook questions. Then small revision of human reproductive health, yes, and the textbook questions. Right now, that's done. Diagrams are the main ones. Then NCRE punch, that one is also important. So yeah, okay, this should give me the F2 now. Sorry, unit square. This is
Finally. Now, what do I need to solve in this basically? Let me just check. So, technically, we need to be solving hmm, phenotype and genotype. Okay. There are pretty much a lot of genotypes if you would ask me. Genotype of F1. But yes, phenotype of this F1 is said to be F1 is violet and axial. Genotype of F2. Genotype, because I wrote the genotype. This genotype of F1, the genotype of F2 now. There are many genotypes of F2. Let me just check. A V, A V small v, A small a V, then this one, A small a V small v. A minute 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So 1, 2, 3, 4, 5, 6, 7, 8, 9. I need 9 of them. Okay. Uh, A small v, A capital V small v. Okay, we got 6 now. A small v small v. That's the seventh. Okay, small v capital V. That's ninth, eighth one. The last one. Okay, it's capital A, small v. So these are the overall genotypes I found for F2. That's crazy, bro. Telling me to ask this. So yeah, let me just check the Google. What does it say? And yeah, look at that genotypes, bro. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah. Really? I mean, okay, I know it's law of dominance and law of segregation, but the main thing derived from this cross will be law of independent assortment, because generally we know that law of segregation and law of dominance are derived from monohybrid. I mean, yeah, it's possible that we can explain this and this. It doesn't mean that, it doesn't mean that a dihybrid cross cannot explain this and this. Okay, I see. Work out a 4 by 4 Punnett square for the F2 cross. No, never mind, I did it. Don't worry. Punnett. Okay. So we have finally done it. Okay, this third part is done. Let's move on to the fourth one. Okay, this is the third part. Let's now do it. So Mendel published his work of characters in 1865, but it sadly remained unrecognized till 1900. This is because communication was not easy in those days, as his work could not be widely publicized. He used mathematics, logistics, and statistics analytics, which wasn't convincing for many biologists and wasn't accepted by many of his contemporaries. And then Mendel's approach of that was then.
Davies, Correns, and von Scherman. Austria, Germany, and what is this guy named? France? I'm not sure. De Vries. France? No, Holland. Who's in Holland? Wait, one of them is in Austria and one of them. Never mind. Let's not discuss about that. That's not important. Okay, so this one is there, and then in 1900, three scientists, Davies, Carl Correns. Okay, I forgot his full name, but yeah, this is Carl Correns and this is von Schermack. Yeah, okay. Their names are incomplete, if I'm being honest. Uh, Independently recover Mendel's results on the inheritance of characters. Also, by this time, due to advancements in microscopy, scientists were able to carefully observe cell division. This led to the discovery of chromosomes, or known as colored bodies. Walter Sutton and Theodore Boveri noted that the behavior of chromosome was parallel to the behavior of genes. Chromosomes as well as genes occurs in pairs. But there's a small difference I've noticed in chromosomes and genes about the independently segregation. That one thing, like how they actually segregate. There's a difference there, but all are same, like almost same. Hmm. Then this one is easy. We can say that they could be grown on a simple synthetic medium, and then they can in a single medium they can produce large number of progeny flies, and then they have they have a great sexual dimorphism. That is, they can easily distinguish between male and female. Then their characteristics are easily, yeah. It has many types of hereditary variations. So these four are the one reason. And guess what? This is small. This is not that big at all. That's great, actually, if I'm being honest. So let's complete this first, then we'll go through the law. We'll memorize it quickly and nicely. Then we have this thing, which is also small. We'll have to understand and learn this, and then we'll complete this too. So we can complete 4, 5, 6 faster, in a very fast way. Then seventh one will take some time, but it will be easy. It will not be that hard. And then we'll learn this one too. We'll learn this one. We have to do the cross for this one too, right? I think I've done enough crossing, like two crosses from the textbook. Then I've done three crosses, then two crosses, so like seven crosses overall, which is pretty much fine, I guess. These many crosses are fine for me to like practice overall, and yeah, let's just check. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to that of the behavior of genes, but the only difference that was the independently segregating. Like that was the thing I've seen. The pairing and segregation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. This is known as chromosomal theory of inheritance. Chromosomal theory of inheritance. He worked with the tiny fruit flies which are known as Drosophila melanogaster. These there are four features about it. They can be slimly grown in in a synthetic medium. They're capable of producing large number of progeny flies from a single mating. They are capable of literally um, they have great sexual dimorphism. That means that female and female can be easily distinguishable. They have many hereditary variations. That's one for characteristics that we can talk about. Now, communications was not widely publicized during that time, so Mendel faced difficulty in publishing his work in 1865. Many biologists weren't confirmed with the. He wasn't able to prove about the factors of work. Yeah, factors that were discrete units. He could not provide any physical proof or evidence for the existence of factors. Now, guess what? I mean, the future scientists were able to do that. Then Mendel's approach of using mathematics to explain biological phenomena was totally new and unexpected to many of the biologists of his time. So this is one thing. So I can say 1, 3. It's kind of the part of number 4, and number 2. Okay, never mind. I was feeling weird, some weird words. Okay, what are the questions though? Uh, Walter Sutton and Theodore Boveri. stated, if it I need to state it, it is the behavior of the the pairing and segregation of a chromosome or chromosomes would lead to the segregation of a pair of factors they carried. Okay, I have to learn that law. Then why was Mendel's work not recognized for many years? What was experimental material used by Morgan? What advantage did this have? Technically, this question, and this question. That's it. So yeah, we can do this, right? We can complete this one too very nicely, very easily. And then we have this. This will take time. Okay. Like what do I think about this? If I'm being honest, this part, linkage and recombination, is a very conceptual part. This one is fine. This is easy. It's not that hard. You just need to learn the law and then the ideas about this. That is fine. Then here, this is also easy. This is not that hard actually. You can just read through it and then complete it. That's it. And then you can actually understand this. For this, then this one can be also read and then studied, but the you have to understand it nicely. That's what thing I need to know. So yeah, mutagen. Okay. Ouch. Then there is frame shift mutation and gen or point mutation. Let's go, bro. Let's go, bro. If I complete these three, I will be able to come at genetic disorders. That's the part I want to wait for. No, not this one. This is very big, I know. But this is worth it overall. We have completed the crosswork. Crosses are done. It's just this part and the theory part left only. Pedigree, pedigree, pedigree. This is just practice. We have to just practice it. I don't see any pedigree based questions in this, in any of the notes. नहीं गर्मी है
ना प्यान ना लाइक कैसा बच्चा ये मैं
फोन को बैठा था मैं बाइक से
ना प्यान है एकदम भाप मार रही
भाप मार रही
बैठा से
क्या
क्या
खा
खुश होकर पड़ा तो क्या करना सर
नहीं सच में मुझे गर्मी होता नहीं लग रहा सच में
खाना खा
क्या है इसका
इडली
बात
बिरयानी खाना
बिरयानी है
इडली होते बच्ची ना
ओके खैर
दो इडली खा तो बिरयानी खा
इडलियाँ खा लेता हूँ इडलियाँ खा
ओए
लेता
मैं दो लग खा क्या
हूँ कौन से दो खाना से दो खाना से चॉकलेट और शुगर
कौन सा होना
नहीं दो खाना से नहीं
तुझे होना है तो दो खा
मैं दो खाया था
ये खा
मुझे दो अच्छा लगता है
खा जा
Finally, I'm back again. I was getting my food. They called me too, so I had to go. Man, I just get a lot of students like these. I don't hate it. Okay, there was one person watching me, 17 views. Like, I don't know why is it like this. Like, sometimes I don't know what happened today. I'm getting views mainly whenever I'm going out of the camera. And sorry, not yay. And I just noticed one thing that I look more clear with the light. So maybe using lights will be much better. Ouch. Okay, so I need to eat and study at the same time. Cannot even like take breaks right now, like most of the cases. Get out, please. Yes, here we go. Ouch. Here. Yeah. The mental work wasn't publicized. Not a deceased, is it? Hmm. Higher level questions. Okay, I see. Then, alright then. Okay, got it. already. Okay. I'm not gonna help this guy. So
prime to just helping out friends, guys. Okay, anyways. Oh, no way, bro. Man,
right here am I. Here I am. 82%. Not bad. 17 weeks. Okay, that's the same thing. My bad. I thought there was an increase in the viewership. Okay, never mind. Though Mendel's work suggests that factors, genes exist and they were discrete units, he could not give a physical evidence or proof of whether they exist or what they are made of. That's one thing. And then he could not. Communication at that time was difficult, so he was not able to publicize or widely um, or reach out to others when he published it in 1965. It was when he also applied mathematics and statistics in his in biology, which was totally unacceptable and new during the time of the for the biologists during that time. Then he was- communications was not easy in those days. Yes, of course. His yeah. Oh, this one was not accepted by many as an explanation for the continuous variation. His concept of dream was not accepted at that time as explanation of continuous variations seen in nature. Okay. Okay, I think that's it, right? The pairing and segregation of a pair of chromosomes would lead to the behavior of the segregation of the pair of genes. Pair of factors they carried, my bad. Pair of factors they carried. Chromosomal theory of inheritance. That's one thing. Chromosomal theory of inheritance. Okay, chromosomal theory of inheritance. Yes, okay. Okay. Factors that was disagreed. The pairing and segregation of a pair of chromosomes would lead to the behavior, would lead to the segregation of the pair of genes or the factor to the pair of factors they carried. Okay, so this one is the chromosomal theory of inheritance, and the pairing and segregation of a pair of chromosomes would lead to the segregation of the pair of factors they carried. Yeah, and if anyone who wants to know why did Mendel's work got rejected or got delayed for a long time, well, after he published it in 1865, it was difficult for him to publicize it in the public, as communications were difficult in that time. He was not able to give any physical proof or evidence for the that genes or factors were the discrete units, and whether if they exist or not, and what they are made, if they about for them and these giving no proof of that the factors would exist, that factors exist, and what they are, and didn't give the idea of what they were made of. That's one thing. Then he was not able to. He applied mathematics and statistics in biology, which was totally new and unacceptable by many biologists at that time. Then he was also rejected with the concept of factors or genes, which is the responsible. Which according to him was responsible for the continuity of variation, but was disagreed. It's in the nature. Okay. Disagreed on basis of the topic. What was the experiment material used by Morgan? T.H. Morgan or Thomas Hunt Morgan used Drosophila melanogaster. The advantages that he applied were that it grows on synthetic medium easily, and then a large progeny flies can be formed from a single mating, and the sexual dimorphism is really there is sexual dimorphism. They show the sexual dimorphism, and then these flies have many hereditary variations. The flies can also, you know, did I mention every point? Yeah, they complete their life cycles. Okay, they have a short span of lifetime, which is about of 2 weeks. Okay, now this is the topic where we need to lock in linkage and recombination, and this is the questions that we need to go through. Okay, so first of all, we just need to read this, and then we'll understand this nicely. Morgan hybridized yellow-bodied, white-eyed females and brown-bodied, red-eyed males, and so yellow-bodied, white-eyed females to red-eyed, brown-bodied males, and observed or intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated from 9:3:3:1. Morgan and his group knew that the genes were located on the X chromosome. When the two genes in a dihybrid cross were situated on the same chromosome, the proportions of parental gene combinations were much higher than the non-parental type. So Morgan attributed this due to the physical association or linkage of two genes and coined the term linkage to describe this physical association of genes on the same chromosome, and the term recombination to describe the generation of non-parental combinations or non-parental gene combinations. Even when genes were grouped on the same chromosome, some genes were tightly linked, showed very low recombination, while others were loosely linked, showed higher recombination. Example, he found that white and yellow linked showed only 1.3%, where
whereas the white and miniature wing were loosely had small linkage, which means that they had a bit higher number of percent of recombination, that is 37.2. This one is 1.3. This is 9. That is 98.7. Linkage, and 1.3% recombination. This is 62.8, right? Yeah, 62.8% recombination. Alfred Sturtevant, who was a student of T.H. Morgan, used the frequency of the recombination of genes between genes to find out and map their positions. He found, and a measure of the distance between genes and map their positions on the chromosome. Thus, the gene maps were used. The application of gene maps were used in the Human Genome Project, or the sequencing of the genome projects. The strength of linkage between Y and W is higher than W and M. So, what are Y and W? Yellow bodied and white eyed. This was miniature wings, and this is white bodied. Okay. So T.H. Morgan and his group knew that the genes were located on the same chromosome, and they also investigated or researched that these two chromosomes were tightly linked, which through which the parental combinations were really high, but the non-parental combinations percentage was really low. So he coined the term of this physical association between two genes as a link on the same chromosome as linkage, and he also coined the term recombination, which is the percent of the non-parental gene combinations to be formed. He found out that he also found out that 98.7 percentage linkage was seen in the genes, and 1.3% of 1.3% of recombination was seen, which means that there was very low chance of recombination. You need to know the. Never mind. What do you understand by gene mapping? Alfred Sturtevant, who was a student of T.H. Morgan, uh, used frequency of recombination between gene pairs on the same chromosome to as a measure of the distance and to map their positions. Thus, gene maps were used to find out the physical association of linkage, and to find out the linkage and the recombination formation seen in the gametes of the chromosomes. Then, why do certain genes tend to be inherited together in a cell at this time of cell division? Tend to be inherited together. I think is it because of linkage, high linkage, right? Yeah, I understand what it says. Uh, this is an A6 chromosome, right? So basically, I would say that let's say this is happening in a gamete. So this should actually, like, when the recombination takes place in the let's say diplotin, not diplotin, is it diplotin? After pachytene, yeah, diplotin. There, what happens is that these genes would not actually, move on. The genetic material is not exchanged, so there the you can say that the certain genes are tend to be inherited together because of the high percentage of linkage, and the combination is very low. So that's one thing I would say about it at the time of cell division. This is, I think, for meiosis, right? Okay, we'll copy this question and check this out. But this one, you have to like read this again just in case to like strengthen this out. Okay. So again, Morgan, so he like hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio we deviated from the 9 is to 3 is to 3 is to 1 ratio. Morgan and his group knew that the genes were located on the X chromosome. I mean, bro, like think about it. That's crazy level knowledge here. Trust me, man. This concept is really complex, and they somehow like try to prove it. That is crazy, bro. Like, I just don't know how scientists do these kinds of inventions or like discoveries. That's crazy. Like, literally involves a lot of your mindset. Like, maybe they say that yeah, you should stay with the question or the doubts with longer so that you can actually get a lot of ideas with. But that is crazy. Next is six determination, right? Yeah, this is easy. This is easy. We should not stress out from this one. We can solve this. We can do this. This should not be tough. This should not be tough at all. This one though, this one though will be okay. It is a good concept, but I think we can just read this out and complete it, and then yeah, Mendelian chromosomal disorders, and this one. Oh yeah, okay, we'll do this too. What are the questions asked? Three questions. Hanking's role, female heterogameity, and haplodiploid type of sex determination, where it is seen in honeybees. Haplodiploid type of sex determination. Here it is XY type for dogs. Oh, okay, it's XY though. Male heterogameity. Grasshopper shows an exotype of yeah. I know that. What? Okay. Okay, we are halfway through, guys. We are halfway through. We are doing it great. We are halfway through the chapter. Ow. Ow, 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 ow. Okay. I just want to check this out, though. Man. Ow.
No, like I'm thinking about anything something else. Like the linkage and recombination is one such factor that is, like deviating and disobeying the law of independent assortment. But what do you guys think about this? Like are there any other factors that disobey this law? Like I was thinking about that too. Hmm. All right, got it. So again, I think we can explain this that through this linkage and recombination thingy. Again, because that parental gene combinations were much higher, the same chromosome, and they were tightly linked. That's why they were deviated by the 9:3:3:1 ratio, because non-parental gene recombination was low, and very low in fact. And then they were not able to separate out independently. So this basically disobeyed the law of independent assortment, if you don't know. And another thing about this is that it says, oh yeah, we did this one, we did this kind of. We'll copy this question and see what's the answer. Control C, Control V. Let's go. Okay, that's the same thing I was thinking about. Due to genetic linkage, right? This happens because when genes are located physically close to each other on the same chromosome, because chromosomes to move as single units during cell division, these tightly genes tend to stick together rather than starting independently. All right then. Okay, well, I don't know whether. Oh man, that's crazy, bro. We have done this much. Half of the chapter is literally done. Literal half. Like, oh my god. Mm. Okay, this is genetic linkage. Then this one, we have an idea based on this. Guys, should we start moving now? Yeah, let's move. I'm confident now. Let's move to the sex determination. And this one, yeah, we'll just read this through and get the ideas. Okay, wait, this one should be. Okay, wait, this one should be known very well so that we get some idea. Hanking could trace a specific nuclear structure all through spermatogenesis in a few insects and observed that 50% of the sperms did not have any structure. 50% of the sperm received the structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanke gave a name to the structure as X body. Guess what? That X body was X chromosome, and the type of sex determination that he observed or we observed was exotype. That exo thingy. Yeah, something like that. But it's not exo basically. It's you may be thinking O could be a type of a chromosome. No, O indicates the absence of the chromosome. So if a male is born, okay, here and he is talking about the spermatogenesis, which means that it is happening in males. 50% did not receive that X body or X chromosome, and the rest 50% body received it, which means that 50% of the sperm did not receive any kind of chromosome. That means the males could actually have only one X chromosome in them. So that one X chromosome that these males will carry means that they will have only one allosome, whereas the females will have two allosomes because the ova would have two X chromosomes. That is possible. That's not type of any problem that we'll see, but this is seen in the males. So if I talk about exo type, we can talk about grasshoppers. Grasshoppers is an example of exotype of sex determination, in which the males have only one X chromosome besides the autosomes, whereas females have have a pair of X chromosomes and the same number of autosomes as that of males. And then we have human beings and Drosophila melanogaster. Now that Homo sapiens and Drosophila melanogaster, both of them
have this XY type of sex determination. And we all know this. I mean, not all. I mean, people may not be studying biology right now, but in 10th standard, we do remember studying about this. Like, that female has XX chromosome, whereas a male has an XY chromosome. In fact, I think that even, wait a minute. In 8th standard, I studied this, like, about this topic, like, XX female and XY male. So yeah, that's one thing. The males have one X and one Y chromosome, whereas female have a pair of X chromosomes besides also autosomes. So that's one thing. And yeah, the different type of chromosomes that we see is in males, so male heterogamity. And there is one interesting case actually, if you noticed, Drosophila's sex determination is a bit different. We know that human beings' sex determination is XY type. Here it is exotype for this grasshopper, and here this is ZW, or that is not ZW, female heterogamity, you can say. And here it's literally haplodiploid type of sex determination. So if I type in, let's say we'll check it out for each and every type. Okay, so let's say I'm gonna get this out of here. Let's just check this out. Okay, so I'm gonna like do this. Okay, so what type of, let's say, how is determined in humans? I want to know that. Let me just check. So it says a pair of chromosomes X and Y, the XY type. That's one thing. All right. What does it say also? There is another thing. At the moment of fertilization by specific pair of chromosomes X and Y, that the sperm would either contain that X chromosome or that Y chromosome, and that X sperm containing either X or Y would bind with the ova to form that gender, or the, what I mean. Then let's say grasshopper. Yeah. See, genetic mechanism known as exotype. It's XXXO means this is females and this is XO. That's one thing. Like humans who rely on X and Y chromosomes, they depend solely on the number of X chromosomes. So only X chromosome, with the zero indicating the complete absence of the second X chromosome. So that's one thing. Then let's go for birds. Like, yeah, let's go for birds. Okay, genetically by chromosomes. Does it say anything else? Okay, not much, but we need to know that it should be female heterogamity for this. Outs. Yeah. Females is ZW and males is ZZ. Let's say now grasshopper. Sorry. Now the fruit flies or Drosophila. This one will be interesting. This one has a different way of sex determination. By the ratio of X chromosomes to sets of autism. Can you see that? Like it says X is to A ratio. That is a bit unique, actually, if I'm being honest. Like, I had no idea that it would be like this too, X is to A is 1. The fly has 2 X chromosomes and 2 sets of autosomes. But this could be this too, like fly has 1 X chromosome and 2 sets of autosome. That will be 1 X is to 2A. So this is male and this is female. Wow. That is also one such way you can do it. Wow. It doesn't mainly depend on XY though. What? I still don't get it why. Let me just check. Drosophila follow XY type of, you know what I mean. Let's check. See what Oh, okay. So this is wrong, right? Wait, this is wrong then. Yo, this is wrong. Drosophila is not determined by the XY type. Okay, looks like we found a mistake, guys. Dang, look at that, bro. Look at that. We found a mistake here. Whoa, whoa, whoa. XY type. Didn't see that coming up, by the way. Oh my god. That is so randomized, bro. Did not expect that coming up. Okay, so it's not Drosophila. We should not have this thing here. The males, okay, we'll just focus on human beings then. Both cases of males. I don't know why did they put this Drosophila. I thought it would be XY type, but I was a bit confused. Okay, okay, that was great. Thank God I knew this. This is something I found as a mistake. I don't know how teacher meant it as XY type. I am confused, by the way. I'm actually really confused. Oh God, what? 18 views. Okay, there was one person watching me. I don't know who it was. Two different types of gametes are produced by female heterogamety. Yeah, that is ZW and ZZ. Then we have this man. So technically, this is undergoing mitosis and this is going meiosis. Yeah. So this will infuse with this to form the female, and this will just go undergo parthenogenesis to form the male. This is known as haplodiploid sex determination. Queen workers females and drone males all fertilized eggs form males. Interesting. So basically, in this case, with the males of the honeybees are haploid, which are containing 16 chromosomes, and the female chromosomes contains 32 chromosomes. Now this, okay, we'll check. Female heterogamety. This phenomenon of female heterogamety is a phenomenon in which the females exhibit a different, unidentical identical pair of chromosomes, chromosome pair, such that is ZW or chromosomal system, whereas the male would have the same Z chromosome with them. That's one thing. Besides the autosome, okay, autosomes are different. Yeah, so that's one thing about it. This is only seen in female heterogamety is seen in birds. Then elaborate on Hankin's role. He traced a nuclear structure and he found out in the spermatogenesis of the nuclear structure all through spermatogenesis in few insects, and found out that 50% of the sperms received the X received a structure, whereas the 50% did not. That is the X body, right? So according to him, yes, the structure after spermatogenesis. Alright, this is one thing. And then through this, he was able to determine the exotype of sex determination, through which it was seen in the form of a sex determination in the insects like grasshopper. That's how we can actually talk about it. Let's go. Don't worry, guys. I'll be doing the lectures soon, and after the exams are finished, we'll actually do some short revision of all the lectures, and then we'll continue with the batch so that we can smoothly run it. I know I'm really tired with this, but can we just do this? No, I'm not tired. Yo, look at that. This sup, bros cooking. Thank you very much, by the way, Super VFX. I know I'm reading it right now. It's so late. I think yeah, he sent it at like 12:46, 3 hours back. That is crazy. And then there was another guy saying sup like horse XD. I have to like give him a reply. I did not reply back to him. Looks like this is also done. Let's go! Oh, finally to this one. 4.7 mutation of a gene. This topic is also nice. I like this topic. It's not that tough. And finally, bro, we are coming closer to genetic disorders. This one is a big. This one is a bit big, so it will take some time. But this is easy. I know that this is easy. This one, okay. I know. It's a bit. Okay, okay. It's not that easy, but this is the tough part. I feel like this is the only tough part. This one. If you like somehow complete this up, we cook. You got me right. You got me. Like literally, I'm literally going through each and every question too. If you don't know, that is taking so much of my time too. What are the causes of variation? Gene causes of variation. Like literally, different characters are formed, right? What? Let me just take my purse out. I don't know why it's literally dangling around my pack, around my pocket. Like it's, I don't know. It's just standing up like very crazily, bro. Any comments or anything that these people want to say? What's up? Because I know that some people are like, hey, yeah, man, how are you doing? Yeah, yeah, I know that, I know that. I know it, bro. I found an error in the notes. 4.6. Let me also confirm it through my textbook because textbook is also the deciding factor here, right? I still didn't see Drosophila and XY type chromosome. What? If I find it, then teacher is correct. That's what I know for sure. But the fact is, I've never seen XY type. Like, I didn't see the sex determination for this Drosophila thingy. One minute, let me just does again last time. Trust Drosophila follow. Yeah, follow. Yeah, just to give me that. Let me ask others and check. Sorry again. I thought I was wrong. Okay. I thought I was wrong here. What? Okay. So confused. It's not even given in the textbook. Okay, never mind. Oh, sorry. Okay, that was a bit confusing if I'm being honest. Google literally tricked me, man. I thought I was being correct here for a while. Never mind, let's just move on. Hmm. Mutation is a phenomenon which results in alteration of DNA sequences and results in changes of the genotype and the phenotype of an organism. Sure. In addition to recombination, ouch, ouch, ouch. In addition to the recombination, what? What's it paining? Yeah. In addition to recombination, mutation is another phenomenon that leads to variation in DNA, causes chromosomal aberrations, loss, deletions, or gain, insertions, duplication of a segment of DNA, deletions, causes frame shift mutations. So chromosomal aberrations, that is, so basically the frame shift mutation causes chromosomal aberrations, that is insertion, deletion, or loss. Man, this is gonna take a lot of time. We only have 2 hours left. I don't know if this is enough for the next part that I'm gonna do. This is important, I believe. That's what it says. Hemophilia, cystic fibrosis, color blindness, sickle cell anemia, phenylketonuria, thalassemia. Here, Klinefelter syndrome, Turner syndrome, Down syndrome, mainly determined by alteration or mutation in a single gene. Here, is mainly determined by the, you know, the excess presence of or addition of or losing of a chromosome. So that's variations of the chromosome number. That's what I know. We'll check this out nicely, so we'll get an idea. Many chemical and physical factors that induce mutations, these are referred as mutagens. Point mutation, example sickle cell anemia. Chromosomal aberrations are commonly observed in cancer cells. Oh, so frame shift mutation is seen in cancer cells. That is, chromosomal aberrations are seen in cancer cells. That is, insertion, delete, or like loss, deletions, or gain, insertion, duplication of segment of DNA. Oh, okay. Of segment of DNA being inserted or added or gained or deleted or lost, um, that is known as frame shift mutation, and that is chromosomal aberrations. The change in a single base pair of DNA is also known as point mutation. That is sickle cell anemia example. This also causes a lot of changes in phenotype and genotype. So that's one thing too. Like changing the shape of the RBCs. So yeah, I can understand. Then, ouch. There are many chemical and physical factors that induce mutations. So that is mutagens. Examples: UV radiations. There was another one. I don't know what was it called. We'll check. Mendelian disorders, and that is determined by alteration or mutation in the single gene, transmitted to the offspring on the same lines and as the principle of inheritance. We're gonna do this, guys. We're so close. We're so close. Phenylketonuria. Okay. Alterations of DNA sequences and also causing the change in phenotype and genotype. Genotype? Yeah. Then, in addition to recombination, mutation is another phenomenon that leads to variation in DNA. So not only recombination, it's you know the mutation that will lead to this change. So insertion, okay, so segment of DNA being inserted or like inserted, added, or lost or gain, sorry, lost or deleted. That is known as the chromosomal aberration, which is also known as frame shift mutation, seen in cancer cells, and that is dangerous though. And then we got the single base pair being changed or altered in DNA is known as point mutation, and this is an example of sickle cell anemia. That is still crazy because a single base pair being changed would be crazy, bro. There are many chemical and physical factors that induce mutations. These are referred to as mutagens. Example: UV radiations, inborn error, metabolic disease, metabolism. Okay. This inborn error of metabolism is basically autosomal recessive. The only thing that is autosomal dominant is myotonic dystrophy. This is autosomal recessive. This is sex-linked recessive. This is, yeah, okay, that's it. It's not given as sickle cell anemia. Yeah, but cystic fibrosis, that is autosomal. Let me just check. I'll check this one. And then color blindness. Color blindness, that is sex-linked recessive. Then phenylketonuria, thalassemia. I remember writing down sex-linked dominant too, if you don't know. In one of my live streams early, when I was studying this, I actually did write down sex-linked dominant disease too. Now I don't know where I did it, but let me just check. Okay, so it is called as Rett Rett syndrome. Rett syndrome. Okay, so if anyone discusses about, okay, so progestin, is that progestin thing you called? Like, I still remember what it was like, if they are asking this, and that's progestin. mm, I forgot that name. Okay, I forgot that name out of the syllabus thing, progestin. Okay, that is a part of basket proteins. Okay, we'll study hemophilia a bit and get the idea of this too. Then we'll do the questions of this. This also kind of has those crosses, so we'll do that. Should be right. Even though the chances are low, it should be. Is it possible to have a hemophilic girl when a cross is made between a carrier woman and hemophilic man? Should be. XHX, XHY. Yeah, it should be. It is possible. But then there are low chances. That's what it says for hemophilia. It is extremely rare for females, but it is not that much rare for males. So a single protein that is a part of cascade of proteins involved in the clotting of blood is affected. Uh, so this is like unaffected female. This is a carrier female, which means that it is carrying the disease, but she is not affected by it. Then this is an affected female of hemophilic. This is which is a very extremely rare condition. This is it is a healthy male, an affected male, and this is an affected male, which is affected by hemophilia. So this is a hemophilic male, male. Then causes of variations, we'll check that. Causes of variations, it should be mutations, then recombinations. If it is mutations of the gene that is being formed, then we can say that the mutations are basically or technically are the ones that are doing the alteration of DNA sequences. And what else? Yeah, that's it actually. It's not much. We'll just go through this a bit. This one. Phenylketonuria, as earlier discussed, it is an example of, okay, 1 minute. Oh shoot! What? That is crazy. Oh shoot! What? Okay, wait. I will. How do you guys? Do you guys can even see that notification? Like what? Nah, I don't know. Like how? Okay. It's fine. Let's focus. Let's focus. Uh, Okay, let's focus on wait. Wait. Okay, so we're so close, guys. We're so close. These two, and then we'll do some diagrams, drawing, and then we'll do neat questions. All right. So let's not give up. We can do this. Wait a minute. Wait. The sickle cell anemia. Okay, let's go for this one. Before that, hemophilia. Let's go. And then the questions, right? Yeah. All right. So I think I didn't expect that I will complete this that fast, though. Whoa. Like, actually, bro, that's so fast. Okay, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Okay. So let's like I don't remember this, but I think it was related to thrombins. I'm not sure. It is an affected individual. A simple cut will result in non-stop bleeding, which is very dangerous. And bro, it's kind of unfair seeing this like heterozygous female carrier for hemophilia may transmit the diseases to son. But like the possibility of female being hemophilic is extremely rare, but then the males have that higher that have a bit higher chances. So yeah, like in autosomal, yeah, let's say that both of them are having like equal chances, but for this sex linked, it is like females having less chance, but male has more. So yeah. Oh guys, you're so close, man. We can do this. Oh shoot, we are so close. Come on, let's go. Wait a minute. Mutagens, physical and chemical factors that induce the mutation is referred to as mutagens. UV radiations. That is known as this genetic disorders. Mendelian disorders and what else? Mendelian disorders, chromosomal disorders, phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. Okay. Okay, then we'll complete this every part. Wait a minute. Yeah, okay. So single protein is affected in the cascade of proteins that is basically affecting the involved in the clotting of blood. Okay. Like I think there was a question in the exam that was asked based on this. Fathers should be hemophilic. Yeah. Well, that's technically true. The possibility of the female becoming a hemophilic is extremely rare because mother of this such a the mother of such a daughter should at least be a carrier, and the male then the father should be hemophilic in this case. That is very rare though, right? This time I think it says at least carrier, so it feels like this is also rare. What do you guys say about that? I'm not sure about it, but it kind of looks rare. Then, oh, okay. Yeah, this is correct. This is Yes, of course, it is possible, but it is extremely rare again. Causes of variation. Now, I think it means, there's mutations and recombination, but let's just check if it is true. You know what I mean? Genetic factors. Okay, this is what it says. Yeah, mutation, meiosis, gene flow, sexual reproduction. Yeah, I mean, this is general, not general. By the means of that, this is the main whole thing. Then, going more deeper, we get to know mutations. Mutations is one reason. So I think they are expecting the answer of mutations here. What do you guys say? And recombination too, because recombination is also one such factor that is like forming variations, right? Like non-parental gene combinations. So that's one thing. So let's go. What do you guys say? X X H X H Y X X H X H Y. Then carrier woman, hemophilic man. This is this. The X X H X H Y. Okay. X X H X H X H X Y and X H Y. So technically we, oh wait, this is the same thing, right? So we get a hemophilic girl here. There was one thing I've read in the textbook too about Queen Victoria. I remember that she was a carrier of this disease. You know, that's what it says. Queen Victoria shows, in yeah, the descendants, the family pedigree of Queen Victoria shows a number of hemophilic descendants, as she was a carrier of this disease. Wait a minute. Descendants. Okay. Okay. Because she was carrier of this disease. I got it. So let's go. We are at this part. Oh god, I feel such satisfaction. We can do this. Autosomal link recessive trait. And yes, color blindness. And then sickle cell anemia. I don't know why there are only two in this one page. They could have, I've thought of that. They will like, put this all thalassemia also in this. Like there is no need to actually like just get thalassemia. But okay, because then they can directly start from chromosomal disorders. That's why. But you know what? Let's just do it. No questions at all here. Let's go. Sickle cell anemia. This is autosomal linked recessive trait. Can be transmitted from parents to the offspring. When okay, this one I should be studying carefully because this is important. As far as I remember, this is important. And the way they are writing it as HB. This one is H. This one this one is XC. Yeah. This is also very rare. 0.4% females and 8% in males. Like the color blindness is actually very rare if you look at this type of thing. It is a recessive sex-linked trait and in which the eye fails to distinguish what? Red and green colors. 8% males and 0.4% females. In females, color blindness appears only when both sex chromosomes carry the recessive gene XC. The females have normal vision but function as carrier if a single recessive gene for color blindness is present as XXC. Yeah. Okay, so I think my notifications are not coming through phone. It's coming through laptop. I'm not sure about that. Okay. Okay, wait, what? Yeah, blind boy is there. Carrier girl is there, and then what else? Normal girl and normal boy. So that's one thing. Wait, if it is extremely rare in females, then this should also be rare in males. Like, just like color blindness. If you can look at this, it's like 8%. That's just pretty much low. And 0.4% is very, very low, in fact. Like, that is very low. So, I'm expecting the same, almost same percentage for hemophilia. Who knows? And hemophilia is very dangerous, by the way. Like, just by single cut, the blood doesn't stop bleeding. That is crazy. minute. It's 4 o'clock, bro. I know the time is going a lot fast, but we are close. We will complete full biology today itself, and then we will be doing diagrams, textbook questions, and this. If I get some enough time, man. No, that's what I feel. What is this? HD. Okay, I don't know what that is. It's a link. It's a link. I don't know. It's a YouTube video, like, as far as I know. Is it tissue? No, there isn't. Okay. Oh, there's a one concurrent viewer watching me. Hi there, man. Okay. Looks like we are doing fine. We are doing fine, bro. Let's go. Tomorrow, only 15 days left for 75 Ultra Hard Challenge, guys. Remember that. 15 days left. Wow. Wait, when does this end, though? When does the challenge end? I don't know the date. And we have crossed over 8 hours, which is pretty great. Okay. Man, I don't like that light coming up, and but still, it's fine. It's giving clear vision to you guys, I guess. That was effective overall. I like it. That was really effective. We did biology nicely, super nicely. XXC, that's cross XY. Okay. So, okay, if we need to understand what color blindness is, because I think there was a question I asked based on this too. You know what's surprising that they asked this question to explain instead of sickle cell anemia and thalassemia. Guys, do you want me to watch a video based on this, because this concept is not easy at all as far as I know from my side. This concept is not easy at all from my side. Like, from my side overall, I feel like this is fine, this is easy. And we are coming to an end of this chapter finally. Like, oh my god, that is a very lengthy chapter, guys. Literally 4.8, and then there's one last one. Because the last topic of this chapter is easy, so I can easily complete that. It's only this genetic disorders like sickle cell anemia, color blindness, and thalassemia. Where is it? Thalassemia. Okay, so let's go. It can be transmitted from parent to offspring. That's true, and both the parent and partners are carrier for the gene. Okay, which means that, or yeah, autosomal, right? So carriers is possible. So this cannot be transmitted from the parents to the offspring, and both the parents partners are carrier for the genes, or heterozygous. The disease is controlled by a single pair of allele, HBA and HBS. Sickle cell, and this is the X chromosome, which is affected by that sickle cell genetic disorder, and this one HBA, Mendelian disorder, and then this one is unaffected. So one of the three possible genotypes, HBS, HBS, HBA, HBS. Oh, this is multiple allelism, right? What do you guys say? This is literally multiple allelism. I forgot about that. Dang, multiple allelism. Like, literally, three genes are governing the same character here. Show the diseased phenotype, okay? Then these are carriers, which means that they will carry the disease, and these are normal. Alright, alright, alright. So this is CTC, GAG, CAC, GTG. Oh, look at that. They just interchanged. Okay, never mind, never mind. This is a normal HBA gene. This is HBS gene. Mutation. mRNA, messenger RNA. That one strand is taken. There is a mutation that one base pair got changed. So U. Okay. So here in the beta globin chain, is it a beta globin chain? Just for reference, I want to know. Yeah, beta globin chain of the hemoglobin molecule. In the sixth position, the glutamic acid is replaced by valine. That is interesting. That is very interesting. At the sixth position of the beta globin chain of the hemoglobin molecule, we get this idea. The substitution of amino acids in the globin protein results due to the single base substitution of the sixth codon of the beta globin chain from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle-like structure. All right. 1 minute, let me just get a tissue paper. Oh man, that is a great streak. So overall, from the biology, we had gone through the textbook, all except for reproductive health. But we gone through the notes. We had like practiced the process, then made notes actually for some chapters. So I think this is overall a great progress. I feel like we had done great. What do you guys say? Oh, I forgot that today is June. Oh wow, May already got completed. June, June. I did not notice the date. I was like, oh, I did not notice that. Literally, bro. If this month gets completed, then we'll literally after this 1 month, we will be literally completing half of 2026. That is still crazy, bro. That is so crazy. Like like passes. Time passes so fast. Time passes so fast. 19 views. Okay, I think that was the previous person who was watching me. That's why. Okay, and I like that. And I've noticed one thing. Like my laptop has changed its WhatsApp logo, and I really like it. I appreciate it. So W WhatsApp sticker. Which the eye fails to distinguish red and green colors. It occurs in 8% males and 0.4% in females. Color blindness appears only when both these sex chromosomes carry the recessive gene XE. Alright. The females have normal vision but function as carrier if a single recessive gene of color blind is present XC. Alright. Yeah, that's true. However, in human females, the defect appears in the presence of a single recessive gene XEY because Y chromosomes of male do not carry any gene for color vision. Yeah, yeah. Wow. Like I still wonder why does Y chromosome do not carry any gene for color vision? Why? That's the question. Why is it not Y chromosome? Like that's the question I usually carry out. But that's interesting to know. Gag to goog, GAG to GOG. Acid in the globin protein results due to the single base substitution, the sixth codon from GAG to GOG. Hmm. Okay. You know what looks interesting about this? Like, um, robots, right? Mainly if they want to like work really highly intellectually. If you want to make some kind of things which should work highly intellectually, then usually you might need some kind of coding work. And I feel like the human body has that, like biologically coding. I feel like those nitrogenous base pairs are like some basic coding, and that is crazy because if you make a single error in a coding, then a whole thing like literally shuts down. Like the not whole thing, some parts will not work at all, and I feel like this is what happens here too. That's interesting. distinguish red and green colors. It occurs in 8% males and 0.4% females. Okay. Uh, Autosomal recessive trait. Okay, we finally moved to thalassemia, are we? Okay, just one thing. Man, okay, this one. We are so close, bro. That's three, then this one. Abnormal absence or exchange, excess or. Okay, you know what? Let's just complete this first. We'll complete this first, then we will move on to, these three will take some time. Okay. So thalassemia, okay, not that one. Chromosomal disorders, absence or excess or abnormal arrangement of one or more chromosomes is known as is the one reason that chromosomal disorders exist. Failure of segregation of chromatids during cell division cycle. That is the reason why we actually face the aneuploidy, that is gain or loss of a chromosome. Failure of cytokinesis after telophase stage of cell division results in an increase of a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. Down syndrome. The cause of this genetic order is the presence of an additional copy of chromosome number 21, trisomy of 21. Okay, yeah, three chromosomes, right? 2n plus 1. My bad. The disorder was first described by Langdon Down. That's why the name is Down's syndrome. 1866. I think Down syndrome is really overrated. I mean, in the sense of knowing about this disease, like I think if you ask any other person about Klinefelter syndrome, any person like who hasn't studied a very good amount of biology, then there's a great chance that he might not know this, but he may know the name of it. But I think he will not know the definition of this, or the idea or the symptoms of this Down syndrome. Why did I hear? Oh, what? Was there any? No. Okay. No. Okay. I thought there was a notification sound. Sometimes what happens is that my laptop would make those notification sounds, but the messages gets cut off so fast that sometimes I might not even see that. So yeah, that happens sometimes. I've seen it. It gets so fastly cut off. I don't know why it happens like that. But hey, dude, let's go. Then we'll close all these windows because at the end we need to oh god, let's okay, sickle cell anemia and thalassemia. That's to differentiate. Okay, this one is male, female, sex unspecified, affected individual. Then mating. This is consanguineous mating, mating between relatives. Then this is the mating parents above and children below, leftmost. Yeah, from left to right. So, this one is basically. They're actually like mating, and then they form this affected male. Then this one has five unaffected offsprings, which is interesting. I don't know why did they give the specific symbol for this one, but yeah, they just gave it. I think because family trees are really big, and sometimes if there are more than five offsprings, then it could be difficult to put them together. So five unaffected offsprings as this, that's fine. But this looks like, but then the problem with this is that how would you know that what's the sex of this thingy? Like you're saying unspecified sex here. Okay, that's fine. But how would we know whether that in those five, which of those are male and which of those are females? So that's one thing. And I don't think so anyone will give sex unspecified. I'm sure about that. Like, that would be very bad if they give this sex unspecified for pedigree. Anyways, aneuploidy means gain or loss of one or more chromosomes, monosomy or trisomy, increase in the whole set of chromosomes, polyploidy. This happens due to the failure of segregation of chromatids. This happens due to the failure of cytokinesis, as far as I know. Karyotype of Turner syndrome. Uh, we'll write it as X, no, 45, because X, yeah, 45XO. 45XO, my bad. And then that's the Turner syndrome. Why does color blindness occur in 8% males and but only in 0.4% of females? Now, color blindness is a okay. We need to explain how this happens. This is due to the color blindness is basically a sex-linked recessive trait in which it will affect the allosomes of the males and females. In females, it can one of the X chromosomes can get affected, and thus the female can become can carry the disease but will be unaffected by the color blindness. However, since males have only one X chromosome, they would be affected by it once it gets the once it is affected by the trait of color blindness. So that's one thing. And then females, if both the X chromosomes of females are affected by it, then the females becomes affected. So that is why the ratio, sorry, not ratio, the percentage of females being affected is less. Okay. Write about the symptoms of Down syndrome. So there is a lot of symptoms. It was discovered by Langdon Down in 1866. How is it caused? That's a good question. I have to check. I did not study that. That's a good one. Okay, okay. So we can answer this. So chromosomal part is done nicely, and even color blindness is. I have a good idea about that now. That's great. That's nice. Also, just let's just look at this again. What is it? Is fails to distinguish. It affects the red and green cones of the eyeball, and it fails to and the failure to distinguish the red and green colors. That is one thing. All right. So that's one thing about answering it. Then what else? Can I actually like see if any notification? Yeah, check for new messages. Check for new messages. Huh? Say no to tobacco. On World No Tobacco Day and always. Today is No Tobacco Day. Bro, that would be diabolical if a person is smoking today. You know, like it says No Tobacco Day and it's like you're literally smoking. That would be diabolical. Literally, it's written in the calendar. I can show it to you. Let me just open my calendar. Where's calendar? Huh? Bro, it was just saying that it was No Tobacco Day, right? Huh? Yo, now you're trolling. What is this? I don't know when is. Wait, okay. Let's just type it here. When is. Okay, this is out of context, but Tobacco Day celebrated. Okay. May 31st. Oh, yesterday. Oh. Bro, that would be diabolical if a person is literally smoking. If the person was smoking yesterday. That would be crazy, bro. That would be crazy. But yes, being serious about this situation, tobacco is really, harmful, and we should stay away from that type of chemical, which is used in smoking. Yeah, symptoms, right? Let me just say the symptoms. Uh, like opened and furrowed mouth, then observable palm trees, then flat back of the face and flat face itself, and then congenital heart disease. Then what else? What else we need to say? Yeah, psychomotor, mental and physical retardation, and what is the other one? There's a lot of symptoms to this syndrome. That's crazy, bro. Um, Is it? And no, 1 minute. Like we know that it is caused because one more. Yeah, I don't know why I forgot about that. That the one more autosome will get added to the 21st pair of the chromosome, or 21st pair of the autosome. That is trisomy of autosome. So basically, due to aneuploidy, this thing is happening. So yeah, let me just check though. Yeah. Additional copy of the chromosome number 21. Trisomy of 21. I still remember it was there in this. Okay. looks like we are cooking, guys. We are doing great. We have completed this. Now we have to deal with sickle cell anemia and thalassemia. That's it. Two things remain. That's it. Two things remain. And 1 minute, I'll go to toilet and come back. Oh my god, man. Okay, so looks like yeah, I came back. I was praying also. Okay, bro, no way, bro. I literally left. I did. I go to toilet. I did ablution or wudu. I just prayed. I came back. 3 viewers. Literally just saw my live stream and then went. Like, what is going on, guys? Bro, what? It's like, I don't know why there are so many viewers watching me at that moment. Like, that's very weird. 3 viewers. Dang. Oh my god. Let me just fix my hairstyle. What is this? And dang, it's literally 9 hours now. Our stream has been going for 9 hours. Yeah, I mean that's true because we have been working a lot. Okay, so thalassemia and sickle cell anemia. You guys are the ones remaining. What do you want to say? Okay, before that, we'll just go through this. Just read it. Due to the presence of an additional copy of X chromosome resulting into a karyotype of 47XXY, then this one, such an individual has overall masculine development. However, the feminine development of gynecomastia is also expressed. Such individuals are sterile. Then Turner syndrome. Such a disorder is caused due to the absence of one of X chromosomes, i.e., 45 with XO. Is this X0? 1 minute. Uh, is it? Yeah. Wait. Okay. Okay. Where should I type? Let's say 0. No, how can I make it bold? Okay. Um, that's pretty weird. Am I supposed to write X0 or XO? Such a disorder is caused due to the absence of one X chromosome, and that's why the females are sterile. That is the rudimentary of their ovary, which means that the four is not fully formed, and secondary sexual characteristics are lacking. This is the question that I've been waiting for. Man, if this question comes, we'll be cooked if we don't study this nicely. Then gynecomastia. Okay, that is done. Only two things are there, and then we'll complete the full biology work here. The rest things that we need to do is like biology textbook questions. We'll go through that just like fast. We'll go through it very fast. Then diagrams. Diagrams we have to give some time because we need to practice those. I'll use my book for that. Like I need to practice ovary, then sperm, then what else? There was one thing. Graphene follicle. I need to open that notes for that first one. XXY. Gynecomastia is also expressed. Such individuals are sterile. Turner's syndrome. Such a disorder is caused due to absence of one of X chromosomes, 45 with XO. Then what else, bro? Such females are sterile as ovaries are rudimentary besides other features including lack of other. Okay. 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 What do you guys say? Like, if we get time, we may use it for the second chapter, where we'll go through this and yeah. I hope this is useful. Questions are good. You have to check. We'll be using AI to like fast do that fast question solving. How many questions there? So this has 21 questions. This has only 12, which is great. Okay. Then oh, I bet this will have a lot of them. This genetics chapter, which is 16. Okay, okay, not bad, not bad. We had done the crosses, so we can skip those. For molecular bases, some parts are not there for the exam, so like I don't know what all questions we have to attempt right now. So let me just check. Um, okay, 3' like this. That's crazy. This one. Ignore this. Ignore this. Ignore this, and ignore this. Okay. Okay, this one can be done. This one can be done. Okay. I'm not sure about fifth one. Okay, so these two can should be done. I hope we can actually like say the writing the answers appropriately. So I have to sleep nicely today, so that I don't forget many of these things that I've studied. That's one thing. Okay, let me just go through this. Okay, so can be transmitted this, okay, or carrier for the gene, heterozygous, controlled by single pair of allele HPA and HBS out of the three possible genotypes. HBS show the deceased phenotype. Heterozygous HBA-HBS individuals are carriers, and HBA-HBA are normal. The defect is caused by the substitution of glutamic acid by valine at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acids in the globin protein results due to the single base substitution of the sixth codon of the beta globin chain, that is JG to GUG, YAG to GUG. Okay, that's a weird way to say it, but okay. Uh, Guanine, adenine, and guanine. This is guanine, uracil, and guanine. Huh? Okay, okay, I don't know what was that. I don't know. There was a change in taskbar. That's why you cannot see the taskbar. That's why. My bad. Mutant hemoglobin molecule undergoes polymerization under low oxidation tension, causing the change in the arch. Okay. Okay. Mutant hemoglobin molecule undergoes polymerization under low. Okay, so mutant means changed, right? I just want to know the name of the mutant because sometimes I got confused. So mutant means changed. What? No, that's not the thing I meant. Mutant, mutant. Oh, okay, okay. Literally, that gave me a direct answer. That's what I needed actually. Wait a minute. Those are so fracking. Yeah. Biconcave disc 2 elongated sickle-like structure. Okay. So I think I'll have to check NCRT Punch because there are some questions which are really good. I have to check. I just pray to God that everyone gets good marks, for tomorrow's exam. الله أكبر. We'll pray. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. رب زدني علما. اللهم إني أسألك علما نافعا. I'm just praying right now because I want to seek good amount of knowledge from God so that I can use that good amount of knowledge to get good marks in this exam. Like I'm doing it right now. Because I don't know why, I just feel less confident for some reason. Like I know that I can do it, but I still feel less confident for some reason. I don't know why. But guys, please don't give up. I know you can do this. Trust me. We are not gonna give up. HBA peptide, HBS peptide. HBA peptide will have this one amino acid different. So in that peptide chain, the glutamic acid of the sixth one will get changed to, valine, and this amino acid substitutions results due to the change in the sixth codon of the base pair of the DNA, that is JG to GUG, through which is the point mutation, by the way, beta globin gene from GAG to GUG of the beta globin gene. Okay, the mutant hemoglobin, that is due to the hemoglobin which is affected by the gene mutation, will undergo this. I didn't know the exact meaning of a mutant. So, literally, mutant is a biology word. Then it undergoes polymerization, getting and then under low oxygen tension, causing the change in the change of shape of RCB from biconcave disc to elongated sickle-like structure. Yeah. Oh. Oh. Okay. Okay. My bad. Into the wrong direction. Okay. This is an example of multiple allelism, right? Could be multiple allelism. What is mutagens? I'm just going through this again because not to forget. Physical or chemical factors that induce changes in the mutation that induces mutation is known as mutagens. Example: UV rays. There is another type of rays which induces this, and I want to know. I think it's coming under the electromagnetic waves. I know that's for sure. Ouch. Okay. Oh wow. Yo, how am I cracking my leg fingers, bro? Like I'm somehow doing that. What? Never seen that coming before. Anyways, I don't know how I'm doing that. Can I do that for toes? No, I cannot do that for toes. However, in human males, the defect appears in the presence of single recessive gene, XCY, because Y chromosomes of males do not carry any gene for color vision. Bro, my bad. Finally, we have done with this part. We have still this thing. Oh god. Alpha thalassemia. Okay, so you can say that we have dealt with this one, and now we have this. Wow. See, there is no people watching me now. That's so weird, bro. Like, there is no increase in views. Like, what? How is it like people are clicking on my channel videos whenever I'm like not there? What? Yo, what? That's like a troll or something. Thalassemia, autosomal recessive trait. I don't know, man. I'm a small creator, so even one subscriber or any one viewership influences me a lot. Trust me. I'm a small creator, that's why. Now, those big creators will. I'm not saying that those big creators are like really greedy or anything. I'm not saying that they're not satisfied by that one subscriber. I'm saying that, they also have faced this. They were eventually that one type of small creator. So I do agree with that. But I'm also grateful with the amount of things I have right now, like laptop itself. I'm capable of like streaming smoothly, like, or else it would be even more difficult if you don't know. I know. I'm still grateful that I have this table too. Like, I don't need a study table for this. Like, this table is so effective. I like it. Even though my leg kind of gets squashed with the ground because of the car because I sit on carpet, it's still fine. I don't sit on bed because I will feel asleep. That's why. But sometimes I do that. That's why, because sometimes some people come for vacuum cleaning, so they have to clean this carpet up, right? That's why. So I have to like go there and sit and study. Plus, it's very cold here. If you look at that AC, it's super cold whenever you sit there. So like that's also one problem. So I get allergies from there. Like I'm really sensitive to cold. So that's one reason. Anyways, let's move on. Involves the genes HBA1, HBA2 on chromosome 16, which is affected. There are two gene loci as and so for alleles. Alpha thalassemia result in decreased alpha globin production, thereby leads to anemia. Okay. HBB genes on chromosome 11 are also inherited in an autosomal recessive fashion. Thalassemia is a quantitative disorder, while sickle cell anemia is a qualitative disorder. Sickle cell anemia is beta globin chain the amino acid is changed at like sixth position. Here there is a mutation on the 11th chromosome for beta thalassemia, and alpha thalassemia it's like 16th chromosome for HbA1 and HbA2. So that's what I can say. Uh, what are the causes of this? Like I want to learn enough things for thalassemia because I don't want to like just miss out many things. I can say that sickle cell anemia's amino acid substitution takes place. This is because of the sixth codon of the beta globin gene gets GH is altered. That is GAG to GUG. That's why that is point mutation. So the mutant gene that we see of mutant hemoglobin reflects in the shape of these has its shape changed from a biconcave shape to sickle cell shape, and causes low oxygen tension. So less oxygen reaches to the body parts, which means that less energy is there, less thinking can be seen, and that's a really broad problem. If you look at sickle cell anemia, that's not a good problem at all. Sorry, that's not a small problem at all, causing the change in the shape of RBC. Okay, under low oxygen tension, my bad. Oh, yeah, mutant hemoglobin undergoes polymerization. I forgot that topic. That's one thing too. It undergoes polymerization, and I know it's stressful, but let's do it. Overall, I would say it's better than the previous ones. Last time in 11th standard, I suffered with one chapter, Animal Kingdom, and that wasn't a good experience. But I feel like this time I can cook all the chapters now. Like I have that confidence. It's because of God. It's because of you people. Because you give me the strength, support. It's because of my parents. It's because of my friends. That I also get the help from them too. And the last person that I would like to thank is to myself. That's it. The last person. Because at the end, I'm not the one being that influential. That's what I believe. I believe that it is you people. However, I like to, have a feeling that it should be only me who should influence this. It should be only me myself who should do this type of work thing. But I want to just say that I should be giving credits to everyone because somewhere some other sometimes like I know my grammar is a bit bad here, but somewhere sometimes and even in any other type you're somehow helping me. You're just somehow helping me, and my parents are helping me. My teachers are also helping me. Let's say that too. Oh, I forgot about teachers. My bad. The reason why I'm saying let's say that, I don't know why I said that. Teachers are helping us. My bad. Then who else? Who else did I forget? Of course, my friends are also helping me. I also help my friends, but I feel like my friends are the ones who honor me and help me a lot, so I respect them too. Then you guys are the also one are the ones, so I really appreciate each and every one of you. And yeah, I actually noticed one interesting and weird fact actually. Like uh, I was like not going for live streaming on for like 7 days straight. I guess I'm not sure. Maybe like 5 to 6 days. Okay, 5 days. Let's say 5 days. So during that timeline, what I noticed is that I don't know it's because of the recommendation algorithm, But my day 29s of 75 ultra hard challenge is one of the most popular ones out of all the 75 ultra hard challenge videos. And I got to know that each and every challenge was consisting of really high amount or value. Like, uh, each and every challenges days are really valuable for me. So that's one thing, because I feel like I'm improving each and every day. Okay, that's one thing. Okay, let's go. Yeah, let's say that I have done it. I have completed this. I have mastered it. I'm happy. But one thing about this is that another thing about I want to discuss is, like, I don't know what happened. There were literally like 192 views in day 29. It was first, I guess, 70 views. Okay, I don't know how I gained these many views actually, because that usually does not happen that fast. And then day one yeah, day 29, I saw 190 views. I got to know this because there was a comment in my channel saying studying 10 hours is mental. I was like, yeah, I mean, I understand that 10 hours is crazy and all, but I'm kind of a person who loves hardworking, so I do it sometimes. Not sometimes, I mean, yeah, you know what, let's say sometimes, because there is a variation in channels. But they didn't see my other live streams. Like, there are some other live streams which I've worked really hard, some live streams which I've interacted with you guys really nicely. But out of all of this, they just saw that one. Which is pretty weird. Recommendations are pretty weird, man, sometimes. I think there is a pattern based on this. I believe that there is a pattern on going viral, but or anything or in a strategy to go viral. But I don't know what is it. Like, that's one thing I noticed. Thalassemia and sickle cell anemia. If we talk about this, they also have similarities. Like, both are autosomal recessive traits. Both are affecting the hemoglobin, right? That's one thing I've noticed. Like, they have similarities too. Thalassemia is a quantitative disorder. Sickle cell anemia is a qualitative disorder. Then it the thalassemia are of two types. Alpha thalassemia are, like, causing a gene mutation due to the gene mutation in the in the beta globin chain. Is it beta? No, no. Due to mutations in the HBB gene on chromosome 11, also inherited in an autosomal recessive fashion. You see what I mean? Like, thalassemia is very complex. Let's have an explanation based on this. What do you guys say? Oh, this one is the renate one. Okay. Who is this? Oh, sample paper. No, 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 not this one, bro. What am I checking? Look at this. Look at this. Uh. Oh, he did memes here. Jackie Chan, Jada Lal. I don't remember which one. Yeah, this one.
Not every student starts as a topper, but
1 minute, guys. Do you want to say?
every student deserves a
Let's see.
chance to improve. At PW Gulf,
Sorry, I'm literally watching PW. Sorry. Five traps that destroy your teenage life. XP her percent growth. Good video. Good video. I like it. I like it. I like it. Okay, never mind. Why is there politics here? Okay, my bad.
Oh, hello everyone.
Let's not discuss about that. Yo, Weepu Bhaiya. Come on, come on, come on. Mutation.
B is the answer. D is the answer. So A. B and D.
जनरेट करने वाले केमिकल या टू डिफरेंट बात, तो आंसर निकल ही गया। क्रोमोसोम इज सेम इन बोथ, है ना, XO
B and
कंडीशन है
D.
उसमें साथ, तो ऑटोसोम के
Okay, so we can cope this question. Okay, never mind. But I'm not here to deal with these. I'm actually here to deal with. Oh,
फिनाइल
look at
कीटोन यूरिया जो
that.
आज
You look at that. Yeah, yeah, that one. Oh, yeah. I can understand that relatability. You can see that relatability, bro. Yeah, this is the one I wanted to discuss. Thalassemia. Phenylketonuria? No, I don't need that. Thalassemia. Okay, so let me go back. This is a bit complex one. So we know that this is phenylalanine, which is undergoing the process of phenylalanine hydroxylase to form tyrosine. This is the enzyme, by the way. But since the gene is influenced, or like if there is a change in the gene, then mutation in a gene that can make enzyme phenylalanine to convert into phenylpyruvic acid instead of going through this process, causing mental retardation, skin pigmentation, and hair reduction.
अब नहीं कर
Skin and hair pigmentation. Okay, hair pigmentation was there. Okay, I don't know why, but in the notice was given as hair reduction. Okay, never mind.
पाती, तो ये सारी चीजें, इट इज ऑल्सो
Let's move on.
ऑटोसोमल रिसेसिव डिजीज, CH यानी कलर ब्लाइंडनेस और हीमोफीलिया एक्स लिंक्ड रिसेसिव, बाकी सारी मैंने बताया हमारे सिलेबस में जितने भी हैं ऑटोसोमल रिसेसिव होंगी। इट इज अ क्वांटिटेटिव डिसऑर्डर, थैलासीमिया क्वांटिटेटिव
Thala
डिसऑर्डर,
for a reason.
जो सिकल सेल एनीमिया है वो क्वालिटेटिव डिसऑर्डर है,
हमें
यहाँ
हिंदी भी आती है।
क्वांटिटेटिव डिसऑर्डर है। जो हीमोग्लोबिन है, हीमोग्लोबिन फॉर्मेशन अफेक्ट हो जाती है ड्यू टू म्यूटेशन और डिलीशन ऑफ मल्टीपल जीन्स। मैंने कहा था मेंडेलियन डिसऑर्डर में एक जीन यूजुअली म्यूटेट होता है, पर कुछ केसेस में मल्टीपल जीन भी म्यूटेट होते हैं, वो आपका थैलेसीमिया की ही मैं बात कर रहा था। यहाँ पर अल्फा थैलेसीमिया और बीटा थैलेसीमिया हो सकता है। अल्फा थैलेसीमिया, थाला फॉर अ रीजन, थाला साहब का नंबर क्या है 7, 1 + 6, तो क्रोमोसोम नंबर 16 पे
Wait, who's Thala? Dhoni? What's Dhoni's number? Seven. That's literally Cristiano Ronaldo's number, right? CR7. Sue. Yeah, no, that's good. That's good. Okay, I like Ronaldo. I don't hate Messi, but I'm a Ronaldo supporter. I like Messi too, but then I'm not a Messi supporter. I support Ronaldo.
जीन्स पाए जाते हैं जिनमें म्यूटेशन होगी। जैसे यहां पर क्रोमोसोम नंबर 16 जो है, ये आपके पास दो होते हैं, एक मम्मी से आया, एक पापा से आया। करेक्ट है, एकदम करेक्ट है गुरुदेव। यहां पर HbA1 जीन है, HbA2 जीन है। Hb यानी हीमोग्लोबिन, A मानी अल्फा, Hb मानी हीमोग्लोबिन, A मानी अल्फा, तो Hb अल्फा 1, Hb अल्फा 2 दो जीन है, Hb अल्फा 1, Hb अल्फा 2 दो जीन है, टोटल 4 जीन हो गए दोनों क्रोमोसोम नंबर 16 पे मिला के। जैसे एक गाड़ी के 4 पहिए होते हैं, तो गाड़ी के 4 पहिए हैं, एक पहिया अगर फेल हो गया तो भी 3 पहिए पे गाड़ी चलती रहेगी। 2 पहिए फेल हो गए, ज्यादा कबाड़ा हो गया। 3 पहिए फेल हो गए और ज्यादा कबाड़ा हो गया। 4 पहिए फेल हो गए और ज्यादा कबाड़ा हो गया। यानी 4 में से जितने पहिए आपके खराब होंगे उतनी ही गाड़ी की कंडीशन खराब होती जाएगी। यानी 4 में से जितने जीन म्यूटेट होंगे उतनी ही आपकी बैंड बजती चली जाएगी, उतना ही सीवियर आपको अल्फा थैलेसीमिया हो जाएगा। तो अल्फा थैलेसीमिया थाला जी का नंबर है आपका 7, तो यहाँ पे 1 + 6 आ जाएगा, यानी 16 नंबर क्रोमोसोम पे 4 जीन है, HbA1, चारों में से जितने जीन का कबाड़ा होगा उतना खतरनाक थैलेसीमिया आपको हो जाएगा। दूसरा है बीटा थैलेसीमिया।
I'm not copying anyone's video. This is just a reaction channel. Just think about it as a reaction. And shout out to PW Neat again. Shout out to PW.
बीटा, B मतलब सेकंड अल्फाबेट, 1 + 1 that is 2।
If you want to look out this channel, you can look out here. It's written as PW Need. Genetics Part 3, complete unit in one shot, D2026, repeat set. Okay? You can look at this. It's there, don't worry.
तो क्रोमोसोम नंबर 11 पे आपके पाए जाते हैं हबीबी जीन। हेमोग्लोबिन बीटा, हेमोग्लोबिन बीटा
हबीबी
ये एक बाइक जैसा है या साइकिल जैसा है, इसमें 2 जीन है। एक क्रोमोसोम नंबर 11 जो मम्मी से आया उसका हबीबी, एक क्रोमोसोम नंबर 11 जो पापा से आया उसका हबीबी। अगर एक जीन म्यूटेट हो गया तो कम बीटा थैलेसीमिया होगा, 2 जीन म्यूटेट हो गए तो ज्यादा बीटा थैलेसीमिया हो रहा होगा। यह है आपका मिस्टर थैलेसीमिया जो हमने यहां पर सीखा, हमने यहां पर देखा। करेक्ट है जी, करेक्ट है मेरे बंधु, करेक्ट है।
Okay, let's try to solve this question. Down syndrome, 21st, 1A3. Okay,
कुछ सवाल देखें। डाउन
16th chromosome.
सिंड्रोम
I'm
को
thinking it's
21st
A. Yeah, it's
ट्राइसोमी कहा
A.
जाता है। 21st
This
क्रोमोसोम
is
पे
the
इफेक्ट
answer.
करेगा तो ए का 3 हो गया, ए का 3 एक जगह पर है, आंसर मिल गया। अल्फा थैलेसीमिया थाला फॉर अ रीजन 16 क्रोमोसोम, बीटा सेकंड अल्फाबेट 1+1 यानी आपका 11वां क्रोमोसोम। लाइन फिल्टर सिंड्रोम में आपका क्या होता है, एक्स क्रोमोसोम बढ़ जाता है मेल में, टर्नर सिंड्रोम में एक्स क्रोमोसोम घट जाता है फीमेल में। मार्क कर लीजिएगा जो भी आंसर आएगा। Which of the following statements are correct about Klinefelter syndrome? The disorder was first described by Langdon Down. Langdon Down ने डाउन सिंड्रोम बताया था, ना कि लाइनफेल्टर सिंड्रोम। तो ए तो पक्का गलत हो गया, ए तो पक्का गलत हो गया, एलिमिनेट कर दो इन 2 ऑप्शंस को। तो such individual has overall masculine development, however the feminine development is also expressed gynecomastia हो जाता है। तो बी पक्का करेक्ट है, बी पक्का करेक्ट है, यानी उत्तर यही आएगा क्योंकि यहां पे बी को करेक्ट नहीं बता रखा है। अफेक्टेड इंडिविजुअल इज शॉर्ट स्टेचर्ड, यूजुअली ये
That's true. That's no wait. Short statured? No, no wait.
डाउन सिंड्रोम में देखने को मिलता है, और फीमेल वाला जो आपका टर्नर सिंड्रोम है उसमें भी देखने को मिलता है, पर मेनली डाउन सिंड्रोम में लिखा हुआ है एनसीईआरटी में। इसको अभी के लिए हटा देते हैं।
Wait. It's Klinefelter's. My bad. What am I doing? I forgot this is Klinefelter.
फिजिकल साइकोमोटर एंड मेंटल डेवलपमेंट इज रिटार्डेड, डाउन सिंड्रोम की ही बात हो रही है। सच इंडिविजुअल्स आर स्टेराइल, ये आपके लाइनफेल्टर और टर्नर सिंड्रोम की बात हो रही है। तो आपका बी एंड ई करेक्ट।
Yeah, these individuals are sterile. Females are also sterile, and Turner syndrome
हो जाता है। ये बात आपकी हम पहले ही जान चुके थे, बस हमने वैलिडेट कर लिया है आराम से।
I need to make a public apology to every grandmother and parent who's bought a robot puppy because I lied. Yes, it's true that this robot puppy uses smart sensor technology to recognize your grandchild's.
ब्रॉड पाम विद सिंगल पाम क्रीज इज विजिबल इन अ पर्सन सफरिंग फ्रॉम। ब्रॉड पाम है, चौड़ी हथेली है और पाम क्रीज देखने को मिल रही है। ये आपके डाउन सिंड्रोम वाले बच्चों का एक बेसिकली यहां पर फीचर है ना, सिम्टम है, ऐसा कह लीजिए। गिवन बिलो आर 2 स्टेटमेंट। स्टेटमेंट 1, सिकल सेल अनीमिया एंड हेमोफीलिया और ऑटोसोमल डोमिनेंट ट्रेट। डोमिनेंट ट्रेट हमारी एनसीईआरटी में एक ही नाम लिखा हुआ है, मायोटोनिक।
Sickle cell anemia and hemophilia are disorders of the blood. Yeah, okay, fine.
Dystrophy. Sickle cell anemia.
But you know, I feel like this should be mainly for hemoglobin. This is okay. You know what? Let's go through.
Hemophilia are
If I get wrong, then what should I do?
disorder of the blood.
Eat myself. Okay, let's do this.
ब्लड में यहाँ पे आरबीसी की शेप चेंज हो जाती है। हेमोफीलिया के नाम में ही हेमो आ रहा है, यानी ब्लड आ रहा है। पहली स्टेटमेंट गंदी है, दूसरी स्टेटमेंट सही है। पहली स्टेटमेंट गंदी, दूसरी स्टेटमेंट।
No, I'm not wrong. Why did I beat myself even though it's not wrong?
सही उत्तर आ जाता है मिस्टर ए।
I didn't see that option. I should have said that.
ठीक है, अब मेरे भाई मैं आपको कुछ खास देने वाला हूं। दैट इज माय बैच लेक्चर। बहुत प्राइम लेक्चर है, बहुत खतरनाक लेक्चर है। एक लेक्चर जिसमें पेडिग्री सॉर्ट हो जाएगी, मतलब एकदम ब्रिलियंट लेवल पे हो जाएगी। इसको मिस मत करना। यहां तक पहुंच चुके हो, कांग्रेचुलेशंस, बिकॉज़
like you know I've fully completed this lecture and I was like watching this. Now pedigree, yeah, this one is very important. We'll go through this
in the chat section. इतनी बातें अगर लिखनी है तो बता दीजिए। यस,
pedigree analysis
करेक्ट है।
The idea that disorders are inherited has been prevailing in the human society since long. Analysis of traits that has been traced in several generations of a family is known as pedigree analysis. That's good. Making family history chart for a particular trait to check probability at its inheritance in coming generations.
बताओ 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 बताओ।
considering a tracing out the trait through in many generations of this same family in the form of a chart is known as pedigree analysis. So that's what I'm gonna say in my own words. So this is phenylketonuria, sickle cell anemia. Then
सकता है डोमिनेंट सॉल्व करूंगा यस अगर मेरा आंसर मिल गया तो मिल गया वरना मैं फिर जाकर के एक्स लिंक
okay. Now here there is one very important line I remember, and we have to yeah, this is the one, pedigree cheat codes. So basically I have to write this down so that I can remember how to solve the pedigree nicely, because this will help me a lot for the pedigree. Even though I have solved some questions, but I feel like it's not enough. So and also we'll deal with this. So first of all we'll just do that. Okay, man, my pen is not working. One minute, I'll check if I have another pen. Okay. Plus my pouch is missing, bro. This is messed up. Never mind, I just find it out. Like, bro, what? I've been finding this pouch for a whole week, and I wasn't able to find it out. The moment I just did it today, started my stream, and after for some amount of hours, when I just check the pouch, I just saw it somewhere here. Whoa, man, what? Bro, come on, bro, come on. Could have been easier for me to write. I mean, I had some pens and pencils which helped me practice, but then that was a bit tough time, I guess. So not I guess, bro. It should be. Not it should be. It must be. You know what? It's not must be. From my experience, yes, kind of. Okay. Oh my god. Oh, look at that, bro. 22 weaves.
घूरा अच्छे से तो आपको पता लग जाएगा कि वो डोमिनेंट है रिसेसिव है आइडिया लग जाएगा एटलीस्ट देखो भाई 4 ऑप्शन है हमारे पास ले देकर ऑटोसोमल
Okay. 22 weaves. There was one weaver watching me. I don't know when. But bro, if it is when I just left the chat, when I just left the stream, I'm gonna lose my mind, bro. That's crazy again. Anyways, let's move on. We'll do this.
कब होगी बीमारी
Do you know that 90% of people don't even suspect that their date of birth has already encrypted their past?
I don't believe in past lives. I'm sorry.
जब दोनों के दोनों रिसेसिव होंगे जब दोनों के दोनों रिसेसिव होंगे मेल में क्या है एक रिसेसिव हुआ खेल खत्म है ना यहां पर देखो
Okay, my bad. Yeah, let's start from here.
ये है घूरो पेडिग्री को पहले हमने पेडिग्री को घूर के देखा ताड़ा अगर हमें हर जनरेशन में कोई ना कोई बीमारी मिल रही इफ वी आर गेटिंग डिजीज individuals in एवरी जनरेशन, जितनी भी जनरेशन दी हुई है, अगर सब में कोई ना कोई बच्चा बीमार है, एक भी बच्चा बीमार मिल गया, if we are getting diseased individuals in every generation, there are high chances that the pedigree is dominating. भाई डोमिनेटिंग होने के चांसेस हमेशा रिसेसिव होने से ज्यादा होते हैं, कभी भी देख लेना भाई। जैसे ये आपने लिया कैपिटल टी स्मॉल टी, कैपिटल टी स्मॉल टी, दोनों बराबर ही लिए, कैपिटल टी कैपिटल टी, कैपिटल टी स्मॉल टी, कैपिटल टी स्मॉल टी, स्मॉल टी स्मॉल टी, एक रिसेसिव बना, 3 डोमिनेटिंग बने, क्योंकि इसमें तो एक कैपिटल टी आते ही काम हो गया ना, तो 3:1 आया था, इसीलिए हमारा मोनोहाइब्रिड क्रॉस में डोमिनेटिंग बच्चों की संख्या ज्यादा होती है। अगर कोई
If we are getting diseased individuals in every generation, okay,
ऐसी पेडिग्री है जिसमें बीमारी आपकी हर जनरेशन में जा रही है, देयर आर वेरी हाई चांसेस, मैंने क्या बोला है, जरूरी नहीं है, पर देयर आर वेरी हाई चांसेस कि वो पेडिग्री कैसी हो,
dominating.
अगर डोमिनेटिंग है ना, अगर आपने सोच लिया कि भाई हर जनरेशन में आपको कोई ना कोई बीमार मिला, अगर डोमिनेटिंग है तो पहले आप क्या करेंगे, ट्राई टू सॉल्व इट
Okay.
as autosomal dominant disease। ऑटोसोमल को सॉल्व करना आसान रहता है, ऑटोसोमल डोमिनेंट डिजीज। अगर समझ में नहीं मिला, ऑटोसोमल डोमिनेंट नहीं निकली, हमारा आंसर नहीं आया, देन ट्राई टू सॉल्व इट एज सेक्स लिंक्ड डोमिनेंट।
Okay.
Sex linked dominant।
You're mastering pedigree, guys, because it's not much given in detail. That's why.
Sex linked dominant, करेक्ट। यानी सबसे पहले आपने पेडिग्री को घूरा, पेडिग्री को देखा। अगर आपने पेडिग्री को देखा, घूरा अच्छे से, तो आपको पता लग जाएगा कि वो डोमिनेंट है, रिसेसिव है, आइडिया लग जाएगा एटलीस्ट। देखो भाई 4 ऑप्शन है हमारे पास ले देकर, ऑटोसोमल डोमिनेंट, ऑटोसोमल रिसेसिव, सेक्स लिंक्ड डोमिनेंट, सेक्स लिंक्ड रिसेसिव। करेक्ट है गुरुजी। अब इनमें से समझ लीजिए, मुझे पता लग गया कि बीमारी डोमिनेटिंग है, मैं पहले ऑटोसोमल डोमिनेटिंग सॉल्व करके देखूंगा, हो सकता है मुझे आंसर मिल जाए, मिला तो बहुत अच्छी बात है, वरना मैं एक्स लिंक्ड डोमिनेंट सॉल्व करके देखूंगा, मिल जाए आंसर तो बहुत अच्छी बात है। तो मतलब पहले वाला आंसर नहीं निकला, ये आंसर निकला। तो भाई चारों सॉल्व नहीं करनी पड़ी ना, 2 सॉल्व करके ही पता लग गया। फिर भी नहीं निकला तो भाई फिर आपका ऑटोसोमल रिसेसिव सॉल्व कर लो या एक्स लिंक्ड रिसेसिव सॉल्व कर लो। हमें तो देखा, घूरा अच्छे से, तो आपको पता लग जाएगा कि वो डोमिनेंट है, रिसेसिव है, आइडिया लग जाएगा एटलीस्ट। देखो भाई 4 ऑप्शन है हमारे पास ले देकर, ऑटोसोमल डोमिनेंट, ऑटोसोमल रिसेसिव, सेक्स लिंक्ड डोमिनेंट, सेक्स लिंक्ड रिसेसिव। करेक्ट है गुरुजी। अब इनमें से समझ लीजिए, मुझे पता लग गया कि बीमारी डोमिनेटिंग है, मैं पहले ऑटोसोमल डोमिनेटिंग सॉल्व करके देखूंगा, हो सकता है मुझे आंसर मिल जाए, मिला तो बहुत अच्छी बात है, वरना मैं एक्स लिंक्ड डोमिनेंट सॉल्व करके देखूंगा, मिल जाए आंसर तो बहुत अच्छी बात है। तो मतलब पहले वाला आंसर नहीं निकला, ये आंसर निकला। तो भाई चारों सॉल्व नहीं करनी पड़ी ना, 2 सॉल्व करके ही पता लग गया। फिर भी नहीं निकला तो भाई फिर आपका ऑटोसोमल रिसेसिव सॉल्व कर लो या एक्स लिंक्ड रिसेसिव सॉल्व कर लो। हमें सिर्फ वक्त बचाना है कि पहले मैं कौन सी सॉल्व करूं। सॉल्व तो करनी पड़ेगी पेडिग्री, कोई ट्रिक नहीं है, लेकिन पहले कौन सी सॉल्व करूं, ऑप्शन देख करके अपना समय तो बचाऊं। भाई समझ लो, आपके पास 4 ऑप्शन है, ऑटोसोमल डोमिनेंट, ऑटोसोमल रिसेसिव, एक्स लिंक्ड डोमिनेंट, एक्स लिंक्ड रिसेसिव। 4 आपके पास ऑप्शन है। गधे की तरह चारों थोड़ी सॉल्व करने बैठ जाऊंगा कि लाइन से लग जाता हूं, भाई ऑटोसोमल डोमिनेंट है क्या, ऑटोसोमल रिसेसिव है क्या, एक्स लिंक्ड डोमिनेंट है क्या। भाई तुक्का क्यों मार रहा हूं मैं, तुक्का क्यों मार रहा हूं, क्योंकि एक एक करके सॉल्व करनी पड़ेगी, ये ट्रुथ है। तो आपको अब एक आइडिया लगाना पड़ेगा कि मैं पहली कौन सी सॉल्व करूं जिसके होने के सबसे ज्यादा चांसेस हैं। तो अगर आपने कोई ऐसी पेडिग्री देखी जिसमें हर जनरेशन में बीमारी आ रही है, वो डोमिनेंट होगी, ज्यादातर चांसेस हैं कि वो डोमिनेंट होगी। अगर वो डोमिनेंट होगी तो पहले डोमिनेंट वाली दोनों सॉल्व करो ना, और ऑटोसोमल झटपट सॉल्व कर लोगे, ऑटोसोमल झटपट सॉल्व कर लोगे। तो इसीलिए पहले ऑटोसोमल को सॉल्व कर लो, उसमें टाइम नहीं लगता। और अगर ऑटोसोमल ना मिले तो एक्स लिंक्ड सॉल्व कर लो। तो आपने क्या किया, अपना वक्त बचा लिया। समझ लो इन दोनों में से भी किस्मत फूट गई भाई, इन दोनों में से भी कोई नहीं आया, इन दोनों में से भी कोई नहीं आया। तो अब क्या करोगे, इफ अ डिजीज स्किप्स जनरेशन, एक जनरेशन में बीमारी मिली, अगले में नहीं मिली, स्किप्स जनरेशन, एक जनरेशन में बीमारी नहीं मिली, तो क्या चांसेस है सर, ये रिसेसिव हो सकती है। देयरफॉर हाई चांसेस दैट द डिजीज इज रिसेसिव। अगर कोई पेडिग्री आपने घूरी और वहां आपकी जनरेशन स्किप हो गई तो हाई चांसेस हैं, बहुत हाई चांसेस हैं कि वो आपकी जो बीमारी है वो रिसेसिव हो जाएगी। करेक्ट है, एकदम करेक्ट है गुरुदेव, हो सकता है वो बीमारी आपकी रिसेसिव हो जाए। अगर वो बीमारी रिसेसिव है तो आप क्या करोगे? इफ दैट डिजीज इज रिसेसिव, चेक क्रिस क्रॉस पैटर्न। चेक क्रिस क्रॉस पैटर्न, या तो आपको सवाल में ही दिया जाएगा कि ये आपकी एक्स लिंक्ड बीमारी है, अगर नहीं तो क्रिस क्रॉस पैटर्न चेक कर लो। क्रिस क्रॉस पैटर्न एक स्पेशल पहचान है, बीमार मेल से बीमारी फीमेल में जाती है, बीमार फीमेल से बीमारी मेल में जाती है, मेल से फीमेल में, फीमेल से मेल में, एंड सो ऑन। इसे हम कहते हैं क्रिस क्रॉस पैटर्न। ये किसकी पहचान है? ये एक्स लिंक्ड रिसेसिव बीमारी की पहचान है। तो अगर इस प्रकार का पैटर्न दिख रहा है तो देखते ही एक्स लिंक्ड रिसेसिव मार दो, सॉल्व करने की भी जरूरत नहीं। एंड इफ क्रिस क्रॉस पैटर्न इज नॉट प्रेजेंट, if criss cross not seen, if criss cross not seen, then solve for ऑटोसोमल रिसेसिव। एक बार ऑटोसोमल रिसेसिव के लिए सॉल्व करके देख लो, ऑटोसोमल रिसेसिव के लिए सॉल्व करके देख लो। तो चारों सॉल्व करके देख लिया आपने, ऑटोसोमल डोमिनेंट के लिए, एक्स लिंक्ड डोमिनेंट के लिए, क्रिस क्रॉस पैटर्न एक्स लिंक्ड रिसेसिव के लिए, ऑटोसोमल रिसेसिव भी सॉल्व करके देख सकते हो। आखिरी जो आपका बचा वो क्या है? इफ बीमारी ओनली इन मेल, इफ डिजीज ओनली इन मेल, ये आपका मेल है, आए, ये आपका मेल है, ये आपकी फीमेल है, बच्चा हुआ ये मेल है, बच्चे का ब्याह हुआ फीमेल से, फिर बच्चा हुआ ये मेल है, ये सारे एक जैसे आपके मेल हैं, बीमार हो गए, बीमारी मेल मेल में फैल गई। मेल मेल में ये जो बीमारी फैली ये फीमेल में नहीं गई, सिर्फ मेल में गई, क्योंकि मेल में कुछ खास होता है जो फीमेल में नहीं होता, वो क्या है? आपका वाई क्रोमोसोम। तो ये बीमारी पक्का वाई लिंक्ड है। एक ऐसी बीमारी जो सिर्फ मेल में है फीमेल में नहीं, इसका मतलब है पक्का वाई लिंक्ड है। ठीक है, एकदम करेक्ट है। इन रूल्स को गौर से सुनो और फिर मैं आप
So that's why if disease only in males means that this is also sex linked. Okay. Okay.
को इन रूल्स को इंप्लीमेंट करना सिखाता हूं, गौर से सुनना, प्यार से सुनना, ठीक है? आराम से सुनना। मैंने एक पेडिग्री की शक्ल ताड़ी, मैंने उसको देखा घूर के, और मुझे हर जनरेशन में कोई ना कोई बीमार इंडिविजुअल मिला। अगर हर जनरेशन में एक भी बीमार इंडिविजुअल मिल रहा है तो बीमारी आपकी डोमिनेंट हो सकती है, हो सकती है। मैं सॉल्व करके वैलिडेट करूंगा, बीमारी डोमिनेंट हो सकती है। तो मैं सबसे पहले क्या करूंगा? ऑटोसोम को सॉल्व करना आसान रहता है, मैं ऑटोसोमल डोमिनेंट के लिए सॉल्व करूंगा। मुझे मेरा आंसर नहीं मिला तो मैं एक्स लिंक्ड डोमिनेंट के लिए सॉल्व करूंगा, ठीक है? फिर भी समझ लो मुझे मेरा आंसर नहीं मिला तो मैं आपका क्रिस क्रॉस पैटर्न देखूंगा। अगर क्रिस क्रॉस पैटर्न भी नहीं है तब जाके मैं ऑटोसोमल रिसेसिव के लिए सॉल्व करूंगा। ये मेरा सीक्वेंस होना चाहिए पेडिग्री सॉल्व करने का। समझ लो मुझे बीमारी नहीं मिली एक जनरेशन में तो इसका मतलब वो पक्का रिसेसिव है, तो पहले मैं रिसेसिव वाली सॉल्व करूंगा फिर डोमिनेंट वाली सॉल्व करूंगा, यानी मैं अपना 50% टाइम बचा लूंगा। मैं चारों केस नहीं सॉल्व करूंगा, मैं एक सीक्वेंस में नहीं चलूंगा, मैं दिमाग लगाऊंगा। अगर मैं पेडिग्री को देख रहा हूं हर जनरेशन में कोई ना कोई बीमारी है, पहले मैं ऑटोसोमल वाले केसेस सॉल्व करूंगा। अगर मैं एक पेडिग्री को देख रहा हूं और मुझे कोई बीमारी किसी ऐसी जनरेशन में, ऐसी भी जनरेशन है कोई जिसमें बीमारी नहीं है, स्किप कर गई जनरेशन, तो पहले रिसेसिव वाली सॉल्व करूंगा, ठीक है? यहां तक की बात समझ में आई? अब ये देखो रूल्स इंप्लीमेंटेशन के है ना, ये रूल्स इंप्लीमेंटेशन के अब करेंगे, प्रैक्टिस करेंगे तो समझ में आ जाएगा, प्रैक्टिस करेंगे तो समझ में आ जाएगा। दोबारा सुनो यार, समझ लो। अच्छा ठीक है, मैं आपको एक क्वेश्चन देता हूं, आराम से उसको देख करके बताना। अभी आपको समझ में हो सकता है ना आया हो, कोई बात नहीं, 10 बार समझाऊंगा। जैसे मैंने बोला है, ये टॉपिक छोड़ेंगे नहीं आज। आज इस टॉपिक का खात्मा करके निकलना है भाई यहां से। रोटी पानी खा लेंगे आधे घंटे बाद सब, है ना, हम भी यहीं पे खड़े हैं। आराम से देखना मेरे भाई, मेरी बात को सुनो। मैं कह रहा हूं ये एक पेडिग्री है, इसकी शक्ल देख लो। ये पहली जनरेशन है, क्या इसमें मुझे बच्चा मिला डिजीज? बिल्कुल। इनके जो बच्चे हुए सारे के सारे, इनमें क्या मुझे डिजीज बच्चे मिले? बिल्कुल गुरुजी मिले। और इनके जो सारे बच्चे हैं, ये भी इसी जनरेशन के हैं, ये भी इसी जनरेशन के हैं, इनमें भी क्या मुझे डिजीज बच्चे मिले? मिले। क्या मैं इस पेडिग्री को पहले डोमिनेंट बीमारी के लिए सॉल्व करूं या रिसेसिव के लिए? चैट सेक्शन भरो एक बार फटाफट। क्या मैं इस बीमारी को डोमिनेंट के लिए सॉल्व करूं पहले या रिसेसिव के लिए? भाई पहली जनरेशन में भी एक बच्चा बीमार मिला, दूसरी जनरेशन में 2 बच्चे बीमार मिले, तीसरी जनरेशन में 1 बच्चा बीमार मिला, यानी हर जनरेशन में बीमार बच्चा है, यानी ये बीमारी डोमिनेंट होने के चांसेस हैं। यस। तो क्या मैं पहले ऑटोसोमल डोमिनेंट सॉल्व करूंगा? यस। अगर मेरा आंसर मिल गया तो मिल गया, वरना मैं फिर जाकर के एक्स लिंक डोमिनेंट सॉल्व करूंगा। फिर भी आंसर नहीं मिला, तब जाके मैं रिसेसिव पे जाऊंगा। सबसे पहले मैं किस पे कूदूंगा? डोमिनेंट पे कूदूंगा। क्या ये बात समझ में आई? बिल्कुल आई गुरुजी, इसमें कोई टेंशन नहीं है, देख के पता लग गया। समझ लो मेरे पास दूसरी पेडिग्री है। पहली ही जनरेशन में बीमारी स्किप हो गई। पहली ही जनरेशन में माता पिता जो है, त्वमेव माता त्वमेव पिता, ये दोनों ही नॉर्मल है, दोनों में बीमारी नहीं है। अगली जनरेशन में बीमारी है, अगली जनरेशन में बीमारी है, लेकिन पहली ही जनरेशन में बीमारी नहीं है, जनरेशन स्किप हो गई। इसको अगर मैं सॉल्व करूंगा तो क्या मैं पहले रिसेसिव के लिए सॉल्व करूंगा? है ना? इसको अगर मैं सॉल्व करूंगा, अगर एक जनरेशन स्किप हो गई, अगर हर जनरेशन में बीमारी नहीं है, तो क्या ये बीमारी रिसेसिव होने के चांसेस ज्यादा हैं? तो क्या मैं अपना दिमाग लगा के डोमिनेंट पे अपनी दिमाग की खपत नहीं करूंगा? बिल्कुल भाई, मेरे पास लिमिटेड दिमाग है, मेरे पास पेपर में लिमिटेड टाइम होगा, मैं डोमिनेंट सॉल्व नहीं करूंगा, पहले मैं दोनों रिसेसिव केस सॉल्व करूंगा, तब जाके मुझे मेरा आंसर मिलेगा। करेक्ट, करेक्ट है। और देखो, और देखो भाई, यहां देखो भाई, ये वाला केस देखो। इस जनरेशन में बीमारी है, इस जनरेशन में बीमारी है, इसमें बीमारी नहीं है। अगर मैं इसको सॉल्व करने पे आया तो मैं क्या इसे पहले रिसेसिव के लिए सॉल्व करूंगा?
यस, रिसेसिव।
अगर मैं इसे सॉल्व करने पे आया, क्या मैं पहले रिसेसिव के लिए सॉल्व करूंगा या डोमिनेंट के लिए? पहले मैं अपना दिमाग किस पे खर्च करूंगा? रिसेसिव वाले दोनों ऑप्शन पे सर।
Listen, listen. Okay, this is interesting. So, this is skipped. So recessive, either autosomal or recessive. So I'm gonna do with AA. AA. Am I gonna get this? No. Let's check. No, I don't think so. I don't get it. So no. Let's start with X. XCY cross. No. Yeah. Okay. XCXC. Let's see. We'll not mess that up. Now it says that it's a recessive trait. So technically, if it is X-linked recessive, what does that mean? Well, XCY. That means if I dumb it, XCY, I separated them. All right. You can also check that one out. Tomorrow the person who's gonna sit next to him is cooked. Okay, I don't know who's it. A capitalized small a. Hey. So this is most probably. Okay, I have to check excellent recessive.
एक्स लिंक रिसेसिव और ऑटोसोमल रिसेसिव पे अपना दिमाग खर्च करूंगा मैं। करेक्ट, करेक्ट। आगे देखो, ये जनरेशन है, इसमें बीमारी है, इसमें नहीं है, इसमें
recessive.
नहीं है, इसमें है, 2 जनरेशन स्किप हो गई। क्या मैं अपना दिमाग रिसेसिव वाले केस पे लगाऊंगा? क्या मैं अपना दिमाग रिसेसिव में खर्च करूंगा? अगर मैं इसे सॉल्व कर
Does this go crisscross? No. No.
Does not. No, does not actually. So.
पे आया, क्या मैं पहले रिसेसिव के लिए सॉल्व करूंगा या डोमिनेंट के लिए? पहले मैं अपना दिमाग किस पे खर्च करूंगा? रिसेसिव वाले दोनों ऑप्शन पे सर। एक्स लिंक रिसेसिव और ऑटोसोमल रिसेसिव पे अपना दिमाग खर्च करूंगा मैं। करेक्ट। आगे देखो, ये जनरेशन है, इसमें बीमारी है, इसमें नहीं।
Recessive, but is it? This looks like autosomal. I don't know how this could be carrier, but no.
नहीं है, इसमें नहीं है, इसमें है। दो जनरेशन स्किप हो गई। क्या मैं अपना दिमाग रिसेसिव वाले केस पे लगाऊंगा? क्या मैं अपना दिमाग रिसेसिव वाले केस पे लगाऊंगा? हाँ के ना? यस, रिसेसिव वाले केस पे लगाऊंगा। नॉट तो करनी ही पड़ेगी, वो अभी सिखाऊंगा कैसे सॉल्व करना है, पर सॉल्व करें कैसे, पहले किसको सॉल्व करें, टाइम भी तो बचाना है पेपर में, वो हम लोगों ने देख लिया। आई होप इतनी बातें सबको समझ में आएंगी। अब सॉल्व करना सीखते हैं, समझ लो हमारे पास पहला केस है, ये एनसीईआरटी का केस है। समझ लो मैं इसे सॉल्व करता हूं, समझ लो मैं इसे सॉल्व करने लग पड़ा। ठीक है, पहली जनरेशन में बीमारी है, दूसरी जनरेशन में बीमारी है, तीसरी जनरेशन में भी बीमारी है, यानी नो जनरेशन स्किप्ड, नो जनरेशन स्किप्ड। Therefore, चांसेस तो डोमिनेंट केस में बीमारी केसेस आप कह रहे हो तो कैसे ना माने, ये तो आपका सेफ होगा ही होगा, ये आपका सेफ होगा। ने भी क्या इनकी सेफ ए छोटा ए डिजीज में दो लगा देंगे, छोटा ए पेडिग्री समझ, छोटा ए होगा कैपिटल ए हो जाएगा, कैपिटल ए छोटा ए होगा, ये भी आपका कैपिटल ए होगा या कैपिटल ए छोटा ए होगा, तभी ये बीमार हो सकता है। अब देखो नीचे स्मॉल ए आ रहा है, स्मॉल ए आ रहा है, यानी दोनों पेरेंट से एक एक स्मॉल ए आएगा, तभी तो यहां स्मॉल ए बनेगा, है ना भाई। अगर यहां स्मॉल ए है तो दोनों पेरेंट ने एक एक स्मॉल ए दिया होगा ना, तभी तो ये कॉम्बिनेशन बनी। तो मेरे भाई यहां पर देखिए कैपिटल ए,
how is this possible? This should be capital A, small a, right? Because capital A and small a will give. This will affect everything. So it's capital A, small a.
स्मॉल ए आना चाहिए ना, यहाँ भी स्मॉल ए है, यहाँ भी स्मॉल ए है, तभी तो बच्चे स्मॉल ए बनेंगे। यस, स्मॉल ए बनेंगे। तो क्या कैपिटल ए होने की कोई संभावना है? कोई संभावना नहीं है गुरुजी। यहाँ कैपिटल ए है, यहाँ कैपिटल ए है ही नहीं, तो ये भी जो बच्चा बनेगा कैपिटल ए स्मॉल ए बनेगा, ये भी जो बच्चा बनेगा ये कैपिटल ए स्मॉल ए बनेगा, क्योंकि कैपिटल ए होने की कोई संभावना है ही नहीं। यहाँ पे कैपिटल ए अगर होता तो सारे बच्चे कैपिटल ए स्मॉल ए बनते, स्मॉल ए नहीं बन पाते। तो ये वाला नहीं है पेरेंट, ये वाला पेरेंट है। ठीक है, करेक्ट। यहां पर देख ले स्मॉल ए भी, तो हमारा उत्तर पहली बार में निकल गया, यानी मैंने पेडिग्री को इतना देखा, स्मॉल ए हो गया। गुरुजी ये तो हमारा उत्तर ही निकल गया, सारी पेडिग्री सेट बैठ गई, यानी हमारा उत्तर पहली बार में निकल गया, यानी मैंने पेडिग्री को इतना देखा, जितना देखा जा सकता था, दो आंखों से जितना देखा जा सकता था उतना देखा, पता लग गया डोमिनेटिंग है। तो पहले ऑटोसोम के लिए सॉल्व कर लिया, क्योंकि आसान पड़ती है, तो इसलिए मैंने सॉल्व कर लिया, उत्तर ही आ गया। तो बचे हुए 3 सॉल्व भी नहीं करने पड़े, सॉल्व मैंने अभी भी किया है, कोई तरकीब नहीं लगाई है, सॉल्व मैंने अभी भी किया है, आपके सामने ही सॉल्व किया। लेकिन पहले कौन सा सॉल्व करना है ये तो मुझे समझ में आया ना, बाकी 3 तो देखनी भी नहीं पड़ी, दुनियादारी को तो मैंने देखा भी नहीं, मैंने जो सॉल्व किया उसी में मेरा उत्तर लड़ गया। दिस इज इंपॉर्टेंट। अब कई धुरंधर कह रहे हैं नहीं समझ में आया, फिर समझा दूंगा भाई, कोई दिक्कत नहीं है, आराम से देखिए। मतलब मेरा उत्तर आ गया, दिस इज ऑटो जनरेशन। ये तीसरे जनरेशन में कोई ना कोई बीमार है, तो मेरे भाई क्या ये डोमिनेटिंग बीमारी होनी चाहिए? यस गुरुजी, डोमिनेटिंग बीमारी होनी चाहिए, इतना मुझे पता लग गया। वैसा है तो ऑटोसोमल कर रहा हूं, होगी स्मॉल ए, आप लिख सकते हो स्मॉल ए बना दो, सारे स्मॉल ए।
What?
जब भी कोई सेफ बच्चा देखूं मेरा दिल दीवाना बोले, स्मॉल ए।
I didn't get it.
स्मॉल ए बना देंगे, जितने स्मॉल ए बना सकते हो उतने स्मॉल ए बना दो, सारे सेफ है, स्मॉल ए कोई खेल बचा ही नहीं है, सब सेफ है, ठीक है, आना चाहिए स्मॉल ए पहले से। और मम्मी डिजीज भी है, यानी मम्मी इन दोनों में से कोई एक होगी, तो स्मॉल ए, ये जो स्मॉल ए है। दोबारा सॉल्व करेंगे, इसको गलत साबित करेंगे, है ना। हिट किया था इसीलिए मैं इसलिए खाली 2 लेके आया था, मुझे पता है ना कितने बुद्धिमान हो तुम, इसलिए पहले ही 2 चिपका के लाया था। एक बार में समझ में न आनी तुम्हारे, इसीलिए कई बुद्धिमान लोगों को समझ में आ गया था, इसीलिए उन्होंने कहा कि सर सेम क्वेश्चन है, उन्हें लगा कि सर वैरायटी ऑफ क्वेश्चंस कराएंगे। पहले मैं तुम्हें संतुष्ट तो कर दूं, एक सवाल को 3 बार बता के, तभी तो आगे बढ़ोगे, है ना। मैंने ही कराया है, मुझे ही चमका रहे हैं सर सेम ही क्वेश्चन है, जैसे मुझे तो पता ही नहीं लगा होगा कि एक ही स्लाइड पे।
Yeah, I know why he did this. He just wanted to like show it in repetition, and also he wanted to like wanted to also explain the sex-linked dominant in this one, just to prove it wrong. How is it not sex-linked dominant? How is it autosome? So he used this one because so people can get reference from here also. That's one thing.
2 बातें लिखी हुई हैं, एक्स लिंक्ड डोमिनेंट के लिए सॉल्व करता, मैं इसको एक्स लिंक्ड डोमिनेंट के लिए सॉल्व करता। एक्स लिंक्ड डोमिनेंट में
1 minute. I have a feeling like taking this all out. So let me just take this out. Bam, 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 and bam. There's this last thing.
आप केस देख लीजिए, एक्स लिंक्ड डोमिनेंट में केस देख लीजिए, फीमेल जो है अगर आए, ठीक है, मेल की अगर आप बात करें, मेल की अगर आप बात करें तो मेल में तो भाई एक ही एक्स क्रोमोसोम है, अगर ये डोमिनेटिंग तो बीमारी भी आपकी हो जाएगी, डिजीज हो जाएगी। और मेरे भाई आपका छोटा सी अगर रिसेसिव तो मेल आपका सेफ हो जाएगा, मेल आपका सेफ हो जाएगा, करेक्ट। सिंपल सी बात देखो।
Oh yeah, that was confusing. That was really confusing when he used that. Oh sure. To be honest, I still remember like studying from this lecture, and this was so confusing when he used small letter C and capital C. They looked a lot like. I think he had to use a bit different one. But again, the reason why we are using this small c is because to show that recessive trait. So I get. But that small c is so small that it looks like the capital C and small c are being together. So yeah. We have 4 more breadsticks.
सिंपल सी बात है, जो मेल सेफ है इसमें तो एक ही केस है, जो मेल डिजीज है इसमें भी एक ही केस है। मेल तो हमें सारे पता है, आओ मेल जोल बढ़ा लें, आओ मेल जोल बढ़ाएं। देखो जितने मेल डिजीज है, जितने मेल डिजीज है इनमें क्या आएगा, एक्स कैपिटल सी वाई, और जितने मेल सेफ है उनमें आएगा एक्स छोटा सी वाई, 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 ठीक है, इतने ही आए, ठीक है। फीमेल में देखिए,
No, please, I want to check. Come on. Huh? Wait, what about the carrier? Ow, bro. My leg is hurting just by sitting on carpet. That happens a lot. What does this C represent? What? Okay, I think this is heterozygous. Both are homozygous. But I'm confused with this case. Like, what about carrier? What about the carrier thing? What will the carrier do in this case? But only in the night. I see. Let me dip it in this chocolate.
Oh yeah, that's now what we call a lot of chocolate.
जो मेल डिजीज है इसमें भी एक ही केस है। मेल तो हमें सारे पता है। आओ मेल जोल बढ़ा ले, आओ मेल जोल बढ़ाएं। देखो जितने मेल डिजीज है, जितने मेल डिजीज है इनमें क्या आएगा, एक्स कैपिटल सी वाई।
This is important.
और जितने मेल सेफ है उनमें आएगा एक्स छोटा सी वाई, 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 एक्स छोटा सी वाई। ठीक है, इतने ही आए। ठीक है, फीमेल में देखिए, फीमेल में अगर मैं देखूं तो एक ही सेफ केस है। डिजीज में मुझे तब भी चुनना पड़ेगा, सेफ केस एक ही है आपका, एक्स छोटा सी, एक्स छोटा सी।
No, I'm confused. How? Then what about the carrier?
तो फीमेल जहां सेफ है यहां एक्स छोटा सी, 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 एक्स छोटा सी। करेक्ट, एकदम करेक्ट है गुरुजी। अब मेरी बात को जरा गौर से सुनना है, अभी मेरी बात को बहुत गौर से सुनना है। डिजीज्ड फीमेल जो है, जो आपकी फीमेल ऐसी हैं जिनमें बीमारी है, दो तरीके की हो सकती है, इनमें से हमें एक चुनना पड़ेगा। ठीक है, यहां बच्चे कैसे हैं, इनमें छोटा सी दोनों में आ रहा है, छोटा सी दोनों में आ रहा है, तो पेरेंट में भी दोनों में छोटा सी होना चाहिए। तो ये फीमेल डिजीज है, तो इसमें एक ही केस ऐसा है जिसमें छोटा सी आए, यानी एक्स कैपिटल सी, एक्स छोटा सी। तो यहां पे आ गई एक्स कैपिटल सी, एक्स छोटा सी। एक्स कैपिटल सी, एक्स छोटा सी जो है ये आपकी फीमेल जो है आपकी कैसी हो गई, यहां पर फीमेल अगर बनेगी तो एक्स कैपिटल सी, एक्स छोटा सी ये बनेगी, या एक्स छोटा सी, एक्स छोटा सी बनेगी। एक्स छोटा सी, एक्स छोटा सी सेफ है, एक्स कैपिटल सी, एक्स छोटा सी डिजीज हो जाएगी। तो एक्स कैपिटल सी, एक्स छोटा सी ये आपकी बीमारी वाली फीमेल बनेगी। करेक्ट, क्योंकि एक्स कैपिटल सी का बनने का कोई चांस नहीं है, क्योंकि यहां कैपिटल सी है, यहां कैपिटल सी है ही नहीं, तो बनेगा नहीं। तो एक्स कैपिटल सी, एक्स छोटा सी बनेगा। अब यहां देखो, यहां बच्चे बने कैपिटल सी, एक्स छोटा सी, यानी ये आपकी डिजीज हो गई एक्स कैपिटल सी, एक्स छोटा सी। फिर आपकी बनी आपकी एक्स छोटा सी, एक्स छोटा सी, यानी अगर एक और फीमेल होती तो सेफ हो जाती। ठीक है, ठीक है गुरुजी, ये बात हमें समझ में आ गई है ना। एक्स कैपिटल सी, एक एक्स छोटा सी यहां पर बना। अब देखो मेरे भाई, मेल देखो, एक्स कैपिटल सी वाई, एक्स छोटा सी वाई बना है यहां पर, एक्स छोटा सी वाई, एक्स छोटा सी वाई बना है।
पढ़ लिया पूरा, खाली रिवाइज करो।
अगर दो मेल बनते, एक्स कैपिटल सी वाई, एक्स छोटा सी वाई बनते, एक्स छोटा सी वाई, एक्स छोटा सी वाई बने। यहां आकर के पेडिग्री हल्की सी हमें कंफ्यूजन दे रही है, इस पे हम उतने कॉन्फिडेंट नहीं है जितना हम पहली वाली पे थे। तो इन दोनों में से एक चुनना पड़ा, तो ये चुन गुरुजी, ये बात हमें समझ में आ गई है ना। एक्स कैपिटल सी, एक एक्स छोटा सी यहां पर बना। अब देखो मेरे भाई, मेल देखो, एक्स कैपिटल सी वाई, एक्स छोटा सी वाई बना है यहां पर, एक्स छोटा सी वाई, एक्स छोटा सी वाई बना है यहां पर। अगर दो मेल बनते, एक्स कैपिटल सी वाई, एक्स छोटा सी वाई बनते, एक्स छोटा सी वाई, एक्स छोटा सी वाई बने। यहां आकर के पेडिग्री हल्की सी हमें कंफ्यूजन दे रही है, इस पे हम उतने कॉन्फिडेंट नहीं है जितना हम पहली वाली पे थे। तो इन दोनों में से एक चुनना पड़ा, तो ये चुनेंगे, ये चुनेंगे। करेक्ट, करेक्ट। यहां तब भी कंफ्यूजन आ गई कि भैया आपका मेल जो बनता वो आपका कैसा बनता, एक्स छोटा सी वाई और एक्स छोटा सी वाई दोनों एक जैसे नहीं बनते, एक बनता एक्स कैपिटल सी वाई, एक बनता एक्स छोटा सी वाई। यहां पे दोनों ही आपके एक जैसे बने हैं, तो कहीं ना कहीं कंफ्यूजन है। इसमें एकदम सेट है, देखो कैपिटल ए स्मॉल ए बन सकता है, कैपिटल ए स्मॉल ए बना, स्मॉल ए बन सकता है, स्मॉल ए बन सकता है, यहां एकदम फिट बैठी है कड़ी। यहां थोड़ी कंफ्यूजन है, दोनों में से एक भी चुनना पड़ा तो ऑटोसोमल चुनेंगे। यस, ये आपने देखा करेक्ट, तो ये ज्यादा करेक्ट है, इसको हम करेक्ट मानेंगे। यहां कंफ्यूजन है एंड में, तो यहां दिक्कत है पेडिग्री में, यहां हमें लग रहा है कि दिस इज नॉट द राइट आंसर, इसको हम लोग खारिज कर दें। करेक्ट है, ऑलराइट। आ जाओ, अगला जो है मेरे साथ साथ सॉल्व करना मेरे भाई, देखना। तो ऑटोसोमल डोमिनेंट निकल गई हमारी ये पेडिग्री, ऑटोसोमल डोमिनेंट निकल गई, एक्स लिंक डोमिनेंट नहीं है, यहां पे आके जो है गड़बड़ा गई। ठीक है, अब मेरे भाई आराम से देखो अगली पेडिग्री, गौर से देखो एक बार। इस पेडिग्री में डिजीज स्किप हो गई इस जनरेशन में, तो क्या ये रिसेसिव होनी चाहिए?
So let's do this. We'll do this fully. This is A, capital A, small a, I believe, which are going under this, as we know that capital A, small a will give us capital A, capital A. See, the thing about this is recessive, so there's a small chance that this will be forming overall as capital A, small a here and capital A, small a. So from here we get a capital A, small a, and what else? Small a, small a. You can get small a, small a. So small a, small a, small a, small a. That's one thing. And then capital A, small a. Okay. Solve for autosomal recessive. So this is small a, small a, small a, small a. This is capital A, small a, or capital A, capital A. This should be capital A, small a. This should be capital A, small a. No, it cannot be capital A, because this one will not give this. So capital A, small a, and that will be small a, small a. So capital A, small a, capital A, small a, small a, small a, small a, small a, capital A, or capital A, small a, or capital A, small a, or capital A, capital A. Here it should be capital A, small a, capital A, small a. Then this will be capital A, small a, capital A, capital A, or capital small a, capital A, small a, capital A, capital A. So in this case, I think it's possible. After 2 minutes, oh, he started from there. Okay. But what if it is capital A, capital A?
बताओ, यहाँ नीचे बच्चे में स्मॉल ए है, यानी दोनों पेरेंट ने एक एक स्मॉल ए दिया है,
It's capital A, small a. Yes.
दोनों पेरेंट ने एक एक स्मॉल ए दिया है, तो दोनों में एक एक स्मॉल ए होना चाहिए. यस, कैपिटल ए इसीलिए नहीं होगा.
What about this? Why not these two? Because capital A is possible, right? No? Nah. Capital A, small a, capital A, small a. However, I know this is autosomal recessive, that's why I got it correct, but still, the genotype would have been wrong here. Okay. Like, you know what I mean?
करेक्ट, करेक्ट. दो में से उत्तर निकल गया, डोमिनेंट के लिए सॉल्व करने की जरूरत ही नहीं.
Let me just check this.
भाई इसमें भी यहाँ भी देखिए, यहाँ स्मॉल ए आ रहा है, तो दोनों पेरेंट में स्मॉल ए होना चाहिए. इसमें भी स्मॉल ए है, तो इसमें भी स्मॉल ए होगा. तो कैपिटल ए, स्मॉल ए यहाँ पर भी आ सकता है, कैपिटल ए, स्मॉल ए यहाँ पर भी आ सकता है, स्मॉल ए, स्मॉल ए है, तो दोनों पेरेंट एक ना एक स्मॉल ए लेकर के बैठे होंगे, यानी दोनों पेरेंट यही होंगे, कैपिटल ए, स्मॉल ए, कैपिटल ए, स्मॉल ए. तो यहाँ पर आपके बच्चे क्या बने, यहाँ पर आपका कैपिटल ए, स्मॉल ए यही वाला आपका सेफ बनेगा, कैपिटल ए, स्मॉल ए यही वाला आपका सेफ बनेगा, कैपिटल ए, स्मॉल ए यही वाला आपका सेफ बनेगा. ये है आपका स्मॉल ए, स्मॉल ए, ये है आपका कैपिटल ए, स्मॉल ए. इस वाले को अभी छोड़ देते हैं, इस वाले को वैलिडेट करेंगे. ठीक है. यहां भी देखिए, यहां स्मॉल ए आ रहा है, तो दोनों पेरेंट में स्मॉल ए होना चाहिए. इसमें भी स्मॉल ए है, तो इसमें भी स्मॉल ए होगा. तो कैपिटल ए, स्मॉल ए यहां पर भी आ सकता है, कैपिटल ए, स्मॉल ए यहां पर भी आ सकता है, स्मॉल ए यहां पर भी आ सकता है. पूरी पेडिग्री सॉल्व कर ली, कहीं पर भी कोई गड़बड़ नहीं मिली. जय हो मेरे भाई, हमको हमारा उत्तर मिल चुका है. ये पेडिग्री है ऑटोसोमल रिसेसिव.
But why not capital A, capital
बात
A?
समझ में आई? यस गुरुजी, समझ में आ गई. ऑटोसोमल रिसेसिव ए कैसे हो सकता है, कैपिटल ए कैसे हो जाएगा मेरे भाई? सोचो यहाँ पे अगर मैं कैपिटल ए लिख दूं, यहाँ पे कैपिटल ए लिख दूं, तो बच्चा स्मॉल ए कैसे बनेगा? इसके पास तो स्मॉल ए है ही नहीं, बच्चा स्मॉल ए कैसे बन पाएगा? कैपिटल ए
I'm
हो
talking
ही नहीं
about
सकता।
this one. I'm talking about this one. Sorry, not this one. This one and this one, and this one.
कैपिटल ए कैसे होगा बताओ? यहां नीचे बच्चे में स्मॉल ए है, यानी दोनों पेरेंट ने एक एक स्मॉल ए दिया है, दोनों पेरेंट ने एक एक स्मॉल ए दिया है, तो दोनों में एक एक स्मॉल ए होना चाहिए। यस, कैपिटल ए इसीलिए नहीं होगा। करेक्ट, दो में से उत्तर निकल गया, डोमिनेंट के लिए सॉल्व करने की जरूरत ही नहीं।
कब है इसमें तुम्हारा ये बच्चा का एग्जाम है? स्कूल, तुम्हारे स्कूल में? हाँ, आवाज नहीं आ रही। संडे है, नेक्स्ट संडे।
अब आप कहोगे कि गुरुजी एक्स लिंक्ड रिसेसिव नहीं है, समझ लो क्रिस क्रॉस इनहेरिटेंस ऐसी कोई चीज होती नहीं है। सर एक बार एक्स लिंक्ड रिसेसिव भी सॉल्व करके दिखा दो। भाई मान लो ये एक्स लिंक्ड रिसेसिव है, मान लो ये बीमारी एक्स लिंक्ड रिसेसिव है, एक्स लिंक्ड रिसेसिव है, एक्स लिंक्ड रिसेसिव है, तो क्या क्या हो जाएगा यहाँ पर? फीमेल को देखो, फीमेल को देखो कैसी हो जाएगी। एक्स लिंक्ड रिसेसिव कैपिटल सी है, यानी दोनों डोमिनेंट है।
I'm actually more confused here because I don't know why.
तो बीमारी नहीं होगी यहाँ पर रिसेसिव केस में, सेफ हो जाएगी। कैपिटल सी स्मॉल सी है तो भी डोमिनेटिंग है, सेफ हो जाएगी। स्मॉल सी है तो ये क्या हो जाएगी? डिजीज हो जाएगी। ठीक है, यहां पर देखो मेल के बारे में। मेल कैसा है भैया? मेल है आपका कैपिटल सी वाई, मेल डोमिनेटिंग है, बीमारी है रिसेसिव, तो सेफ है। मेल अगर आपका रिसेसिव है एक्स क्रोमोसोम पे, तो ये क्या है? डिजीज है। यानी मेल में अगर आप देखो तो एक ही केस है आपके डिजीज का, एक्स छोटा सी वाई, तो यहां हो गया एक्स छोटा सी वाई, एक्स छोटा सी वाई, है ना। और यहां पर आपका फीमेल में भी डिजीज का एक ही केस है, एक्स छोटा सी एक्स छोटा सी एक्स छोटा सी एक्स छोटा सी। ठीक है, मेल में सेफ का एक ही केस है, एक्स बड़ा सी वाई, 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 एक्स बड़ा सी वाई। ठीक है, ये जितने हम कर सकते थे उतना हमने कर दिया। अब मेरे भाई बहुत गौर से सुनना, ओ मेरे मुन्ना गौर से सुनना। यहां आपका जो बच्चा बन रहा है अलग अलग प्रकार का बन रहा है ना। यहां पे बच्चा बन रहा है अलग अलग प्रकार का, जैसे यहां पे एक्स छोटा सी बन रहा है। मेल जो है इस पे तो छोटा सी है ही नहीं, फीमेल के पास समझ लो छोटा सी होगा अनडाउटेडली, क्योंकि यहां भी छोटा सी आ रहा है, तो फीमेल के पास आपका एक्स छोटा सी होगा, क्योंकि ये सेफ है और छोटा सी भी है, तो एक ही केस बनता है, एक्स कैपिटल सी एक्स छोटा सी, एक्स कैपिटल सी एक्स छोटा सी, एक ही केस बनता है। ठीक है, होगा, मना नहीं करें। अगर भैया एक्स बड़ा सी और छोटा सी है इसके पास, यहां सिर्फ बड़ा सी है, तो छोटा सी जैसी तो कोई कॉम्बिनेशन है ही नहीं, ये बन ही नहीं पाएगा, यानी ये गलत पेडिग्री है, इसीलिए एक्स लिंक्ड रिसेसिव नहीं थी। यही पहले स्टेप पे, पहली ही जनरेशन में हमें गलत मिल गया।
Yeah, that's true.
तो हमने ये भी प्रूव किया कि ऑटोसोमल रिसेसिव क्यों है, हमने ये भी प्रूव किया एक्स लिंक्ड रिसेसिव क्यों नहीं है। यस।
Okay, so I don't know, man. Sometimes pedigree kind of causes controversy, but for me myself, like this teacher is the ours are like, I mean, not this teacher, he's my teacher too. So capital A is not possible for this one, and this one, it makes me kind of confused because we know that capital A can also bind, so I mean, like, you know, can also get crossed, so that's also possible, right? For this one and this one, I understand that, yeah, there should be capital A small a, so yes, that's true. This one is also capital A small a because that's how we can only get this capital A small a, small a. But I'm so confused about this one, bro. Sorry, this one and this one. Like, I don't know why. This one also has a chance of being capital A, right? Because capital A small a, I don't know why.
ये हमने देखा।
That's confusing.
करेक्ट यार, इसमें कैपिटल ए लिख लो यार, एक ही बात है ना। इससे हमें मतलब नहीं था, इसीलिए हमने ऐसे किया था, है ना। कुछ भी लिख लो इसमें, कोई दिक्कत नहीं, यहाँ लिख लो कैपिटल ए, एक ही बात। ऊपर कैपिटल ए नहीं आएगा, यहां लिख सकते हो, कोई दिक्कत नहीं, पर इससे हमें मतलब नहीं है ना, ये पेडिग्री सॉल्व करने में हमारी हेल्प नहीं कर रहा है, दैट्स व्हाई हम लोगों ने लिखा नहीं।
Okay, never mind.
क्रिस्टल क्लियर है मेरे भाई? क्रिस्टल क्लियर है?
Okay. That was
भाई
making
देखो,
me
जिसको
so
जिससे हमें मतलब
confused.
नहीं है वो कुछ भी लिख दो ना यार, इसको छोड़ दो, है ना, हमें तो इससे आगे की पेडिग्री मिल रही है ना, सॉल्व करी हमने, हमें ओवरऑल आइडिया है कि क्या करना है, राइट? तो यहां पर मैंने क्या किया, पहले केस में मैंने देखा कि पेडिग्री देखने में मुझे डोमिनेटिंग लग रही है, मैंने आपका ऑटोसोमल डोमिनेंट को सॉल्व किया, मुझे आंसर मिल गया, नहीं मिलता
Let's do some
तो
questions.
इंतजार है, वो सवाल मैं ले आया हूं, ये वाले।
Guess what? This is all from the book. If you don't know, fun fact, I'm just gonna tell you. This book right here that you see, right, this book right here has those questions which are there in this lecture. So basically, I'm doing those questions in here which are already there in this book. So that's one fun fact if you don't know. Like, yeah, you can see this. It's pretty thick and big, bro. Like, very big. Where's my cursor? Yes, here we go.
नहीं, ये वाला सवाल, सवाल को गौर से सुनिएगा और मेरे साथ साथ सॉल्व करने की कोशिश करें।
Carrier. Carrier. Oh, if this is, I think that whenever it, you know what? Never mind. Not so true. Let's try. So both of these they are crossing out with XY. Wait, what? What's the seating arrangement? I have no idea about the seating arrangement.
मैं आपको बताऊंगा कि इसको सॉल्व कैसे करना है, आराम से देखिए।
Oh, this is a contemporaneous meeting. I did not know this that. CX.
मैंने यहां कहा विद द हेल्प ऑफ गिवन पेडिग्री फाइंड आउट द प्रोबेबिलिटी ऑफ द बर्थ ऑफ अ चाइल्ड, ऐसा बच्चा जिसमें कोई डिजीज ना हो और कैरियर हो, ऐसा
1 in 4 chance. Is it D? Sorry, A.
कैरियर फीमेल है, तो इसका मतलब ये एक्स लिंक्ड रिसेसिव बीमारी है ना, गौर से देखो।
Yeah. 1 minute. No disease and being a carrier. That is 1 by 4. I'm going for A actually. I'm not sure.
कैरियर फीमेल है, इसको भी मेल और फीमेल एक्स लिंक्ड रिसेसिव बीमारी है ना, तो कैरियर है, अगर बीमारी हो मेल का बीमारी है, क्रोमो एक अगर कुछ जीनोटाइप छोटा एक्स कैरियर की छोटा नॉट अ आपका क्या है उत्तर के बाय 4 आ या
Yes, 1 by 4.
नहीं, हमें टेक्निकली ये सॉल्व भी नहीं करना था, हमें सिर्फ आपका कैरियर फीमेल देख के।
That's a carrier. Oh, I did not know that. Okay. Wait, what? 1 minute before he wrote
अगर
that.
कैरियर फीमेल दे रखी है तो यहाँ पे एक्स कैपिटल सी और छोटा सी के अलावा कुछ नहीं हो सकता हमसे।
Okay, so now I got it. So that's what you mean. Oh.
That means this is excellent recessive, right? Because you can also see that it is getting skipped. So yeah, excellent recessive. To this, we come to the end of biology. We completed this at 6 o'clock. Wow, that was a great speed run. Not speed run. That was a great journey. I see. I don't know why I'm keeping my book here. Let me just check. Whew, let's go. Okay, now we have less time for other things like drawing the diagram. Okay, any other thing I need to do? Okay, so 1 minute, guys. Man, I'm kind of tired about this, you know. Okay. Wait, what should I write? How do I reply? Yes. Okay. Okay, no problem. Take your time and don't worry. I've been studying my tags. Think about it. Okay. Mm, perfect. questions. Okay. Wait. Okay. So what is this going on, bro? Yo, what are these chats, bro? Oh my god. How many messages? One unread message. But it says there are many of them. What is this? Yo, what the? Bro, what is this? It's like a person shooting and then he just scored a goal. Like, that was so random. Who even took that shot? Yo, what is this? Yo, the chats are crazy, bro. Assalamu
Assalamu alaikum, मेरे बेटे हिशाम अब्दुल रहमान. How are you, my son?
Alhamdulillah, अच्छा.
स्कूल कैसी थी बेटा?
स्कूल नहीं था आज. मैं नहीं गया.
स्कूल अच्छी थी बोलो.
अच्छी थी.
मेरे बेटू स्कूल बोलो.
स्कूल अच्छा था.
बस. What are you eating? What is it?
कुछ न्यूटेला के साथ ब्रेड स्टिक्स.
Okay. पानी पियो अच्छा करो. हिबा बेटी इधर आओ. हिबा बेटी come here.
Okay. I don't know whether you guys saw that. Sorry.
हिबा इधर आओ जी. तो स्कूल ना एग्जाम
Hmm?
तो फर्स्ट एग्जाम को सारा बेटा
In bio.
ओके बेस्ट ऑफ लक
Oh, thank you very much. Let's go, guys. We are doing great. What is this? Let's start off with the neat questions with each chapter, and then let's do this one. 1 minute. तुम्हें लेना है तो ले लो पूरा खा लो
पूरा
वो हो रहा नहीं पता नहीं क्यों बोल को खाओ Okay, this is okay. कौन ना जाको बोलो उनको You need to try hybrid plant. What does that mean? Three phosphatized linkage. Okay. So where should I stop doing this because I need to know the topic. Practicing questions, guys. Questions. Don't worry. This is going great.
Tin. Chromatin is further coiled and condensed at which phase? Prophase, anaphase, telophase, and metaphase. Chromatin is further coiled and condensed at which phase? I will send it away. Sooner or later we have to start maths work, bro. Sadly, I don't know when I will get time for biology diagrams, bro. I need to give that time too. Especially for ovary. Ovary looks really crazy, bro. Some people literally told us to draw that structure. So we need to know how to draw it. Okay, this is general knowledge, right? Prophase. That was this is 11th biology. Man, wait. What is this? What? Helical structure of protein stabilized by. I would go for peptide bonds. It's hydrogen bonds. Okay. Learning a lot of new things. Okay. This is not there in the textbook, I'm guaranteeing. Which of the following pairs is incorrectly matched? So, metaphase, yeah. This is the one. What this is. Ouch. Yo, what? Yo, what is this? Chargaff's rule, okay, sensible. The Le Chatelier's principle. This is literally in chemistry equilibrium, 11th standard chapter. Coulomb's law is literally physics chapter. Van 't Hoff's rule. Okay, I think it could be Van 't Hoff factor, but yo, Or for Chargaff's rule. That's for sure. 16, 1. That was a bit too, you know, I would say too obvious, yeah. 45%, okay. Ouch. Where's eraser? I need eraser. Yeah. मगरिब क्या था हो गया बा? Huh? Okay, so it's actually 27.5. But how is that making sense? Okay, wait, it's in percentage. So, okay. Maybe, yes, that's possible. Nope, that's true. That's false. That's false. One. Cytosine is not a one, bro. One. It's one. 21st. I'll be shocked if 21st is not one. Come on. I've been working really hard for this. Anyways, I wanted to know 18th. It's 4. Yep. 21 is 1. 21. Yeah. 21 4. Yo. Stop. Bro. Oh, nah. What? Nah. Stop the cap. Come on. How's it 4? Let me just check. Wait, 21st, right? Oh, does not give the topic wise question. Nah. Nah, how is it 4? Come on. You're 5. That doesn't make any sense. Come on. Bit of DNA helix. Okay, that's wrong too. How is that possible? Nah, I'm doubtful on the first 21st one, man. You know what? Let's send it to ChatGPT. I know I'm being too AI dependent, but there's something wrong with the book. Wait, I forgot how to send the photo. Photo. Yeah. To- Yeah, what? Oh, I'm stupid. Next time I should. Man, my. Why do I do this? It says which one is false. Nice, nice question. Man. Okay. So what is this master following telling us? It's 2.2.
منظرين إنا نحن نزلنا وإنا له لحافظون ولقد أرسلنا من قبلك في شيع الأولين ما يأتيهم إلا كانوا يستهزئون كذلك نسلكه مجرمين يؤمنون به وقد خلت سنة الأولين ولو فتحنا عليهم بابا من السماء يعرجون لقالوا إنما سكرت أبصارنا بل نحن قوم مسحورون ولقد جعلنا في السماء بروجا وزيناها للناظرين من شيطان رجيم والسمع فأتبعه شهاب مبين والأرض مددناها وألقينا فيها رواسي وأنبتنا فيها شيء موزون وجعلنا لكم معايش ومن لستم له شيء إلا عند خزائن أرسلنا الرياح لواقح فأنزلنا من السماء فأسقيناكم ماء ثم إنا لنحن نحيي ونميت ونحن الوارثون ولقد علمنا المسلمين تأخيرين وإن ربك هو يحشرهم إنه حكيم عليم ولقد خلقنا الإنسان من صلصال من حمأ خلقناه من قبل نار السماء سموم وإذ قال ربك للملائكة إني خالق بشرا من وإن بقدر منا للسمون
1 minute, guys. I'll pray and come back.
وإذ قال ربك للملائكة لا تحزن عليهم واخفض جناحك للمؤمنين وقل إني أنزلنا على المقتسمين الذين جعلوا القرآن عضين فوربك لنسألنهم أجمعين عما كانوا يعملون فاصدع بما تؤمر وأعرض عن المشركين إنا كفيك المستهزئين الذين يجعلون مع الله إلها آخر فسوف يعلمون ولقد نعلم أنك يضيق صدرك بما يقولون فسبح بحمد ربك وكن من الساجدين ويأتيك اليقين होगा
Oh yeah, I was just charging it up. That's it. I'm doing maths assignment right now because this is very important. Um, this is the only thing that can stresses me out. I can do the rest work nicely and easily and in a fun way, but this thing is gonna stress me out. So I have to do it as fast as possible, guys. So let me do it this fast, and I have to lock in very nicely. My bad. Oh, oh, I made a small mistake. I by 4 plus x by 2.
Practice your paper.